prestigious event marked by the gracious presence of our honorable chief guest, guest of honor, and the insightful speeches of the experts who set the tone for innovation and collaboration. The signing of MOU between Nirmala College of Pharmacy and Helwan University, along with the souvenir release, added prestige to the inaugural session. The academic sessions were nothing short of extraordinary. We had the privilege to hear from the esteemed speakers like Professor Dr. Mohammad Dikhil, Professor Sisir Nandi, Professor Asmita, Professor Amit Roy, Dr. Janes, and many more, and who enlightened us on molecular parasitology, pharmaceutical education, and computational drug design. The enthusiastic participation in the poster and oral presentations further demonstrated the collective dedication to advancing research. We also witnessed cultural performances in the evening that added vibrancy and joy to the day, highlighting the spirit of our diverse talents. Today, it promises to be just as enriching with a lineup of inspiring sessions by the keynote speakers. We have here today with us Professor Nagaraju sir, Dr. Srinivas Reddy Bonam, Dr. Srinivas Pentiala, Dr. Jitendra Patil, and we have the young researchers' presentations lined up, along with the online lectures by Dr. Nebosa, Dr. Raghavendra, and Dr. Uttam Prasad. So these are going to be a very entertaining and enlightening presentations for all of you. I hope you'll make full use of it. So let's begin day 2 of the symposium on this energetic note. Once again, a very hearty welcome to every one of you. Please give a big round of applause to all the speakers over here today.
Once again, a very warm welcome to all the dignitaries, eminent speakers, faculty, and student delegates. Hearty welcome to the second day of International Symposium. Without any delay, we are going to start on day 2 scientific sessions. Before that, I'm going to introduce the speaker, eminent speaker of this session. Today, the eminent speaker of this session, Professor Dr. R. Nagaraju sir, Institute of Pharmaceutical Technology, Padmavati Mahila Viswavidyalayam, Tirupati, and Dr. V. Satish Kumar is going to be act as a chairperson, and Dr. Y. Ratna Sindhu ma'am acted as a co-chairperson for this scientific session. So I would like to introduce co-chairperson of this session, Ratna Sindhu ma'am, presently working as Associate Professor and HOD of Department of Pharmaceutical Regulatory Affairs and NAC Coordinator. Madam has completed PhD at Apex University, M.Pharm at Hindu College of Pharmacy, affiliated to Acharya Nagarjuna University, B.Pharm at ASN College of Pharmacy, Tenali. So Madam has some of the achievements like 3 research papers and 25 review papers. Hearty welcome, Madam, to this scientific session. We are going to invite onto the dais and introduce your chairperson for this scientific session. Ladies and gentlemen, give her a big round of applause.
A warm welcome to all of you to the International Symposium. Respected dignitaries on the dais and off the dais, and eminent speakers, and respected faculty members and students, a very good morning to all of you. Myself, Dr. Y. Ratna Sindhu, working as a professor and HOD, Department of Pharmaceutical Regulatory Affairs in Chennupati Indo-American School of Pharmacy. I take a pleasure to introduce a chairperson of this session, V. Satish Kumar Garu. Sir is working as a Associate Professor and HOD in the Department of Pharmacy Practice. Sir completed his PharmD in the Nirmala College of Pharmacy and completed PhD in ANU. And sir qualified as a RSET. He secured 19th rank. And sir having 21 publications and 5 research articles. And sir achieved many international awards, and sir achieved excellent young researchers in pharmacy practice department. And sir achieved not only this and many more. By this, I conclude this with the chairperson. Welcome, sir. I welcome chairperson onto the dais to introduce the eminent speakers.
Thank you, madam. It is my privilege to introduce today's our keynote speaker, legendary person, Dr. Professor R. Nagaraj sir. He is the Institute of Pharmaceutical Technology, Sri Padmavati Mahila Viswavidyalaya professor and doctor. Sir was having 20 years of research and 33 years of teaching experience. Sir also ranked as a top 5 scientist in Sri Padmavati Mahila Viswavidyalaya. And sir also guided 10 PhD and awarded, and although is pursuing 8 peoples and M.Pharmacy was 49. Sir was published around greater than 85, and also sir has received so many awards, and also sir delivered at greater than 150 of lectures, scientific sessions. Sir also receives around greater than 2 crores 37 lakhs from different DST and UGC sponsored and CSA sponsored. And also, sir also awarded with 2 patents in their dental implants. Sir also working as R&D consultant to various organizations and member of governing council of few education institutes, and also editorial board for few reputed journals. And also sir received Best Pharmacy Teacher Award by Association of Pharmaceutical Teachers of India, and also sir received State Best Teacher Award in 2017 by Government of Andhra Pradesh. Sir received Pharmaceutical Scientist of Year Award 2017 by Andhra Pradesh Council for Science and Technology, Government of AP. Sir also received Best Teaching Pharmacist Award 2017 by IPA, Indian Pharmaceutical Association, Tirupati Local Branch. Sir also awarded as National Level Pharmacist Award 2017 in the category of Scientific and Innovative Research Award 2017 by Seaman Drug Deliverers Association and the Andhra Pradesh State of Pharmacy Management Association. And also sir Achiever of the Year 2002 Award by Avopa Bangalore. Thank you, sir.
At the outset, I thank the management, the principal of this Nirmala Institute of Nirmala College of Pharmacy for selecting me as one of the keynote speakers in this prestigious conference. And also thank the funding authorities also. And also I have met a wonderful team of resource persons, very rare to get such type of people in one platform. And all that, the most wonderful audience. Majority seem to be only girls. Yes, and very silent. So keep the silence and try to listen more and have the interaction with the speakers in case it is what do you call it, have any doubts. I just wanted to start, I should say one more thing here. Whatever the photograph shown in the first slide reminds me, it was 10 years back, how I was looking like that 10 years back. So now I look like this. Thank you for the organizers reminding my younger days. Yeah, I just wanted to start my talk by just giving just 3 minutes video for all of you. Kindly watch this video. This is about our university. is the self-explanatory. This is the administrative block. This is our guest house. Physical science department. Yeah, this is our pharmacy department. Animal house, what I am seeing on the left side. Artificial intelligence lab. Law department. Our prestigious lab, Curie laboratory we call, all sophisticated instruments we have. This is biotechnology incubation facility. Now the vice chancellor is changed. This is the old photograph. Even the registrar is changed. Thank you for your patient watching. I would like to know how many of people have visited Sri Padmavati Mahila Vishwavidyalaya in person. Can you raise the hands? I know, sir, you have visited. Students? Not visited, yeah. Yeah, we have my own students here, they have done it, fine. So this is for those people, what is the university, and can go to the website, and if you are planning the higher studies and incubation and innovation and anything related to biotechnology and also technology business incubation, you are welcome to us. Okay. Sir, can I have my presentation? My presentation. can I have the slide changer? I had to do or? Oh, slide changer is here. Idena? Huh? Right. Come back. I am going to talk to you about this particular topic for another 30 minutes or so. I just wanted to briefly put a focus on the only the basic part and applied part. I am not going to talk about any technology part, right? As the proverb says in Telugu, ఏ దేశం ఎగినా ఎందుకు కాలిడినా పొగడరా పొగడరా నీ తల్లి భారతిని. So I believe that in the true spirit of it. So I am very fortunate enough in mid-career of mine, after having 19 years, put up 19, 20 years of experience in a private setup, I have got the opportunity to work in this great institution, for which I am almighty and thankful Almighty God, Balaji, and the opportunity given to me. I hope I am enjoying it, and this is a great opportunity for me. Right. So the topic what I have chosen to discuss with you is going to be an emerging trends in novel drug delivery research. And my focus is going to be, as your theme is going to be novel drug delivery systems, nanoparticle systems. I am going to focus more on nanoparticles and leaving out other novel drug delivery systems. And I am very proud to say that pharmacy department established 1983, our university established in 1993 and pharmacy established in 1992. And we are consistently ranked in NIRF ranking, and this year we got 60. One is, of course, Andhra University. So that is number one. So this year we have submitted the data. We have to see what is going to be the rank. I have divided my presentation into these following headings very briefly, as we started little late. So I just wanted to condense some of it, where I am going to talk about what happened. Where is the pointer? Pointer? Light. Pointer. Right, I am going to talk a little bit of introduction and advantages of novel drug delivery research and why the novel drug delivery systems basically, and coming to some brief introduction about the nanoparticular drug delivery systems. And I have taken liposomes and neosomes as the basic case study for that, so that the level of audience here will be able to understand better. I didn't take any advanced systems, and also the challenges and the future. And finally, what are the research, what is happening in SPMVV in the area of nanodrug delivery research, and also, of course, the final conclusion. All of us know the novel drug delivery systems are not conventional. That's why they are called novel. So why the novel drug delivery systems are the need is going to be because we require to improve the drug potency, control of drug release, sustain therapeutic effect, and then provide greater safety, target a drug specifically to a desired tissue. So these are all some set goals and objectives for which the whole research in the novel drug delivery systems is based on. And then, as per the definitions of various textbooks is given, novel drug delivery systems are approaches, formulations, technologies, and systems for transporting a drug in the body as needed to safely achieve its desired therapeutic effect. This is the book definition of novel drug delivery systems. So the main aim of these novel drug delivery systems goes into getting a delivery system which is better than existing in a non-conventional way. So compared to conventional way, how these nanodrug delivery systems are say based on nanoparticles, how they are going to be advantages. They have more solubility and bioavailability and protection from the toxicity and enhancement of the pharmacological activity is possible. Of course, enhancement of this. So, it prime most important one. The question comes to the young researchers, those who are sitting in the audience, researchers I call it, because the pharmacy is more the blend of research and education. The new curriculum in the pharmacy, we include, the Pharmacy Council included practice school and then project work. And of course, M-Pharm we have completely project work. All these are important, are essential for the young minds to put you in the research field. So why these novel drug delivery systems are going to be the thrust area in the research? It is going to be because you all of you know very well that development of a new drug molecule is very expensive and almost touching $120 million. And then, of course, the time consuming, it is almost 10 years. Even after 10 years, is it guaranteed to get a new drug? No. That's why in India, till now, we are not able to get any new drug in the market at any particular stage of it. In the inventions, we are not able to bring it to the market even today. In August of last year, there is one molecule has come out in the Indian origin. So I forgot the name of that. Except that for last so many years, we don't have any drug molecule which is made in India and for the world. So therefore, this a decade or more is a very time consuming. Probably the result is not sure. Usually 10 to 15 years, and we don't know whether you are going to get a molecule or not. So that is what very uncertainty is there. So therefore, the focus has gone on to what is the alternate ways to improve the therapeutic efficacy. How we will be able to do a better treatment and therapeutic response and so on. So therefore, to improve the efficacy of the old drugs, in the focus of individualizing the drug therapy, dose titration, therapeutic drug monitoring, and existing drug molecules can get a new life. So with this focal theme, the nano drug research is being taken forwarded, and how we are successful many occasions in proving all these characteristic features in the system. And then this is a history, the decline of introduction of new chemical entities since late 1950s, and we don't have a latest antibiotics even today. And then only very few companies are conducting pharmaceutical research, and pharmaceutical giants in India are closing their research institutions and shifting to generic drug. And then erosion of what I should say, effective patent life. So therefore, the novel drug delivery systems are going to be based on nanotechnology, is going to be taken a very significant role. And also, when you see the projected market of these particular drugs is around 40 to 50% growth is there in the US marketed products, in these products. So amongst the popular nano drug delivery systems, transdermal and transmucosal are the most defining one, nasal and buccal aerosol systems, and then drug impregnated lozenges. And then we also see in controlled release technologies where one such thing which is working mostly is the nanoparticles. This last line I have put, that encapsulated shells, oral subgels, ionophoretic devices, and variety of programmable and implanted drug delivery devices, and then nano device, nanomedicine, nanoparticles, and nanosomes. This is what the theme of your conference. So I am leaving out the other aspects of it and directly going into nanotechnology, how it is going to be is useful to us. So what is nano? The now whole pharma research in pharmaceutical technology is going into around nano. Whichever the journals you see, we see the preference is given for in advanced technologies like nanoparticle formulations. So here the nano 10 to the power of 9, very small, that's why dwarf. We see tera, giga, mega, kiloi, milli, micros. Now we see in the computer terminologies, we have the terabyte. Gigabytes are gone, now terabytes are there. So whereas we are going the reverse, we wanted to start with nano, very small particles. So the fundamental question is the very small particles, okay, why? Why it is going to be? By now you understand particle size reduction will alter some very important parameters in the drug. Those are the creation of the functional materials or devices at the systems, control of the matter in the nanoscale range, that is 1 to 100 nanometer length scale, and these are the very important thing I wanted to mention to you. Exploitation of novel properties and phenomena developed at that scale. So when we try to convert the larger particles in the nanoparticle range, we have some properties in the drug molecule has been altered. So let us see what are those properties. The properties are physical properties, melting point and such things, chemical properties, say reactivity, electrical properties, say for example conductivity, mechanical properties and optical properties. So these properties has been changed when we convert normal drug particle into nanoparticle. So we are going to exploit these properties in advantage of ours so that the therapeutic efficacy, solubility, bioavailability, targetability and all such things can be addressed by simple reducing the particle into a 10 to the power of -9. So that is only you are going to do. So therefore, we get by nanotechnology, nanoparticle formation, a high surface area to volume ratio. When you see, of course, this is a very old picture, predicted in 2001, you can imagine. So there is an industrial revolution, all of you know, second information revolution. Now we are in information revolution. Gone are the things, textile revolution in 18th century, rail and road revolution in 19th century, then automobile innovations in 20th century, and then computer revolution in 20th century started 1939 itself. But in India, we got it very late. So it is going to expected to continue. They expected in 2001 up to 2025. But even beyond that, it is going to go. We are seeing it, the new vistas where the computer science has been going ahead. Then the nanotechnology started somewhere in 1997. It is going to rule the whole world in all the fields people expected in 2001, 2081. And I wanted to update this, even the latest predictions I could not find. So therefore, I am using the old information, but it's very much relevant how the nanotechnology revolution is happening around us. And then this is also an old slide. Let me skip this. Yeah. So which are the areas where the nanotechnology can be applied? So when you see the application of nanotechnology, the first one, medical and pharmaceuticals. That's what we are interested. Chemicals, automotive industry, energy production and distribution, aerospace technology, environment, defense, textiles, agriculture, information technology. This is another area where we are also interested to amalgamate the artificial intelligence in drug delivery and discovery, where you require revolutions which are going to be manifested in this area. Then material science, telecom industry, where all of us are the Yeah, now we are getting 5G very shortly, almost, and the world is, the China has gone ahead of us. I hope, subject to correction, they're already given the 6G and 7G, and I don't know about America and other countries. I don't know which Gs are going on there, but all these are telecom revolutions which one way or other connected to this field. When you talk about the, in particular about the nanoparticles, which already I have given in 1 or 2 slides, I thought I can repeat it. Repetition does not do any harm. So the methods of preparations are simple and easy to scale up. So any industry wanted to adopt this technology, the industry look into a simple technology. Whereas in the laboratory, we may be successful in bringing out some in innovations which sometimes are not possible in the actual production scale. Whereas nanoparticles have been adaptable, they are simple and easy to scale up. That's why they are popular. And then they are stable and easily freeze dried. And we can also look into the tissue targeting, successful tissue targeting like antibiotics, cytostatistics, and peptides, proteins, nucleic acids, and etc. We see the anti-cancer drugs, the nanotechnology and targeting is going to be very much innovative, and things are coming up in this field, and few of them are in the pipeline, and anytime it is going to hit the market. And then production of the drug, when you do nanotechnology and make it again, stable against the chemical and enzymatic degradation, which prolongs the shelf life of the drug. And we can definitely reduce the side effects, some of the very toxic drugs, because we are going to go for nano, and the dose is going to be very, very less. And then this is a slide I repeated again from the previous one. The nano sizing, increase the targetability, increase the solubility and dissolution. Where we work most on this in the laboratory, in our laboratory, to use this technology for these purposes. That's why I repeated this slide. And we do targetability increasing, we work improvement of the solubility and dissolution, thereby the drug bioavailability is improved tremendously, and of course the adhesion of the tissues are going to be very much better than the molecules or the larger particles. And we also see the delivery of peptides, the large molecules such as peptides, proteins, oligonucleotides, and plasmids are improved using nanotechnology through mucosal delivery. So we have great advantage of simply reduction of the particle size to a 10 to the power of -9, except that we are not doing anything. So therefore, we see a greatest advantages in these particular areas. So why it is relevant to pharmacy? See, anything which is useful to the society, taken up by the industry, are only the successful projects. You make 100 inventions which is only go in the form of a book or lying on the cupboard of any people, either patenting, whatsoever it is going to be. If it is not useful for the society, there is no point of doing so much of money. That's why our Honorable Prime Minister always says many a times, do research, focus research, which is going to be useful at the end to the society. And with the latest RUSA, Rashtriya Uchatar Sarveksha Abhiyan, where the research grants are given to various organizations, both in the first phase, they are given to government colleges and universities. They are planning to give for research organizations. They are planning to give for private colleges. That's why the NAC and accreditations and all NRF are all going on with them. So the next phase, even the private colleges will get this funding. The one condition is that you have to do a multidisciplinary project. So that was the first and foremost condition they have laid down. So, and also the project should be at the end useful for the society. And if not only that, the industry should be able to make it in a large scale. So that is going to be a very game changer, not the game changer of Ram Charan. Yes, this is another game changer for us where how we are going to put ourselves in the competition of the drug delivery research. And then we also see the new life for existing drug molecules. By doing this, we'll be able to improve the therapeutic drug monitoring part of it, and also making a therapeutic response more and more effective, reaching the people. And then it is going to be increase the commercial opportunity by distinguishing the drug from competitive threats posed by Me Too drugs. That means in the place of a particular drug, a new drug has come for the same purpose. So that competition can be avoided when you got the nanoparticle you are going to go. And we see nano carriers allowing the branded drugs to be rescued from abyss of generic competition. That's called as resurrection of the drug. When you see the generic popularity, the government of India, in association with the Pharmacy Council of India, has brought out many schemes, and they encourage the generic drug stores. And Pharmacy Council of India is going to make mandatory that each and every pharmacy college should start one generic medical store. So that rule is there, but it is not implemented. Probably that may come at any time. And Prime Minister's fund is supporting the generic drug stores, where the generic drugs are pushed into the thing, and many of the people don't understand the generic and the branded one, and we need to educate the people about this efficacy of that. Therefore, the burden of the medical treatment is going to be so much relieved for the patients when you go for the, generic medicines. So in the nano devices, the work which has been resulted into the final acceptability, I just wanted to give few success stories here. That is the development of 1 dose of a day of ciprofloxacin. This is by nanotechnology. Tumor-targeted Taxol delivery using nanoparticles is in phase 2 clinical trials. Improved ophthalmic delivery of formulation using smart hydrogel nanoparticles. And then that is also under work. And oral insulin formulation using nanoparticle carriers. A worldwide lot of research is going on this oral insulin part of it based on nanotechnology. And then we see the most success story is the liposome-based amphotericin B formulation, where it is really helped us during the COVID time, and we have seen use for the treatment of fungal infections with the black fungus infection, which has been the side effect of these things. So these are some success stories where it is already happening. So you must be wondering whether is it okay. As I said, I am not going to touch upon the technology part of it. The technology part in any textbook, you will get it. So I just wanted to give you some insight about some of the basic understanding of nanotechnology. So whether is it okay to make only the particles to reduce the nano size range, or we should use any carriers for that. So this slide will answer that question. We can use the vesicles, we can use the multifunctional dendritic polymers, we can use the nanospheres, nanocapsules, liquid crystalline crystals, and micellase. So I am happy to inform we are working in our laboratory in our own way using all these technologies. And then especially micellase and liquid crystals, last time one of my students has done a project work in MFOM. She got in one of the conference a best paper award, where the liquid crystal, really we have made some breakthrough, and I am preparing based on the information, I am preparing a project based on liquid crystal nanotechnology for funding of the DST and RUSA and all. So probably 30 to 40 lakh project I am making it. Therefore, we have got already the fundamental results. 2-3 students worked on that, and it is very promising. Only thing is little bit of fine tuning is required to go at it. So what I wanted to tell the young minds here is the research is only the way you can make the difference in you. And you think only reading, studying a B.Pharm degree and getting an M.Pharm degree will get you a job. Definitely you may get some job, but that is not important. You should see a decent job to suit your caliber. So that is very, very important. Not, I don't like B.Pharm students going opening a medical shop. Yeah, for that why do you study 4 years? 2 years diploma is enough. I don't endorse a pharm.D person going for medical scribing or say pharmacovigilance. And then for that, why do you require to study 6 years? You know, people say the scope is only that in India. So therefore, you need to identify, get a suitable position, make yourself a competent person, and of course, the government and other ecosystems to support you, to uplift you. So that is what. So that's why our Honorable Chief Minister always says the skill development. The skilling is very, very important. So we are going to go for some basic idea and how we are going to hone our skills if you have. And if you don't have the skills, how to acquire the skills. So those are all the things are going to be very, very important. So what I wanted to tell from this is we are working in all these areas, either at PG level or PhD level, and we are getting a promising results, and also some projects we are converting into the what you call funding authorities, and maybe if it is approved, maybe 3 to 4 years, probably we may bring out some breakthrough findings through this. And then I just wanted to share this slide. It is, even though this is old, I verified there is no much progress in this. They are still continuing. These are all the phase 1 and 2, the vincristine, and then paclitaxel, cisplatin, and lortocan, where the basic novel drug delivery systems with the nanotechnology has gone to this particular level. I didn't get any information about the second drug, the paclitaxel. I wanted to know what happens, the next progress. I could not get anything. I didn't update this information, but this is way the success stories are already there in the literature. So what are various ways or the dosage forms which are based on the nanotechnology, nanoparticles? So the simple liposomes and neosomes come under the nano range. We call them as nano drug delivery research. And then microemulsions, nano suspensions, solid lipid nanoparticles, and then simple nanoparticles, micelles, nanocrystals, microspheres, and so on. So these are all some of the drug delivery systems based on nanotechnology and nanoparticulate systems. So I am going to quickly to understand, I came to know that principal sir has told me that even some diploma students are in the audience. So for them to understand, I took a very simple conventional liposomes and neosomes for 5 minutes, and then jump into my final conclusions with my experience in our laboratory, what we have done. These are the liposome eccentric rings, and where we have bilayer lipid layer, and then these are all the inventions, what is happening in the liposomal drug delivery. Yes, the most noted one is amphotericin B and the doxorubicin, and then the CDDA. There is a very promising fields where the liposomal drug delivery systems most of it are successful. And the medical applications are listed here, and where immunosuppressant, anti-nausea, topical estrogen therapy, for appetite stimulant, and so on, and antimicrobial. So in these areas, we see the liposomal drug delivery systems are still working. And then some market, this is the little latest information, 2014. These are also marketed preparations which are available for the purposes indicated. Next is neosomes, the twin brother of liposome. Yes, in the liposome, we have lecithin and phospholipid. In the neosome, we have only surfactant, except everything is same. So many a times, the field appears to be very good. Many a times, you get motivated by reading some articles and seeing some people. But actually, when you try to do something, any task, not only the research, you have the challenges ahead. So the challenges ahead are all known challenges or unknown challenges. In the research, always we have the challenges which are not known. We don't expect. A simple expectation also we don't mind. So unless until you work in the laboratory and see what is going on, then only we are. So we are not supposed to be frightened or what I should say, get disregarded if at all you have some challenges which you are have to face it. That's why I didn't call as a problem. The problem is different, challenge is different. Problem is a problem which is going to be a different meaning that of a challenge. So the challenge is going to be the laboratory, you are successful, to the pilot scale, you are also successful, full scale commercialization, this is a big challenge. So when you go for technology transfer, people will work it out on whether is it going to be possible to put it to the market. That is still remains challenge with all these basic nanoparticle drug delivery systems. The next important challenge is, the bridge the gap between the science involved and engineering required and the scale up. So in the universities and academic institutions and research organizations, both central and government organizations, we can develop the science behind it. And we don't do the economics part of it because somebody is giving the money, somebody we are going to do, we are going to establish the concept. But the real challenge is, what is going to be translation of this new science, what is being generated into a reality. That reality changes in the scale up. So always many successful stories in the laboratory will not come in the light of the day because of the problems of the scale up and the, what I should say, the success in the industry. And then many a times it is a high cost. So any people, especially I am not degrading my own country people, the India light, we want everything the best, but it's cheap. The most example one is the China phone. Remember when mobile phones are invented, the China phones are so popular. And you used to blast your ears and all that, they are blasting and all. So slowly, people have moved to consciously to the quality now. So now the present generation, for last 5 years I'm seeing, people are not only conscious about the economics, but also conscious about the quality. That's why wherever you go, the quality is going to be taken as a first priority. So, but we should be able to give the quality product at, I will not say at a cheap price, but at an affordable price. Then only the product is going to be successful. There is no point, say for a drug A normal drug useful and is costing around 100 rupees, and when you keep 1 lakh rupees, the next invention, so who will buy? We will not be able to do it. So therefore, the cost is going to be very much, and the size and composition of the nanoparticle formulation is going to be a little challenging to handle at very high levels. I know audience are little saturated. I'm coming to the last few slides. That will be interesting. So of course, I'll skip this. What is the future? This is what many a times the students in the mind they will have. You joined pharmacy course, what is the future of it? The future of it, you have a very bright future. That's what we say, always. Yeah, the people who are going to explore will have always, as I was talking to one of my resource person, you should come out yesterday, you should come out of the comfort zone. So Dr. Srinivas will talk to you about how he struggled to achieve this position. You should listen to him. Sir, please tell briefly about your struggle. How did you achieve this position in the, in central government organization, that compared to subject what you teach, that will be more motivating. That's what I feel. Maybe 1 or 2 minutes you spend about it. How did you start? How did you get this? How did you make the reality and out of it? So it is going to be your hard work, and working out of the comfort zone takes you to the different levels to achieve your ambitions. So here, the most fascinating areas of the research, that's why my current area of research is nanoparticle nanotechnology, and filing, it is not filling, filing of almost 1,500 patents. Even I have 5 patents filed in this area, waiting for the response. And then consistent uniformity and drug loading release capacity is going to be achieved by properly manipulating the procedural work. And also, the gold nanoparticles appear to be well absorbed the tumor tissues and making tumor susceptible to radiation. But the gold price is very high, so most of our Indian homes, so we need to take out some gold out of it, then only we make a project out of it. Otherwise, whenever we have very little money, sir, in Indian tradition, any good occasion is there, we buy gold, yeah, and keep it in the locker, safely for future generation. Yeah, otherwise, wear it, yeah, men, women will wear big ones. But anyway, that is not a bad idea, that is an investment where for the future. So, but the gold nanoparticles, one of our scientists, a biotechnology colleague in Mahila University, she is working on the gold. And I said to her, how you are getting money? She gets some grant, that's why she is working on gold. It's going to be even now very promising to work on it, but because it's very expensive, the research is very slow in this particular area. But there is a lot of future in this. It's not going. Nano robots are there, nano sensors are there, nano medicines are there, and natural biomaterials or biodegradable compatible materials and renewable low toxicity materials should be explored, so that you have these areas which can be worked out. And then these are all some of the, after going to the literature, we see these are some potential drugs which are available, which are unexplored, not much explored, like berberine, curcumin, ellagic acid, rivacetal, and quercetin, and the nanoparticle, the formulation of these drugs has little low explored. We can take this as a challenge and look into the further one. And integration of the therapy and diagnosis by the cancer disease model is also a good future. Yeah, I'll skip this part of it because it's already, we are running late. Yes, so this is one case study of floating microballoons, which we have done in our laboratory and received much applause, and a good publication has made out of it. I thought I will share it to you. Yeah, little animation. Yeah, you like it? This is the approaches of drug delivery systems in the gastric systems. One is floating systems, mucoadhesive, swelling and expanding devices, high density system. These are the approaches. We have worked on all 4 approaches, and this approach what we have worked is on based on swelling systems. We have worked on this. Anyway, I don't explain anything, I just give it the bioadhesive systems. Swellable systems. This is the drug targeting using microballoons to the GI tract, I mean especially the stomach. This is the high density systems. This is the various types of floating systems, what has been there. And these are all the types of floating systems. The hollow spheres or hollow micro balloons are the things where we explored in this study. These are the methods of preparation. We use riboflavin as the drug of choice here because it has got the therapeutic window in the later distal part of the, proximal part of the intestine. That's why I use this. This is what the concept is. Whatever you see the absorption window where the drug vitamin B12 is supposed to absorb, because of conventional drug passes this absorption window very quickly, we will not be able to see the absorption of this particular drug in the conventional dosage form very much. But however, when you make the gastroretentive drug delivery systems preparation based on micro nano technology, we see the drug is going to remain in the stomach for a longer time and slowly releases the drug as you see in the animation. And it passes the the proximal part of the intestine and the drug is absorbed very quickly. So this is how any plan of the work will be done. Evaluation. This is how the formulation chart is prepared. This is the SEM pictures of these microballoons. I'll spend only 2 minutes in this few slides where I wanted to focus on what type of work we are doing in this NDDS research in our laboratory. I listed the most important ones, successful ones here. So we work on this project, the microballoons, and then she is awarded in the 2019 PhD, and then we got 2 patents out of this project work. And then she is also worked on pulsatile release, and then where we see she got 1 patent. This is, we worked purely on nanosponges and nanoparticles based on the chitosan, and then she completed in 2019, and we applied for 2 patents. 1 patent got approved, but 1 still inquiry is going on, so we need to answer that. And the next one is the hydrogels. She is still doing the project work. Something.
Sorry to interrupt, sir. Please take this.
Okay. Here the Jansi is, I think yesterday she might have come and presented one of his research work here. She worked on some formulation development using nanotechnology based drug delivery systems she is pursuing. And this is what NDDS M-Form research. This paper has got the best publication in one of the international conference. We made liquid crystals. I was talking about the liquid crystals, no? The same thing is this. These are some organogels, chitosan-based nanoparticles, colon-specific delivery, intranasal microemulsions, ocular nanosuspensions, nanoparticulates, scaffolds of various antifungal drugs, nanocrystals, nanosponges, controlled release, delayed release, emulgel. This is actually the emulgel for psoriasis model. This is, we applied for a research funding. This has given a very good results for us, and we are waiting for the approval. If that gets approved, probably we will work further. And then moxifloxacin nanoparticles, and then floating in situ gels. The list is this. So what I wanted to tell message to the student audience and also young researchers is going to be, do the research with passion. And whatever the little facilities you have, you can do wonders. And the basic facilities, if it is created in the laboratory, and then advanced facilities, we can always outsource. So SEM we don't have, but we get the SEM pictures from some other research foundation. So likewise, students also should, I will use the word again, come out of the comfort zone, try to work hard and bring out, show your passion to the research and bring out some project which is going to be novel and talked about. So your guide should be what you call proud enough to present like this. Yeah, to that level you should come. In international conference, I am very confident that these are all my significant contribution in nanoparticle, nanotechnology level where my PhD and PG students have done it. So you should make your teachers proud and the college proud and taking such projects and going ahead. This is my message to all the youngsters. Last slide, last but one slide, one more is there. Yeah, this is what the wake up, those people who are sleeping, seeing your mobiles, please look at this slide once. All of you, your attention please. Look at this picture. What did you see? What do you see? Images nearby and images far. Can any one student who are very confident can take a mic and talk about it? Madam wanted to, you know, you will talk. Yeah, because of the accessibility. One of our resource person is going to add the contribution to this. Yeah, you can tell. Madam, you can tell.
So on the first hand, I could see a person with the instrument on one end and the person with something catching on top of his head. And also I can see two old person also. I can see the person with the instrument as well.
Nice, madam. Only that much you can say?
With
the
a
guitar?
guitar
Yes.
instrument.
And
Playing the guitar.
And then one young lady is coming out of the ear. Can you see that?
Huh.
Yes?
Young lady is also there.
Yeah. Then? So-
Then we can see in between a vase is also there.
Correct. Correct.
And
Right.
a bottle is there in the ear of the young, old person. Old lady, a bottle from the, as earring.
Yes, yes, yes.
Many things are there.
I am
Doctor,
seeing a romantic meeting between a man and a woman.
Correct.
Yeah?
Correct. Yes. Very nice observation. I hope the delegates, both
I absolutely agree with Professor Dikil. It's a romantic meeting. The gents means Asik, he play the guitar and she is enjoying the guitar.
yes,
So it's a very nice picture. Means you open your heart.
yes. I want to, my aim is any students or delegates are sleeping, at least open now and see this and remember the earlier part of it. Yeah? I have one more slide. Yeah. I give the question, find the baby in this. There is a baby there inside the picture.
It's between the picture what we see
Oh,
itself
no,
is
no,
an
no.
answer.
Now teachers know. I want students. A young couple is there on the seashore. There is a baby. Can you find the baby? All of you? How many of you can find the baby? Please raise your hands. Yeah, majority. Those people who have seen the baby, say yes. Very nice. One more time. Yes. Yeah. So I consider all of you have seen this baby. If not, please go to the ophthalmologist. I thank you, all of you, for your patient listening. I hope you have at least understood or say got motivated or say the purpose of coming here and meeting the galaxy of the speakers. It is very important that to make all the speakers here, even on the phone, we will not be able to get them to speak. That includes me also. Don't think, sir, మీరు ప్రొఫెసరే కదా ఏముంది పని మీకు కూర్చోంటారు కదా అనేసి కాదు. You ask your principal how many times he called me. I will answer only after 2 hours. I will say sometime busy at a meeting. I have this, I have that. Only evening I will. So it is all of us are busy in our own routine and other things, and keeping aside everything. Sir, professor has come from Egypt, and you know, he come to first time to India, and people have come from West Bengal, people have come from Hyderabad, people have come from Maharashtra. See, this is what making. I should appreciate the college, principal especially, and sir who is helping him in making all these things happening. Is going to be. It is the I should say again the galaxy of the speakers. You try to interact with them. Yes. How many of you wanted to have an interaction with what? You should decide. Go to our principal, get their email, and converse in the email. Sir, I am very much impressed. Madam, Madam will get angry. Madam, I am very, very much impressed about your lecture and about your this thing. I want to further continue my advice with you. So this is the way you have to, the purpose of coming over here will serve. Otherwise, the Professor Reddy Garu and the management and the whole team of the college is working for last 2 months. I know. 2 months back they asked me the date. And they have come to the railway station in the midnight, 12 o'clock. Why here? What is there for the college? One day news people will forget. But all these things they are doing it for the profession, for the benefit of you. Right? Explore that and have a communication to the speakers, the things what you like or like to be, and extend your conversation with them, take their guidance and get those things. I wanted to read this. I am very proud to be a teacher. I came to the teaching profession by choice. Even though many opportunities were there in after my completion of M.Pharm 1986 and PhD, I took up the teaching profession and I continued because teaching is my passion. Teaching is a profession that creates all other professions. And I'll take here Abdul Kalam's very favorite word, very apt for the students who are having, the inferiority complex. Yes, fear has got 2 meanings. Forget everything and run like us. Tomorrow exam is there. Tomorrow, no problem. I know. Today we'll go to movie, Game Changer undi. Repu exam edo radamle. That is a fear of exam. రూమ్లో కూర్చుంటే రూమ్మేట్ చదవంటాడు ఇంకా ఎందుకు చదవంటాడు ఎందుకు గోలా పోయి సినిమా చూసేస్తే అయిపోతుంది. ఐఎమ్ సారీ, ఐఎమ్ యూజింగ్ ద లోకల్ లాంగ్వేజ్ టు మేక్ దమ్ మోర్ ఇన్వాల్వ్డ్. సో దాట్ ఈస్ ద ఫియర్ ఆఫ్ ఫర్గెట్ ఎవ్రీథింగ్ అండ్ రన్. సో యూ షుడ్ నాట్ బీ ఇన్ దట్. యూ షుడ్ ఫేస్ ఎవ్రీథింగ్ అండ్ రైస్. దట్ షుడ్ బీ ద యాటిట్యూడ్. ఫేస్ ఎవ్రీథింగ్ అండ్ రైస్. సార్ అగైన్ Srinivas Reddy Garu, I am very much your rise in the field. You have come to this level. I become a fan of you on this dais. I am telling you, speak about yourself for few minutes. From such a background and the hard work, he is now the scientist in central government organization. Just see his successful man, how he has done. He belongs to the next category, face everything and rise. That's why he has come out of the comfort zone and success has been there. So remaining, I didn't know what is your things. Probably you also might have your own success stories. I know Dr. Reddy has really struggled very hard for his PhD, and I know I happen to be one of the part of that, and I know his hard work, how, even though little late, is rewarded with a good professional position and all. I can see the whole conference is organized very well. A good leadership in you has shown the conglomerate of all the things, which is going to be very much definitely a very memorable event in the history of Nirmala College of Pharmacy. Yeah, with this, it's already 11 o'clock. I was supposed to complete by 10:30. Yeah, sorry for the delay. I hope I didn't bore you much. If the chair allows, the students can ask any questions, one or two,
Yes, sir. Yes, sir.
or we'll postpone to lunch break.
Thank you, sir. Thank you for valuable presentation, sir. We are also so much inspiration like younger lecturers like us, sir. Thank you, sir. Thank you for a very valuable presentation, sir. Any questions regarding the session? Please, any discussion time. This is. You have any questions? Ask like legendary persons or like persons.
Hello.
Sir, ask
Thank
easy,
you.
Sir, ask easy questions, please.
Hi, yes. Yes, sir. So first, thank you very much, sir. It's our very good opportunity to have very young and energetic speaker, very good friend. I would like to share, yesterday I met with him around 10:30 p.m. in front of the hotel. And my feeling was, I met him first time yesterday night for a moment, and my feeling is, I met him long years back. So this is his personality. So what Dr. Nagaraju is presenting to us, that you must have such kind of personality and presentation for easy access and friendly. Thank you very much, sir. I really pray to God for your long, healthy life. You are the living example.
Another 5 years extension, please give.
Acha. So, can I see the under clinical trial slide? Under clinical trial. Under clinical trial slide.
You have some information? Information on that?
Sir, you know, development of computer science, peak. So, programming, computer-aided drug discovery, computer-aided drug delivery, computer-aided drug design, application of informatics. Nowadays,
Yeah, continue, sir.
clinical trial, last, yeah, keep, back, yeah. Sir, nowadays, computer-aided drug design algorithm can predict the lifespan of the cancer patient. Oncorox, one computer-aided programming has been developed. It is based on incorporation of the patient's profile properties, drug efficacy, etc. Then that software informatic skill can explore the life of the patients. So this is the beauty of research. So, sir, in case of when a patient is suffering from cancer, multi-combination like DNA alkylating agent, DNA intercalators, multi-component tablet formulations are given for the treatment. Does nanoscience can develop any nano formulation incorporating the synthetic component that can increase the patient's life? Is it reported? I don't know, sir. That's why I am asking you. Is it reported?
My first response is, I don't know. Because I have more and more pharmaceutical technologist rather than the therapist. So my area of specialization is developing nanotechnology based products. So remaining part of it, probably I don't have expertise to talk on it. Anybody
Okay,
can add, anybody can add if anybody, sir?
My question is there any nanomedicines, anti-cancer medicines taking synthetic component in nano formulation that can increase the cancer patient life? Is it reported? Is it reported, sir? Okay.
Sir, take the mic.
There are so many drugs like that, nanoparticles, particularly nowadays they are using like even for CAR T cell therapy, you know, like T cells adaptive therapy. Now from drugs to cells we have come up, so there are many drugs, but definitely there is, I mean, we cannot increase the longevity of life, but at least they are increasing like 5 to 6 years. That's why still we are struggling to come up with the new drugs, new formulations.
Okay, sir, this is a great knowledge. Huh?
Thank you.
We have
Thank you.
achieved. And does the development of nano formulation possible taking multi-component?
Yes, yes.
It is possible.
Yes,
Okay, sir.
we can do it.
Okay. Thank you very much, sir. Thank you.
First of all, let me conclude with the 2 sentences. I'm thankful to all of you for your patient listening. More to Dr. Reddy and his team, the management, and of course the support which has been given. To be frank with you, I was little nervous because all resource persons are there. First time I am speaking in front of the elite people who know better than me, or sometimes I was little tensed up. Then I thought, okay man, you have given 136 lectures, this is 137, no problem, you do it in a way. So I consider them as they don't know anything, like how we tell, I was looking only behind, right? So if at all I am too basic, if at all somewhere some conflict of interest, please ignore that. So in the overall, I took it 2 basic things and went ahead. Thank you very much for the opportunity.
Thank you so much, sir, Professor Dr. R. Nagaraju, sir, for coming us to the International Symposium and giving very valuable and knowledgeable session over today. Once again, thank you so much, sir. Ladies and gentlemen, give her a big round of applause. It's time to honor our eminent speaker, Professor Dr. R. Nagaraju, sir. On behalf of this, we'd like to call upon chairperson and co-chairperson onto the dais, please. And I would like to call upon Secretary and Correspondent, Reverend Sister G. Nirmal Jyoti Garu onto the dais. And I would like to call upon Dr. P. Pamul Reddy, sir, Professor and Principal onto the dais. I would like to invite Organizing Secretary and HOD of Pharmaceutics, Roy sir, onto the dais. And also request Nikhil sir onto the stage, please.
Thank you so much, Professor Nagaraju, sir, for your time. Thank you for sharing the current researches on nanotechnology with all the participants and delegates here. Give a big round of applause for the wonderful personality here. We are going to have a collaboration with Professor Nagaraju, sir, in future. You are going to see him now and then in our campus. I request all the speakers here to come on to the dais, please. Ashmita ma'am, Srinivas Penchala sir, Srinivas Reddy sir, Amit sir, Sisi sir, Jitendra Patil sir. It's a pleasure to have such eminent personalities at a time on the stage. You could see here all the eminent speakers who have already shared their knowledge with you and who are about to share. Give a big round of applause for all the wonderful personalities here.
It's time to honor our chairperson of this session, V. Satish Kumar Garu. Please accept our token of gratitude.
It was an eye feast for all of us to see all the bunch of banks who are having lots of knowledge in their minds full. Thank you all the respected speakers for coming up.
It's time to honor our co-chairperson of this session, Dr. Y. Ratna Sindhu Madam.
Thank you to the speaker, chairperson and co-chairperson for all the time and efforts you have put in for the successful completion of this session. Thank you all respected dignitaries. It gives me great pleasure to welcome you all to this much anticipated technical session. Let us begin by introducing the key figures who will guide us through this intellectually stimulating session. We are privileged to have this session by Dr. Srinivas Reddy Bonam, Senior Scientist, Vaccine Immunology Laboratory, Department of Applied Biology, CSIR, Indian Institute of Chemical Technology, Ministry of Science and Technology, Government of India, Hyderabad. And this session will be presided. We'll be having the chairperson for this session, Dr. N. Ramanakumar sir, accompanying his co-chairperson, Dr. Syed Supriya madam. I request the chairperson and the co-chairperson of this session to please take your chairs. First and foremost, I would like to thank our speaker, Dr. Srinivas Reddy Bonam, for being here with us. Thank you so much, sir, for your presence. On this behalf, I would like to take the opportunity to introduce the co-chairperson, Dr. Syed Supriya madam, to all of you. She is currently the head of the Department of Pharmacology in KVSR Siddhartha College of Pharmaceutical Sciences, Vijayawada. She has completed her PhD in 2022, her master's in biotechnology in 2008 from Andhra University. She completed her bachelor's from Deccan School of Pharmacy under JNTU, Hyderabad. She has a teaching experience of 13 years and above. She has done publications in various national and international journals, up to 50. She has 4 published patents. She has authorized a book and 2 book chapters. She has received few awards and applied for various grants. Ma'am, we are privileged to have you here with us today. Thank you so much. Now I request you to take over the session by introducing the chairperson to all of us. Give a big round of applause and welcome the co-chairperson onto the dais, please.
Good morning, one and all. First of all, I thank the management and principal sir and all the organizers of this conference for giving me this opportunity to act as a co-chairperson for this coming session. And it's my privilege to introduce Dr. Poppala Ramanakumar sir, who is working as Associate Professor at St. Mary's College of Pharmacy, Chebrolu, Guntur district. Sir has completed his B Pharmacy at Vishwabharati College of Pharmacy, and sir also completed his Masters from Wales University in the year of 2009-11. Sir has completed his PhD in the pharmaceutics from Wales University, and he received the Best Paper Award for his research work. Sir has 15 years of industry and academic experience, and sir also worked in various multinational companies including HCL Technology, Smilex Laboratories, Arabindo, and so on. And sir has published around 50 publications in various peer-reviewed journals. Now I would like to invite the chairperson, Ramana Kumar sir, to come on to the dais and to start the session with the resource person.
Hello everyone, good morning one and all. It's my honor and privilege that I have acting as a chairperson for this prestigious event. Thank you for the management for inviting me. Thank you so much. Today it's my privilege to invite Dr. Srinivas Reddy Garu. So he is one of the eminent scientists in CSIR. So I would like to introduce Dr. Srinivas Reddy Garu. He is an immunopharmacologist and graduated and postgraduated from Chalapati Institute of Pharmaceutical Sciences, and he received a PhD from AASIR at IICT, and his PhD focused on vaccine adjuvant development against infectious diseases. During his PhD, he was awarded Raman Charap Fellowship by CFIRA. So, and for 6 months to work under the guidance of Professor Selvin Muller at IBMC, University of Strasbourg, France, where he worked therapeutic strategies for autoimmune disease including P140. And he also worked as postdoctoral fellowship research with Professor Selvin Muller. Later, he continued his postdoctoral research on various aspects of immunology. So, mainly he then moved to University of Texas Medical Branch in Department of Microbiology and Immunology, where he trained under Dr. At8, where he developed novel mRNA vaccines for emerging infectious diseases, including COVID-19 and therapeutics. At present, he is a senior scientist at CSIR, Indian Institute of Chemical Technology, Hyderabad, and also holds honorary assistant professor position at AASIR, Ghaziabad, Uttar Pradesh. His current research focuses on development of mRNA-based vaccines and therapeutics, including CAR T cells. He has published 90 research and review papers, 7 book chapters, and he has 5 patents to his credit. He has licensed 1 patent to private organization. He has been serving as editor-in-chief for Advances in Pharmacology and Pharmacy and an editorial board member for PLOS 1, so Frontiers in Immunology and different journals. And he has been serving as a guest for General Vaccines, Frontier Immunology, Journal of Immunological Research, and others. His publications has received 4,000 citations and boosting a 35H index and 60 I-10 index. Sir, it's privilege and honor to welcome you onto the dais to give your valuable speech to the students. Thank you, sir.
Yeah, I want to follow, Nagaraju sir, a short video about my institute.
The Indian Institute of Chemical Technology, located in the capital city of Telangana, Hyderabad, is an avant-garde research institute that specializes in chemical sciences and technology. Set up in 1944, it is one of the oldest research institutes of the country. CSIR IICT has been playing an important role in supporting India's chemical technology sector and related industries, including MSMEs, by developing chemicals and products that help reduce dependence on costly imports.
Our vision is to serve the society by creating outstanding knowledge base in chemistry and chemical technology. This was originally established in the year 1944 by Nizam's government at that time, and just before independence. In the 1956, again, our institute is renamed as a Regional Research Laboratory, Hyderabad. This is probably one of the first RRL created in our country. Again, in the year 1989, our institute renamed with the present name, that is IICT, the Indian Institute of Chemical Technology. We are in the service of almost 8 decades for the nation through chemistry and chemical technologies.
Being a premier research institute of CSIR, or Center for Scientific and Industrial Research, under the Ministry of Science and Technology, Government of India, IICT is active in research and development in cutting-edge areas of chemical sciences. It has state-of-the-art research departments which carry the vision of an Atmanirbhar Bharat in chemical sciences. The state-of-the-art facilities and a consistent focus on developing innovative solutions to national and global needs in the areas of drug discovery, agrochemicals, catalysis, material sciences and environmental science makes IICT as the foremost chemical research institute in the entire country.
IICT is located in the very city of Hyderabad. Hyderabad is also called pharma capital of world. So basically many drugs are being synthesized here and exported to various countries. And in coming back to our IICT facilities, we have excellent infrastructure, the trained manpower, and particularly infrastructure. If you look at analytical facility, we have NMR and we have X-ray and we have excellent mass spectroscopic facilities we have. And in the lab, experimental lab, we have starting from milligram scale to we can go up to kilo level, then commercialize. And we also have mole bank where we store the compounds what we synthesize in our laboratory, and also we procure from outside laboratories, outside institute also we'll get, and we store these molecules. We use those chemicals, that molecules what we stored in mole bank, and screen for various diseases. We also have another fantastic facility, we have animal facility where we can test some of the molecule what we synthesized in our laboratory.
In addition to carrying out state-of-the-art research, IICT Hyderabad also caters to the scientific human resource needs of the country in the areas of chemical technology. It offers prestigious academic programs at PhD and postdoctorate level, where budding scientists learn the most advanced techniques of the science of chemicals. Identification and precise structural determination of molecules are pivotal for developing new products with desired functions. This is carried out by highly qualified and experienced scientists and engineers of CSIR, IICT, who use state-of-the-art facilities and infrastructure. Some notable facilities which are available at CSIR, IICT include Mass Spectroscopy Center. This is a centralized facility of the institute which has most advanced mass spectrometers. These highly advanced mass spectrometers not only provide molecular weight information about any chemical or element, but also its structure, composition, and other physical features. Gas chromatography. Volatile compounds such as gases, amino acids, environmental toxins, and those chemicals which turn into gaseous form at normal room temperature can be analyzed using gas chromatography coupled with mass spectrometer. Liquid chromatography mass spectrometer. Liquid chromatography mass spectrometer is used for the analysis of non-volatile chemicals such as chemicals obtained from natural products and herbs, human and plant metabolites, synthetic organic molecules, agrochemicals, proteins, etc. High-resolution mass spectrometers. CSIR IICT houses different types of sophisticated mass spectrometers such as LC high-resolution mass spectrometers, LC quadruple Orbitrap mass spectrometer. Similarly, the institute is equipped with NMR spectroscopy, X-ray diffraction, thermal and surface analysis and other advanced analytical techniques which help in chemical and physical analysis of chemical and compounds in the country. CSIR IICT has made significant contributions in the field of chemical sciences and technology. Some of the key contributions include drug development. One of the important themes of CSIR, Indian Institute of Chemical Technology, Hyderabad, is healthcare. The Department of Applied Biology is involved in basic and translational research areas related to cancer, cardiovascular and metabolic disorders, neurobiology and inflammation. This department also caters to the needs of the private stakeholders in the above areas.
Try one more time. If you are not getting, it's fine. Yeah.
IICT has made significant contributions to the development of drugs for the treatment of diseases such as tuberculosis, Alzheimer's and cancer. Catalysis and chemical synthesis. IICT is known for its metabolic disorders, neurobiology and inflammation, Alzheimer's and cancer. Catalysis and chemical synthesis. IICT is known for its contributions in the areas of catalysis and chemical synthesis. The institute has developed several new chemical reactions and catalysts that have applications in the pharmaceutical, petrochemical and agrochemical industries. Chemical process development. IICT has also made significant contributions to the development of chemical processes that are more efficient and sustainable, including 2-ethyl hexanol, a key ingredient in the production of plasticizers. Renewable energy. IICT has been working on the development of renewable energy technologies such as solar cells and biofuels. The institute has developed new materials and processes for the production of solar cells that are more efficient and cost-effective. Waste management. IICT has also contributed to the development of technologies for waste management. The institute has developed processes for the treatment of industrial waste and wastewater that are more effective and environmentally friendly.
I can talk about very recent one, that is a hydrogen hydrate plant establishment in Gujarat, at Day-2. So this hydrogen hydrate is a very important chemical that is used as a rocket fuel and also key starting material for APIs, crop chemicals and many other ways. This is useful chemical, and right now we mostly depend on importing. In our institute, almost like 2 decades ago, this plan was initiated, and now recently established that plant and dedicated to the nation by our Honorable Prime Minister Dr. Narendra Modi ji. This is going to save us a lot of foreign exchange. Almost I think 400 crores probably it is going to save us.
CSIR IICT has a grand futuristic vision to make India a global leader in chemical technology sector and give a push to the green chemistry by exploring alternative and eco-friendly chemicals. IICT's larger vision for the future is to help India become a global leader in the chemical technology sector and importantly as a research and development hub for green chemistry by developing eco-friendly chemicals and products which are sustainable, safe and non-polluting to help fight the growing challenge of climate change and global warming.
Yeah, thank you. Slides, please. Yeah, first of all, I must thank to Nagaraju sir because he mostly covered my topic because I am also a nanoparticle, going to discuss about nanoparticles, but he discussed most of the basics. So I will explain a little bit practical, my practical experience, what I have been doing, okay. As Nagaraju sir suggested, let me first tell me about, about myself. So my father was an auto driver when I was doing B Pharmacy at Chalapati Institute of Pharmaceutical Sciences. So it was very hard for me because I got a free seat in, during that period. So I immediately entered into the pharmacy profession. Then after B Pharmacy, I qualified GPAT. Again, I took same college because I love my faculty, how they teach. So I joined there and I continued M-Pharmacy also. After M-Pharmacy, I immediately got a project assistant professor, assistant position in Indian Institute of Chemical Technology. So I joined as a project assistant. After 1.5 years when I worked there, so my PA was very impressed and he immediately offered me a PhD and through ACSR and he bought funding for me for 3 years. So I completed my PhD in next 3 years. So total my PhD was 4.5 years. In 4.5 years, I had an opportunity to visit France through Raman Charpak Fellowship. Immediately I went University of Strasbourg through 6 months fellowship. And again, the French professor, impressed by my work, immediately she offered me postdoc. She asked, Srini, go back and submit your PhD thesis, come back. So immediately I went back. 2 years I continued my postdoc with her. She supported from her company because she owned a company. Then after that, I moved to USA because now those days in 2020 COVID pandemic time, so my expertise is purely on vaccine development. So USA visa also it was difficult those days, no visas, nothing. But French embassy took my case as a special case and they approved in 1 day. It's unimaginable for getting visa for US in 1 day. Morning I submitted, evening I got a call, sir, your visa approved, come and collect. In any country you ask, it's highly impossible. With a hard copy, not soft copy. That was the case, my case for going and attending the UTMB. So then I moved to the UTMB where I worked with Professor Haitava Hood. We worked on the development of novel mRNA vaccines for COVID-19. My fortunate thing is my professor was directly a first student of Nobel laureate, Drew Weissman, but when I joined his lab, he didn't receive Nobel Prize. We closely worked with him for 2 years. Later, after 2 years, he awarded Nobel Prize. So that was my, I can say, my fortunate. Oh. Yeah. We know central dogma of life, okay? How DNA to DNA, DNA to RNA, RNA to protein. Okay, in our biological system are viruses, when they, we don't have pointer? Pointer is not there? Okay. So, we know if it is a DNA to DNA replication and when DNA to RNA transcription and after RNA to protein translation. if you want any protein, the process is like using RNA and making a protein. So even virus use the same strategy, like RNA viruses, DNA viruses, all these pathogens, they use same strategy to make their DNA content into RNA, RNA content into protein and replicate. That's how virus survive in our body. Then scientists thought, okay, virus is doing so, why can't we make our RNA in vitro in the bench, not in cell, making RNA on the bench and putting that RNA into the cell, so that from RNA protein will be produced. If you want a cancer protein to be expressed or cancer antibody, an antibody against cancer if you want, make an RNA and put it into the cell. Then our cell will read this RNA and make into the antibody. So you have a drug. So similar strategy those days, it started long back, but it has some limitations. That's why people never used it. So due to the advances in the technology, people come up with various modalities. Then now we have mRNA technology, full-fledged mRNA technology into the market. We know we all witnessed the COVID-19 pandemic. Imagine without COVID-19 vaccine, mRNA vaccines, it's miserable. So thanks to this technology. So what is mRNA? So this, you see the life of mRNA, how it goes. mRNA is a single-stranded RNA. Everyone knows here, like nucleotides, ATGC, AUGC, right? So this is a single-stranded RNA. It should have cap. Cap generally protects the RNA, and it is having UTR region, untranslated region, and it is having ORF, okay, open reading frame. This is where the protein sequence has. If you want any protein or any drug, if you want to generate, drug means synthetic biological drugs. We need to insert our sequence there, ORF region, and again 3-UTR and poly-A. Poly-A is it has a function to protect the RNA, both cap as well as poly-A. So this is how it goes. For example, in cell, DNA to RNA, we know RNA polymerase takes place, and then once it forms RNA through the cap process, it exits the nucleus, and immediately ribosomes capture this RNA. Once it captures, it starts reading. When RNA reads what it comes, then protein. That's how what we are learning from the first B form onwards, or intermediate. DNA to RNA, RNA to protein. That is what we have now, protein. So what scientists are doing, we know now if you give RNA to the cell, whatever we want, RNA to the cell, if you deliver RNA to the cell, then it makes a protein. So scientists started using in different aspects. Like if you want to develop a vaccine we know, for example, COVID-19 vaccines. We have SARS virus. SARS virus has a spike protein. So people took spike protein sequence and put it into the mRNA sequence. Then this mRNA they delivered to the cell. So cell will give the now spike protein. So once it is in body, cells will start memorizing this, recognizing these peptides. And when we infect with SARS-2 virus, then immediately our immune system kills that virus. So that's how vaccines are developed. And also these days, people are using for cell therapy, particularly cancer cells. Imagine now, cancer is like, the drugs are not suitable for cancer, because now personalized medicine has come into the picture. Because once we start treating cancer with the drugs, they are mutating. We have so many mutations in the cells and start expressing different antigens. So researchers identified, why don't we adapt patient cells? For example, if a person having cancer, they are collecting cells from the patient and they are engineering on the lab, hood, they are engineering in the bench cells and again injecting back. Those are like adaptive cell transfers. Those are called CAR T cells. And also gene editing. If people, born with any genetic diseases, we can modify their genome by using this RNA technology and make them targeted protein produce. Similarly, like antibody production and protein replacement also. Fine, this is a RNA components, how this RNA looks like what I explained, cap, UTR, ORF, and UTR and poly-A. Somehow, through the years of research, researchers are identified what cap is required, what how the UTR, what UTR required, and how poly A is required. Based on that, scientists are fixed. Okay, fine. RNA is ready. And when you have, this is the overall picture how people generate mRNA vaccine. So we should first design DNA, because RNA comes from the DNA, right? So we have to first design a DNA, and DNA we have to grow in the bacteria, we have to transform into the bacteria and culture, grow the bacteria, and you have amplified the DNA, required DNA, then linearize the DNA. So you provide the enzymes, RNA polymerase enzyme. So it makes RNA. You have RNA in your hand. But the difficult part is RNA is not stable, because we know nucleases, they cleave the RNA. So for that, we need nanoparticles. That's why I of nanoparticles. So when you put this RNA into the nanoparticles, they protect the RNA, and when you deliver it to the cells, directly cells take up the RNA and make the protein. Let me show you how it does. See, this is the picture. So we make lipid nanoparticles by using these lipids: ionizable lipid, helper lipid, cholesterol, and lipid and PEG. So this is the mRNA we made. Imagine whatever mRNA. You can make a cytokine, chemokine, vaccine, or cancer vaccine, or antibody, anything, mRNA. And you take this lipid nanoparticle and use this nanoassembler. This is like a microfluidics machine. When you inject these two from the two angles, they mix them, the machine mixes very well, and it produces nice lipid nanoparticles. The advantage of the ionizable lipid is, ionizable lipid having a positive charge in acidic pH. So it interacts with the negative charged mRNA, because we know mRNA is like nucleotides having phosphate group, and it has a negative charge. So we need a positive charge to bind. So ionizable lipid have that positive charge at lower pH. So once you give the nanoparticle to the cell or inject into the human, the first cell, when it encountered the nanoparticle, we know the phagocytosis process, right? Through phagocytosis, nanoparticles goes into the cell, and after forming a phagosome, it has a acidic pH. So acidic pH has a positive charge again. Then in the acidic charge, the LNPs are more, the ionizable lipid take up the positive charge and release the mRNA inside the cytosol. Then now mRNA is available for the ribosome. Ribosome do its action. It makes again protein. That is how all the system works. If anybody says mRNA technology, this is what whole mRNA technology, how it functions inside the cell. Okay. Again, we made mRNA, we put it into the lipid nanoparticles, we delivered it to the cells. That's fine. Because this is outside made, not cell made mRNA. So outside made means we don't have post-transcriptional modifications. Generally, inside the cell, if our cell makes DNA to RNA, we have post-transcriptional modifications. So cell won't recognize, because it's our own RNA. But we are making RNA outside on the bench, so cell will recognize this as a foreign material. Okay, some foreign material comes, immediately our sensor system activates and suppress the translation. So, coming to this entire chart, if you see 2005, they demonstrated that nucleoside modified RNA is non-immunogenic. So this is how it senses. So when you use unmodified mRNA, means we prepared mRNA on the bench, we delivered to the cells through the nanoparticles. Immediately what happens, it detects our sensors like TLRs or cytosolic RNAs, they recognize this. And they suppress the translation mechanism. So our required protein won't come out from the cell. So at the 2005, what researchers have discovered, they discovered pseudouridine modification. Means we know uridine, AUGC, we know uridine, but they modified uridine into pseudouridine. They identified that in our body, when DNA makes as RNA, we have pseudouridine in the RNA structure. They identified that, and they started using pseudouridine in vitro. While making RNA, they are incorporating pseudouridine instead of uridine. So now that is safer, and also easy to translate RNA to protein. Who did this? These people did this. And Dr. Weizmann and Dr. Carico, these two people identified that if you incorporate uridine and AUGC into the RNA sequence, our immune system recognize and suppress the protein translation. But if you put a pseudouridine instead of uridine, so our immune system won't recognize our whatever targeted protein translate into the cytosol. Okay? So then that's why these people got Nobel Prize in 2030, in 2023. Recently, these two people got. So this is the one structure. This is common uridine, and this is pseudouridine, modified one. And also both this technology used by Pfizer and Moderna. So how the vaccines works generally? Okay, we have now mRNA, we encapsulated the mRNA, we have nanoparticles with mRNA, and we put mRNA as a spike protein for the vaccine. So how does it work? When you inject into the mice or human, how does it work? See, when we inject into the mice, it generally goes like a phagocytosis process and makes the protein. That protein again goes to the proteosomes and cleaved by the, proteosomes cleave the peptide and also presented to the immune system. Like T cells, immune system. Forget about these T cells, B cells, okay? Imagine, okay, immune system. Immune system recognizes these peptides and generates antibodies and protective mechanism. This is how vaccines work. We are lucky, we always take, when we were kids, like BCG vaccine, polio vaccine, many vaccines we took, all they act in the same mechanism. Okay? Now advantage is, compared to the conventional vaccine, the mRNA vaccines gives more CD8 T cell responses. CD8 T cell responses, they are very much important for the cancer treatment. If you make a cancer vaccine with mRNA technology, they are more affordable and also we have long-term activity. Yeah, when I joined with Professor Hu at UTMB, we started working on mRNA technology, and along in collaboration with Professor Drew Weizmann. So we used to do many mRNAs, monovalent, tri means not only one mRNA. We used to put many mRNAs with many constructs in one lipid nanoparticle and test against mice and hamsters and even monkeys. So these are all publications. So after getting Nobel Prize, he immediately visited our lab. So because we have been working for him, it was fortunate, and even I went there. After, once he visited, I immediately went his lab, and because I have some technical difficulties in the technology, then I was there for one week to learn those technologies. And these are the publications we did along with Drew Weizmann. So all our top publications like Science, PNAs, and NPJ vaccines, Science Translational Medicines. Okay? Yeah, let me take as an example this one, like PNA's paper, the top one, how we come up with a novel COVID-19 vaccine, like a case study. So we had collaboration with Tufts School of Engineering. So they have, during the discussion, because we know everyone took COVID-19 vaccines, Pfizer and Moderna, but they are not active because we got different waves, Omicron wave 1, 2, 3, like that. So we were thinking how to give a broad protection against these SARS-2. So during that discussion, these people, chemistry people, they come up with a novel lipid, ionizable lipid 88. As I told you, these ionizable lipids are important to bind to the RNA, negative charged RNA. So they have come up with the new novel lipid, and also we wanted to bring novelty to get a broad spectrum of activity. So we changed these UTRs. We didn't change the sequence spike because spike is like protein what we wanted. But we changed these UTRs, 5 UTR and 3 UTR, different sequences, and put that mRNA into the nanoparticles with that ionizable lipid. And we wanted to check the efficacy of these nanoparticles. We know Pfizer uses ionizable lipid ALC0135, and Moderna uses SM102. But they are only localized in the liver, spleen, and lymph node, but it has humoral immunity, means antibody response is more, but not cell-mediated immunity, not T cells. So what we did, after changing the UTRs, we treated these mRNA to the cells, and we wanted to look out how our strategy is working. We can see the red spotted ones having more expression when you change the UTRs. I know this is too scientific, you don't understand, so let me go fast, okay? So we made mRNA with changing the UTRs, and we encapsulated with lipid nanoparticle, and then we injected into the mice subcutaneously. So after injecting, we wanted to see how our mRNA is working. So we used reporter gene. Instead of spike protein, we used luciferase and GFP, so that once it is translated in mice, so protein, the fluorescent proteins we can detect through the imaging technology. So this is live imaging, we can see. So after finding two best matches that we have very good expression, these two best matches we took further, and we again made LNPs, and we wanted to see how these lipid nanoparticles distribute in the animal tissues, like where they are localizing. And we collected all spleen and lymph node liver after 5 hours of injection, and this is the one expression we can see. Like they are more localized in the liver, spleen, as well as lymph node. And we compared this with the standard like Pfizer vaccine, okay? And also we wanted to see, okay, now mRNA is ready, lipid nanoparticles ready with novel lipid. So whether, if you put a spike vaccine, whether they are really acting as a vaccine or not. So then we made a vaccine and we injected into the mice, like 1 microgram, and we gave booster dose. Everyone took booster doses, right? Same way, we gave booster dose to the mice. And then after some certain period, we collected the lung and spleen. Then we evaluated the immune response, how our vaccine is acting, again, how our vaccine is immunogenic. So we have seen a good immune response compared to the Pfizer vaccine. And later the question is, okay, your vaccine is working and immunogenic, immune response is good, whether vaccine is protecting against SARS-2 or not. So efficacy we wanted to test. What we did, we used a hamster model. This is a very sensitive model for SARS-2 because mice was not working those days. And then we used hamster, we injected we immunized with our vaccine, and again booster dose, same like mice. And after 35 days, we challenged with the real virus through intranasal route, because everyone, COVID time, those days, everyone infected through the nasal route. So same way, we infected virus through the nasal route. And after infection, we measured the weights, whether after infecting, weights reduced or not. It's an indication of like a fever or disease. So then after every 2 days, we collected the organs to assess whether virus is inside, persistence virus is there or not, how our vaccine protected from this virus. So we can see here, we have good antibody response with our vaccine, and when we collected a hamster lung and as well as nasal was, with vaccine, the red and blue dots indicates suppressed viral titers and viral RNA. So we can estimate viral content by RNA because it is RNA virus. So this is a whole picture, what we did in the study. I know this is too much science, that's why I summarized in a few slides. So this is how entire picture, what we did. We developed lipid nanoparticles and novel mRNA and tested in mice and hamster and evaluated how the immune function works with these nanoparticles. And later what we did, as I told you, we worked on many vaccines, many therapeutics. So one more challenge was C. difficilis vaccine. So this is like a pathogen which causes intestinal infection. In US it was very, more burden for people. So what we developed, we took three antigens from the C. difficilis, toxin A, toxin B, and JMP, and we again did same process. So first we checked with the cells, then injected into the mice, then injected into the hamster for protection, then injected into the monkeys. Then again we evaluated how our vaccine is working. The vaccine is working very well, and we published this study in Science, Real Science, and the technology is taken up by Pfizer in US. So in my lab now in IICT, we are currently working on different vaccines. We have collaboration with Bharat Biotech and other small companies. And also recently we started collaborating with Pulse Pharma, a small company where they are working on CAR T cell therapy. So what is this CAR T cell therapy? As I explained you, for cancer patients, we need personalized therapy. So we need to take cells from the patient, we need to collect cells from the patient and collect the T cells, because these are the cells fight against cancer, and engineer these cells in the lab against that cancer antigen, and when we have the cells ready, infuse back to the patient. So for this, US is known because US, they are using this CAR T cell technology for person. They are costing, I think, $100K, I think. One person, if attended hospital and wanted to go for this therapy, he has to pay $100K. In India also we have this technology. Recently one company launched, but we need to pay 40 lakh. If a person is suffering with cancer, if you want to go with this therapy, 40 lakh we need to pay. But from IICT, what we come up with, we wanted to reduce that 40 lakh to 10 lakh or at least possible extent. So we are using mRNA technology. What we can do, they are using lentivirus to engineer these T cells. So we wanted to use mRNA and express the same protein. So mRNA is cheap. As I told you, we have DNA, make linearize and make mRNA in vitro. So we wanted to use same technology and make our construct and use for the patients. So now we are working on this and we are expecting big grant also on this. So we are expecting soon. Yeah, definitely we have some advantages and disadvantages also, like every therapy has, so like similarly. And finally, I want to thank my mentors, like Professor Ramarao Sir Nadanla and Sampath Kumar, my PhD mentor, Professor Selvian Muller, first postdoc mentor, Professor Jagdish Bairi, second postdoc mentor, and Professor Hayato, who gave me a non-tenure track position as a faculty position at UTMB. And recently I got a funding from the Ignite Life Science Foundation to work on self-amplifying mRNA vaccines. That is again a big story, I'll explain you later. And this is my team, 2 PhD students and 1 project assistant and 2 dissertations. And I thank IICT as well as CSIR. Thank you.
Yeah, it is a nice, valuable presentation to our students, sir. Thank you very much for your session. It is very nice interaction section with our students. So one small question is there, sir.
Yeah.
So researchers using artificial intelligence to personalize cancer treatment right now, nowadays.
Yes.
So any kind of softwares are there to identify earlier prediction of cancers?
Yeah, now AI, ML, like artificial intelligence and machine learning, we have predictions. We can identify definitely. Nothing but a biomarker identification. Based on the biomarkers difference, they could identify whether how far he is getting, how quickly we are getting cancer. Not like exact precision we are lacking, but there is a provision. We can easily detect, like, okay, next 5 years you are going to get this cancer, like that. The AI ML is so advanced, we have this technology now.
Okay, thank you, sir. One more thing.
Yeah.
So last week, I think so, I had heard a news that Russia claims to develop new mRNA vaccine for cancer.
Yes.
And according to one of the agency, the government had also decided to give vaccination for their people.
Yes.
So any developments for government of India, especially I want to know, as you are working in CSIR, any central government is in your prestigious institute, can we know any further developments and challenges in preparation of vaccines for India?
Yeah, Russia recently launched a breast cancer mRNA vaccine. So we have seen in news, but Russia is too, what we say, possessive. It won't release the technology details, what protein they are targeting for cancer, because you need to target some protein when it is an mRNA. So when it translates as a protein. So it never, disclosed what protein they are targeting. But we are working on mRNA, definitely cancer vaccine in IACT. We have our own target. For breast cancer as well as ovarian cancer, we are working. We are in the preliminary phase. We have already prepared mRNA and we have seen the expression of our protein in cell cultures, but not into the animals. We are soon going to the animal model. So once we have animal results, definitely we will first make a patent, then followed by publication. Yeah.
Thank you, sir, and all the best for your research work. And hope CR rise in India, CSIR. Thank you so much.
Thank you.
Thank you, Dr. Reddy. It was splendid hearing you. We
Thank
had
you.
a chat also last night. It's remarkable that mRNA concept and because I also work in the area of nanotechnology, so this liposomal encapsulation of mRNA is a nice one. I just have, it's not a question, I just have a query kind of, like I have used for the oral routes and I have seen that you have used the nasal route as well as oral route, right? So which route do you think is the best suitable one? Means you find it's more convenient. And another question you then answer. Another is that you have, what I have gone through the slide, it's a very interesting thing, means I heard, but the way you projected, I really liked it. Means I understood the concept.
That's what my intention.
Actually, I understood the concept. So, what if we can prepare the mRNA or we can design the mRNA? So I think that liposomal or encapsulation of that mRNA is possible for targeting any protein of any disease. What I learned from this, right? So there again is my question that for suppose say for I work in diabetes or Alzheimer, so for that nasal route, if I suppose, so the drug has to go for the Alzheimer's, the drug has to go to the brain also, as well as there is a relation between Alzheimer and diabetes relating to insulin pathway, insulin regulatory pathway. So it has to be multi-targeted, right? Because it is a multifactorial thing. So please give an insight, because I am very keen and I'll definitely have a talk after, because I'll definitely want to have a talk regarding this. But this oral and nasal route, please give an insight regarding this.
So first of all, the nasal route what we gave is virus, not the nanoparticle. Nanoparticle, if you see, we injected subcutaneously here. So when we wanted to check against virus, because generally SARS-2 entered through nasal route, so we used the same mechanism and we challenged it with the virus through nasal route. But as you said, there are so many ionizable lipids now recently come up. Those lipids selectively target we can use through the nasal route. So if you want, you can check his publications, my collaborator publications, like Mohammed, because he's a big guy. Now he's publishing at least every week, one science paper, one nature paper. Yeah, this fellow, Mohammed, if you check his profile, Mohammed Alme, he's from the Penn State, University of Penn State. Recently, they published many papers. One paper is on intranasal delivery of mRNA. They used ionizable lipid that is having mucoadhesive properties, and it delivers the mRNA intestine through the mucosal route. Okay? Thank you.
Hello, sir.
Yeah.
Very nice presentation, energetic presentation.
Thank you, sir.
And I try to understand very little bit knowledge on this kind of. Regarding that novel coronavirus vaccine, as you have told. So very devastated situation around the world created
Yes.
during the COVID-19. For Covaxin, that virus taken from, suspension taken from the asymptomatic person, virulence kill, then injected into the healthy human.
Yes.
For development of the antibody.
Yes.
But
Not
for
development of-
Covishield, yeah.
Not development of antibodies. Antibodies they inject into the healthy, infected people. Simple. How it goes, let me show you. Just to give the idea to the. See, every vaccine generally, whatever we are taking, it acts in two different ways. One is producing antibodies. These antibodies are specific to antigen what we are giving, or vaccine what we are taking. If you take BCG vaccine, you get an antibody specific to BCG. Next time when BCG or any pathogen enters, these antibodies neutralize. This is called humoral immunity. But the other one is cell-mediated immunity, called these T cells and CD4 T cells. These T cells, they are also antigen specific, but they produce cytokines to kill the virus, to kill the infected cells or direct virus. So this is the two pathways immune system works. During the COVID, what happened? Few people got a COVID, but they are asymptomatic or they recovered quickly because of their each immune system. Each have little variations in the immune system. So, but recovered patient will have antibodies, right? Because they infect, they have the antibodies against virus. So what doctors thought immediately, we don't have vaccine. Let us take antibodies specific to virus from that person and inject it to the ICU patient, because patient is suffering in ICU. When we inject these antibodies, what they does, these antibodies go and bind to the virus, right? They neutralize. That's what people was using, and this was like antibody therapy those days, initial days.
Okay, so Covaxin is based on antibody for the asymptomatic person.
Vaccine is acts in both ways, T cell as well as antibody.
Okay.
Yeah.
And for Covishield, that chimpanzee adenovirus vector, right? That can able to generate the spike protein.
Yes. I think Oxford
Yes,
vaccine is adenovirus. Again, if I don't know, did I present first slide? Let me show you. Sorry, this is the first slide. So vaccine can be prepared in different ways. So we can use either whole vaccine, like you can see live attenuated vaccine, because when virus is there, you can directly inactivate virus with some chemicals, okay? Inactivate it so that it won't proliferate in the body. But inactivated vaccine virus, when you inject into the human, it's inactivated, but our immune system immediately capture and process it and make antibodies against it. Similarly, either live attenuated, but all we cannot do that because some viruses are pathogenic. If you inactivate also, they get the opportunistic way. Again, they bounce back and maybe infectious. That's why people have started working on whole inactivated vaccine. Or if it is also having drawbacks, go with the split vaccine. Or if it is two, then go with the mRNA or DNA and subunit. So these are the different vaccine modalities. But the concept is same. Whatever the vaccine we take, we will develop pathogen-specific immune response, antibodies and T cells, cell-mediated response.
Okay. Sir, for main protease,
Yeah.
COVID-19 main protease,
Yeah.
and like you know that this part, split inactivated virus.
Yeah.
So spike protein, it is attached with the membrane
Yes.
towards the surface.
Yes.
So that mRNA has taken
Yes.
for the development of Covishield.
Covishield,
Yeah.
yeah.
And regarding that mRNA main protease,
Same, spike protein.
mRNA main protease I am asking, which is responsible for synthesis of genetic material. That is not targeted for the development of vaccine, right?
Yeah. Why we wanted to target only spike in this SARS-2? So virus is, completely, when it is in active shape, if it has to enter into the cells, it needs spike protein.
Yes.
Right?
Yes.
So people...
For to occupy the receptor,
Yes,
human
to
receptor.
to bind to the ACE2 receptor, it needs only spike. The later comes to the other protein functions. So then researchers simply thought, okay, target spike, don't, block the attachment. So why you wanted to go for the next target? We have spike. So that's why most of the vaccines targeted spike. We wanted to block the attachment. First instant, we wanted to block the attachment. Then no infection.
Okay,
Yeah.
thank you. Thank you very much. Thank you.
Sorry, sir.
Thank you, sir. Thank you for your wonderful presentation related to the immunology and synthetic vaccine preparation. I have a small doubt, sir, related to the mRNA vaccine preparation. So you are saying that an mRNA vaccine preparation, the uridine is replacing by pseudouridine.
Yes.
So pseudouridine, does that pseudouridine will give the accurate antigenic protein expression, sir?
Yes, that's what they got Nobel Prize, because in normal human physiology, from DNA to RNA starts post-transcriptional modifications. So during the transcriptional modifications, uridine convert into the pseudouridine. But initially they didn't realize what's happening inside. Simply they thought, okay, making RNA is easy, AUGC, okay, make RNA outside. So they used AUGC and they injected into, put it in the cell. So once it started, they started seeing, they don't see after 24 hours, expression is lost. There is no protein. Then they were thinking, why we lost this protein expression? We gave whatever needed, AUGC and other factors, why we lost the expression. Then these people, whoever got the Nobel Prize, what they identified, okay, after making RNA, there is a little change in the uridine nucleotide modification that is called pseudouridine. What happens if you put that, replace uridine to pseudouridine, whether expression is increased or not? They immediately made mRNA with pseudouridine and they have seen increased expression.
Okay.
Yeah, because
then how could that pseudouridine will get protection from immunity imparting elements, sir? If both the codons are having similar activity.
Yeah, this is, let me show you. So if you use only uridine in the normal, like without pseudouridine, this is a unmodified uridine part. Many viruses, daily we encounter many viruses. Not only flu or not only COVID. COVID is rare condition, it emerged. But every day we encounter so many viruses. But our immune system is so defensive that recognize foreign RNA as this is not modified, because the virus doesn't have capacity to modify uridine. So it comes as a native uridine. Then our sensors in our immune system with these sensors, they recognize uridine for the virus and immediately suppress the protein translation. So the RNA to it has to come protein. If protein comes only, virus particle can form. If protein is not coming, virus won't form. So our immune system suppress the proteasome system. So considering this, what they decided, okay, this is not a virus, but this is a therapeutic purpose. We should mimic our physiological system. So then they identified pseudouridine modification. They input pseudouridine. So we have easy translation process. So our proteasome system won't suppressed in this condition.
Okay, sir, with your permission, I want to clarify the last question which was given by our resource person. Sir, you are asking about spike protein, no, sir? With your permission, just I want to give that clarification. So in each and every virus or in the bacteria, the toxin fraction will be lies in any one organelle of the particular organism.
Yes.
In case of coronavirus, spike protein is acting as antigen.
Yeah.
So that's what we are preparing, spike protein related genetic fraction.
Because each and every virus is unable to produce the spike protein. Adenovirus can generate and this COVID virus can generate the spike protein. That's why spike protein is a big target for the drug development and vaccine development.
That is what we don't have vaccines immediately. We took some time to identify what is this virus, what is the components on outside proteins expressed, and whether they are stable or not. To make stable proteins, what to do, that's why we took some time to make. Otherwise, target is easy. Okay.
Sir, back slide please. Back one. No, that picture you have shown. For leave attenuated, inactivated, that slide. Leave attenuated, inactivated. Yeah, this one. whole inactivated virus, this one, na? So, COVID virus, 14 to 21 days in human cycle, reported, right, sir? So, for the development of vaccine, asymptomatic person, it's after the recovery or before it?
I didn't understand.
Means that serum was taken before 21 days or after 21 days? For the
After 21 days.
after 21 days.
Because we know that person is infected and he rested for some time. He generated antibodies against this virus, and virus neutralized, antibodies neutralized the virus. So we know a gap is there. Then we used to collect the blood from the recovered patient. So we have antibodies against this virus. Then we can inject these antibodies to the ICU patient who is in critical condition.
And that serum might have contained dead particle of the virus body?
No,
No.
it won't, because
No.
that's why we give gap.
We need
Doctors precisely estimate with qPCR, and they see if negligible amounts of RNA, viral RNA is detected, then only they inject. Yeah.
Thank you so much, sir. Before that, I would like to welcome the director of our institute, Reverend Sister D.U. Shaurilu Garu, who has arrived here. Sister, welcome to you. Now it's time to thank our distinguished speaker, Dr. Srinivas Reddy Bonam, for valuable insights. I request the chairperson and co-chairperson to come on to the dais, please. I request Dr. P. Pamal Reddy, sir, Hare Krishna Roy, sir, Professor Nagaraju, sir, Professor Dikhil, sir, onto the dais for felicitating a chief guest, for the speaker. I request sir to please take your chair and accept a small honor on behalf of our institute. Please give a big round of applause for such a wonderful session, for making us such an insightful and making deep thinking into the subject. We request the speaker to accept a small token of gratitude on behalf of Nirmala College of Pharmacy. I request all the dignitaries, the chairperson and co-chairperson and director sister to continue with the proceeding. Give a big round of applause, guys. All these subjects, what you learn from textbooks is different. What we get to learn through the research is different. We should be thankful and grateful for such eminent personalities who are sharing their knowledge with us and making us think deeper about the subject. Thank you so much, sir. Now it's time to felicitate the chairperson of today's session, Dr. Ramana Kumar, sir, to accept the token of gratitude. Sir, thank you so much for your acceptance as the role of chairperson in introducing our speaker to us today. Thank you so much for your time and effort. I request the chairperson, co-chairperson to accompany the same. Thank you, Raman Kumar, sir, for your support. We hope this will continue in future also. I request the co-chairperson of today's session, Dr. Seth Supriya, madam, to accept our felicitation on behalf of the institute. Thank you, ma'am, for your presence here today and for your role as co-chairperson. Special thank you to the speaker, chairperson and co-chairperson, and my heartfelt thanks to our director, sister. I request all the dignitaries to move off the dais. Thank you so much, sister, for your presence. To all the participants and student delegates, we apologize for the delay, but still, if the delay is giving us something fruitful, I think we all should accept it. Whatever is happening here is nothing which is going waste of time. It's going to be a fruitful information what you are gathering today. So I hope you all will accept the delay what's going on in the sessions. So thank you so much for your patience. I have few announcements. We'll be having a short tea break now for 10 minutes, sharp 10 minutes, because post tea break, we are going to listen from the very eminent personality whom we were waiting after interaction, sir. We had an interaction with Dr. Srinivas Pentyala, sir, who's come long way from US for all of us. So we'll be having a session by Srinivas Pentyala, sir, followed by Dr. Jitendra Patil, sir. So these two sessions will be continued after the tea break. After the two sessions, we'll be dispersing for lunch break. Post lunch break, we'll be continuing with your poster and oral sessions. Those who are here for participating from the departments of pharmacology and pharmacy practice poster and oral competitions, you will be going and presenting after the completion of two sessions post lunch. I hope I'm clear. If you still have any doubts, you can contact reception or the hosting committee during the break time. In the afternoon, we are going to have young researchers presentations, short research presentations. We have two research scholars who will be sharing their research work with us. After that, we'll be having 3 online lectures, which are short again for 10 to 15 minutes. So afternoon will be an enlightening sessions for you again coming up. Then we'll be ending up with cultural programs. After cultural, we'll be proceeding with the valedictory. I hope I'm clear with the schedule. So please take a break of 10 minutes for your tea. It's 12:20. I request all of you to be back by 12:30 for the sessions. Please try to be back in 5 to 10 minutes. I promise you that the coming up 2 sessions are going to be very interesting for you. Thank you all. Once again, a kind request to all the participants, please.
Welcome back, everyone. Without any further delay, we'd like to start another scientific session. The eminent speaker of this session is Dr. Srinivas Pentyala, Professor and Director of Translational Research in Anesthesiology, Faculty in Orthopedics, Urology, Health Sciences, Physiology, Biophysics, USA. Hearty welcome, sir. We are very fortunate to hear. Ladies and gentlemen, welcome our sir with a big round of applause, everyone. Once again, hearty welcome, sir. And for this session, Dr. Surya Prabha Madam is going to be acted as a chairperson, Department of Pharmaceutical Sciences, Associate Professor in Vignan to be deemed. Hearty welcome, Madam. I request chairperson to occupy the chair. And for this session, Dr. G. Swapna Madam is going to be acted as a co-chairperson. Madam, I request you to occupy the chair. It's time to invite our co-chairperson of this session, Dr. G. Swapna Madam, onto the dais to introduce the chairperson of this scientific session. Dr. G. Swapna Madam, M.Pharm PhD, Professor and HOD, Nirmala College of Pharmacy. Madam is having 15 years experience, 108 research publication, 10 patents, 19 awards, 12 book chapters, scientific presentations in national and international conference. We welcome you, Madam.
Respected dignitaries, dear faculty and students, it's my immense pleasure to introduce the chairperson, Dr. Surya Prabha Matangi. So, Madam is a pharmaceutical biotechnology specialist and she's working in the Vignan University, Vadlamudi, Guntur district, AP. Madam has secured her PhD in Acharya Nagarjuna University and she has 18 years of experience and she has made several scholarly contributions. Over her professional life, Dr. Surya Prabha Madam has actively guided over 25 undergraduate projects. Her main areas of interest in study include biological activity of phytoconstituents and in vivo in vitro evaluations. Madam has 25 research and review publications in esteemed national and international journals and her considerable research in the field. She has also written a book chapter that has been published by domestic and international publishing firms. Her inventive contributions to the field of pharmaceutical biotechnology and phytopharmaceuticals are evidenced by the publication of 2 patents. Dr. Surya Prabha Matangi constantly participates in national and international conferences in addition to her research and publications. She has given several papers and several conferences, and her presentations earned her prizes. She holds her leadership roles in committees at national and international levels like chairperson and adjudicator. So Madam is invited as keynote speaker at 2017 Innovate Pharma Conference in Sydney. And also she has presented at Spain on the subject of herbal banquet of products with different pharmacological activities, and gave a virtual lecture at KPG Health University in Malaysia. And also, Madam has given the title of presentation on drug discovery process on potential phytoconstituents for nucleoside analogs and viral enzyme inhibitors. As evidence of her active participation and dedication to professional organizations in her area, Dr. Surya Prabha Matangi is a life member of both the Association of Pharmaceutical Teachers of India and Indian Pharmaceutical Association. With this, I invite Dr. Surya Prabha Matangi, the chairperson, to come on to the dais and introduce the resource person. Welcome, Madam.
Very good afternoon to distinguished guests and then esteemed speakers. Very good afternoon. Before introducing Srinivas Panchala sir, I was informed that we want his session we need his session. Because of his session, we want to came over here. And I was also informed that you just give a very small and short introduction about him. Why? Because people and then delegates want to get the insights from you, sir. So with short introduction, it's my both privilege and then honor to introduce Dr. Srinivas Panchala, sir. He completed his degree from Sri Venkateswara University, Neurobiology, Tirupati, and PhD from Sri Venkateswara University, Neurotoxicology, Tirupati. And then he handed so many grants and then various research supports, such as molecular actions of volatile anesthetics on G proteins, then second messenger screening of peptides, interactions between opioids and general anesthetics, interaction of product R with G proteins and biological membranes, and identification of CSF in biological fluids, methods and kits to detect the CSF, treatment of Parkinson's disease, integrated fluid collector and disease marker concentrate. So like that, so many research and grants are there. Without reading that, I want to get all these insights from us, sir. And also, sir has so many patents, nearly 8 patents are there. And he is a consultant for various institutions, such as Care Solutions, Care Health Services, and then SIP Incorporated, Brookhaven, like that. So many institutions he is in consultancy. With this short introduction, as I was informed to give, as I don't want to waste time to introduce, sir, we welcome you and we want to get your insights. Thank you, sir.
Good afternoon, everyone. I think I am the one who is standing between you and your lunch. I and the other speaker. So don't hate me and my colleague. I'll take less of your time. You tell me when to stop. I'll stop. How about that? Okay? So instead of giving a talk on a project which will put you to sleep, I thought I'll talk about projects that are close to my heart, which are small stories. you can get excited with any one of these stories. Let's see how it goes. It's not working. The advancer is not working. Okay. Okay, this is my topic. How many of you are interested listening to the title of my talk? Raise your hands. You are very much interested in this? I'm not interested to give this talk because it will be so boring for you. So what I say is, what is this about? Let's say what is this about. Volatile anesthetics allosterically modify the alpha subunit of receptor linked whatever it is. You are already bored to death, right? You think this is so boring, I cannot understand this. So most of our projects are like this. If you go into advanced studies like your postgraduate studies or PhD programs, these are the things which you will be doing, and many of our students never understand these things. So same thing. All my projects have long titles, they get good funding, we get good publications, but when we talk to a general audience like undergrad students, they all say, what is this? So you can go on and on and on, and I'm going to talk about this one first. This is a project on a protein called phospholipase C delta 1. It's a big name to undergrad students. What this protein does is simply raise the calcium levels in the cell. That's all you have to remember about this title, okay? So what we did was, we understood how this enzyme works. We understood how the calcium level rises in the cell because of this enzyme. Then we did microscopy work with cell biology, and we were able to identify the exact location of this enzyme or protein within a cell. And we also monitored where this protein travels in the cell in response to drugs. That's a great finding for us, because if you understand how a drug can affect a protein within a cell, then you can target the particular disease. And to do this work, it took us 18 years. And my daughter actually teases me that I won't pass this exam because I'm constantly doing the same work for 18 years. But interestingly, what we found was, when you take a cell and make this protein expressed in high levels, the cell goes into cell death. Simply, you raise this protein, the cell dies. And we published it. I get invited to many places to talk about this. But one year, one of my colleagues in neurology, he sent me a sample of Alzheimer's brain. A patient came into the clinic and the patient died. So we took the brain of the dead patient, Alzheimer's patient. We actually made a homogenate of the brain, and we checked the levels of this protein in the brain. And to our surprise, in the Alzheimer's brain, this protein level is very high. 18 years of work targeting this one thing. Now I want to go back and tell one thing. Calcium level goes up, cells die. In Alzheimer's brain, this protein level is going up. What does it mean? Because it is also increasing the calcium level. And that's why the brain cells are dying and the patient is getting Alzheimer's. And it took us 18 years to figure that out. And so now what we are doing is, we are trying to make compounds with the help of medicinal chemists to actually target this protein and enzyme, so that if we decrease the enzyme activity or decrease the protein levels, the calcium won't go up and the cells will survive. So are we finding a cure for Alzheimer's? Maybe. And that's one of the small stories which I wanted to share with you. It's a small story, but 18 years of work on this small story, okay? And for every project as a translational researcher, we have an endpoint. We need to plan ahead. Within 2 years, 3 years, can we finish this project? That's the real definition of a translational research. So in our case, we are now going into clinical trials, and hopefully in the next 2 years, this will actually come into phase 2 trials where we'll actually recruit patients to actually see if this molecule works. And I'm a faculty in anesthesiology. Let me ask you, how many of you had surgery? Anyone had surgery? Okay, ma'am. So when you had surgery, did you get anesthetic? Did you ever ask your anesthesiologist, how does this anesthetic work? Did you ever ask? Did you ever try? If you ask that question, we put in more drugs so that you can sleep quickly. Because there is no answer. This is the only class of drugs, anesthetics, which don't have a target. Imagine if you are trying to develop a drug, you need to know the target and how it interacts with it and see how it works. But anesthetics are the drugs that don't have any target. So for a few years, by a mistake done by an undergrad student, we found the target of this anesthetic drug. And this sorry, there is no pointer. This target is a protein called G protein. Anyone heard about G protein? So this G protein is made up of 3 subunits, alpha, beta, and gamma. And we found that this anesthetics bind to the alpha subunit and it won't allow it to separate. Now I'll put it very simply and finish this story. Imagine you are lying on the bed as a patient. Imagine your surgeon coming and cutting your skin. When you cut your skin, this complex, 3-piece complex, separates into 2 pieces, and then you'll get pain because somebody is cutting your skin. But what we found was the anesthetic that we give binds to this subunit, changes the configuration, puts the subunit back into this resting stage so that the surgeon can cut the skin, no signaling is happening, and you won't feel the pain. And that's the story that we actually were able to discover. And interestingly, when we proposed this story for the first time, with lots of mistakes, we got $1.2 million grant. Because nobody knows how anesthetic works. And people thought this is the start of finding a target for anesthetics. Okay? So again, each slide is a story of 5 to 10 years. I'm trying to finish in like 2 minutes of each story. And this is the most important thing I want to tell you, this story. I love to take high school kids into my lab. We get lots of high school kids, of course, undergrad students, B.Pharm students. One of the high school kids who came into my lab, normally you write an email or call and take an appointment. And this girl barged into my office and asked me, "Are you Dr. Pentyala?" I said, "Yes." And she said, "I want to work with you." And I was thinking, who is this high school kid? I'm a professor in a big shot medical school. She barged into my office and she said, "I want to work with you." And I asked, "Who are you?" Okay. And then she said, "I am your teacher, I am your student's student." So her science teacher is my MS student. So I thought, okay, it's my grand student, so I'll entertain her. But, I mean, we don't entertain really high school kids. But I said, okay, come into the lab. I gave her a table. I made her sit in the table and I said, "Do whatever you want." I didn't see her for 3 weeks. And then after 3 weeks, my whole lab came into my office and said, "What did you do to Rachel? She's not coming." I mean, the girl's name is Rachel. I said, "I didn't do anything. I didn't even see her for 3 weeks." And you know what the lab people said? "We want her back in the lab." You know what Rachel did? Every day morning she comes at 7:30, cleans the lab, and started helping everyone in the lab to make a solution, to treat the cells, to actually dose the animals. Within 3 weeks, she took over the whole lab. And then when she stopped coming, my whole team said, "We are going to quit if you don't bring back Rachel." So I called Rachel and said, "Can you please come back?" You know what she said? "I'll only come back if you pay me." So I paid her some money because we had some grants. And she came and she actually had an argument with me saying that, "You are not giving me a science project. I want to do something, not helping others." Then I said, okay, here is your computer. You think about your own science project. Because that's what we do with the kids. Like here, we are PhD students. We give our projects to PhD students. There, we don't do that. We ask them to think and develop their own projects. So, to sum up everything, after just 2 weeks, she came to me and she said, look at this blue colored ring. This is a toxin called polychlorinated biphenyl. And she said, and it is very prevalent in Long Island waters where we live. It comes in the drinking water and it causes lots of bad effects. So what she said was, she looked at this compound, and in our lab we have a chemical called cyclodextrin. Okay? And look at the ring structure of the cyclodextrin and the ring structure of the polychlorinated biphenyl. And she said, as a high schooler, she doesn't understand chemistry. She doesn't understand good science maybe. But her thinking is, I'll take a big ring, the cyclodextrin, and trap the small ring, the PCB, and pull it out of the water. And I thought that is the most stupidest idea I ever thought about. Then, without telling me, she went to a computer science professor. She gave those structures, and she was able to prove by in silico docking that she can trap the small PCB with a big cyclodextrin ring. And then she came to me and said, Srini, this is what I did. Then I got excited because this is working now, right? So we did a very simple experiment. Somebody was talking about we need big equipment to do good science. You don't need any big equipment. What we did was we took a small test tube, put some water in there, contaminated with PCB. Then she added cyclodextrin on top of that. As you can see, the turbid water is with the toxin. When she added the cyclodextrin, everything got trapped and sunk to the bottom. And this kid got so many awards and prizes. And wherever she goes, as a mentor, I also go. And whenever she is applauded, they ask me to stand up, and I also say, as if I did everything. Actually, I didn't do anything. And that's what we want to encourage in young kids. And actually, after that, she went into environmental law. She interned at the White House, and now she is an environmental attorney in Washington, D.C. And just because we gave the kid a small opportunity to think, not just feed, she thought about this. Even though it might be simple, it can lead to great things, okay? And by the way, this project helped us to get another $1 million grant. A high school kid helped us to get a $1 million grant. Because in New York City is surrounded by a river called Hudson River. The Hudson River is contaminated by PCB and by a company called GE. The GE company is paying $250 million every year to purify the water in the Hudson River. So when we proposed this idea to the GE company, they said, use this and see if we can clean the water quickly. And that's the power of young minds. You need to think. If you just sit there not thinking, nothing will work. So another story is, you stop me please whenever you want, okay? So because these are stories, I can stop anytime. And this is another story. As a kid who was born and brought up in India, we are taught to respect elders. One day, a gentleman actually who retired, is a peptide biochemist. He used to come to all our lectures, and he used to stop everyone and bore them to death with his science. When he talks science, nobody understands. So one day he caught me, and he kept on talking for hours and hours and hours, which I never understand anything, because he is such at a high level. But after 3 months, I thought what he is talking is sense. So I called him back, and he and me, we developed a project together. I'm going to make it very simple how this project is. Imagine this is our protein. My hand is your protein. And when proteins do their function, they are cleaved, they are cut off in the cell. And these proteins are cut off in a sequential manner. That means they are not randomly chopped off. If it has to be cut off in the shoulder, then the wrist, then the finger. So what he is saying is proteins are chopped off in a sequential manner, and we can write a one-line algorithm to see whether we can find the small pieces like my black strap, and it can act like a drug. That's the whole story behind this. So what we did was, we actually identified 122 peptides. One of the peptide was able to cure Parkinson's in vitro. And actually, we collaborated with University of Lexington. They were doing monkey studies, where we treated a Parkinson's monkey with this peptide, and this 90-year-old monkey is now running like a 15-year-old kid. So we are on the verge of actually putting out a drug to cure Parkinson's. Now, here is another big story about what we are doing now. We were able to identify a peptide which is called osteogenic peptide. That means it can improve the production of bone. So what we did was, we took the peptide and we treated some bone cells with the peptide. That's the first panel. And to our surprise, we found that the bone cells are making more bone. And then we also took some stem cells and added this peptide that we discovered. And we found that these stem cells are now being transformed into bone cells. So with that, what we did was, if it is making more bone, can we heal fractures quickly? Anyone had fractures? Right? Ma'am, looks like you have everything happening to you. So when you have fractures, particularly in old people, they heal slowly. So what we did was, we took a rat, break the leg of the rat, obviously under all ethical considerations, and then we treated the fracture site with our peptide. And to our surprise, the fracture healed really quickly. And when we proposed this and asked for money for funding, they said, why don't you try working on osteoporosis? You know osteoporosis? Weakening of the bone. So what we did was, we took a female rat, we took out the ovary of the female rat. If you understand physiology, women who undergo menopause, their bones become weak. So if you take out the ovary, the rat will become osteoporotic. And we created an osteoporotic model, and we injected the peptide that we discovered into the rat. And for 35 days, the treatment was being done, and the rat's bone started becoming stronger again. This is the first osteogenic peptide that has been ever reported, because all the drugs that are there in the market to cure osteoporosis is to stop the breakdown of the bone. But there is no drug that will help to create bone. All the drugs will stop the breakdown, but we are the first one to report a molecule that can create bone. And this project is going on really, really good with a lot of funding. And here is another interesting thing for undergrad students, like your B.Pharm students. One day, one of my undergrad students, John, he came into my lab, and everyone stopped working in the lab. So I went into the lab, and he is showing a small machine. He has a small 3D printer gifted to him by his dad during Christmas, and he's showing off to everyone. And I grabbed the 3D machine and yelled at John and said, you're disturbing the whole work. And I took the 3D machine into my office and started working on it. And I got mesmerized. I called John into my office and said, how much did this 3D machine cost? He said, $800. So I thought maybe I should get a 3D machine for my lab. So we wrote a good project, and I'll explain to you again, 5 years of work in one slide. Our idea is to create a bone scaffold, like a skeletal structure of the bone, seed the scaffold with stem cells, treat the stem cells with the osteogenic peptide that we discovered, hoping that the scaffold will now become a real bone. And then we actually, how we did it? We took the CT scan images of the bone, created a scaffold, and seeded the stem cells. This is the first time ever reported that stem cells can be transformed into bone cell on an artificial scaffold. And actually, when we reported the first red-colored slide, the reviewers said, no, those are not cells. They are just dots. So we actually double-stained them to show that the nucleus and the actin fibrils are there in the cell. And we also did some scanning electron microscopy work to show that these cells are talking to each other and making bone. And now we actually tested this material in biomechanics, and we found that it is as strong as the real bone. And now we are planning to implant it into humans. So shifting gears, how much time do we have? You can stop me anytime, okay? Like I said, these are short stories. I want you to get inspired about these small, small things to see what you can think about. And this is now close to my heart because just before I came to India, I got a $250,000 grant on this. A simple story. Okay, how many of you had nausea and vomiting sensation? Anytime? Okay. Did you ever take a lime, squeeze it and smell it? You all do it, right? In India, it's very common. In US, nobody knows about it. So one day, my daughter when she was young, she was coming to us in the car, and she has motion sickness. My dad actually gave a lime and asked her to squeeze and smell. And that was the idea for this project. So what we thought was, if there is something in that lime rind, if I can isolate it and put it in a bottle and market it, I can make a lot of money as an anti-nauseating drug. So what we did was, for this story, it's a true story. I stole the grater from my wife's kitchen, bought some limes to the lab, and told the students to grate the lime rind. And we did some mass spec studies and isolate the pure aromatic D-limonene from the lime rind. And we actually encapsulated the D-limonene into nano beads, and these nano beads are put on a paper with a glue, and now you peel open the paper, you smell it, your nausea is gone. And when we proposed this as a patent to a university, our university jumped on it. Reason, where do you use this product? Anti-nausea, anti-chemotherapy. When you have chemotherapy, you have lots of nausea. You are going on a train, sometimes motion sickness. You're going on a ship, motion sickness. You're going on an entertainment ride, you have motion sickness. Janitorial staff who clean our bathrooms, nauseating smell, they all can use this product. Now we are doing clinical trials with this product, where we are now recruiting patients who underwent surgery, who are coming to the ICU, not ICU, PACU, where they recover. We actually give them this product and ask them to smell and see if their nausea is gone. And we are getting some good results, okay? So, we have moms. How many moms in the audience? Any moms, mothers in the audience? Okay, if you had babies, did you ever get an injection in the back called epidural? You all get epidurals, right? Epidural is given as an injection in the back to relieve your pain. So when that is given, anesthesiologist doesn't know where the epidural injection is going. If it goes beyond that region, then you have a mishap where the drug can go into the spinal fluid, and then you cannot deliver the baby because the whole drug is in your system. So to prevent this mishap, we found a marker called prostaglandin D2 synthase, and I'm just speeding it up. We cloned the gene, we expressed the protein, we injected it into mice and created antibodies, and then we created like a COVID test kit, we created a CSF leak test kit. So in that way, anytime you go into the delivery room, when an epidural is given, we collect the fluid and make sure the injection is given in the right spot. So that's one project. I'll end up with a couple of things, if you don't mind, because this is very interesting for the kids. Prostate cancer. Prostate cancer is only men will get it, obviously. So prostate cancer means the prostate gland becomes cancerous. How do you detect prostate cancer? This is a funny story. If you go to a urologist, they'll inject a finger into your anus and then palpate the rectum to see whether you have any tumors. Like your breast cancer. You palpate your breast to see whether there are any lumps. One day, one of my colleagues, Dr. Khan, he passed away during COVID, unfortunately. He came to me and said, Srini, did you ever feel a prostate tumor? I said, no. He dragged me to the clinic and he made me wear gloves, and he called a white gentleman, Bob. He asked Bob to lean on the desk and bend over, and he asked me to put my finger into his anus. because Bob had a big tumor. So when I put my hand into the anal region and palpated the rectum, I found the big tumor. That made my day because I'm working on prostate cancer, but I never touched a prostate tumor. So when I found that, it gave me more inspiration to do something about it. So what we did was, we took a prostate tumor tissue, grind it up, injected into a mouse, and the mouse created antibodies against this tumor. So we took those antibodies and screened lots of patient samples. And to our surprise, every time we get a designated patient sample biopsy, our antibody lights up the tissue. Not only that, we also found in the urine, you take a sample of urine from a prostate cancer patient, we stain it with our antibody, and the cells, cancer cells, will light up. While this is going on, another high school kid came into the lab. This will be my final project. Her job every day is to take human urine samples, which come in a cup, put them into a centrifuge tube, and spin them in a centrifuge, and separate the urine from the cells, and give it to our senior people so that they can do the analysis. So after 2 days, she came to me and said, Dr. Srini, I'm not going to work with you. Who are you? I mean, you are a high school student. Why do you don't want to work with me? She said, you and your lab stinks, because she is processing 50 urine samples a day. But none of my senior people, my MDs, residents, postdoc fellows, they never complained about it. But this high school kid came to me and said, I'm not going to work with you. And I got really mad at her. I said, get out of my lab. And after 2 days, I got a call from my chairman. She went and told on me to my chairman, to my boss. And my boss called me and said, Srini, why did you yell at her? And I told the whole story. And he said, encourage the kids. Try to make her understand. I'll give you $10,000 for this project. And I said, okay, let the kid come back. And I told this kid, you think about something that can relieve this urine smell if you want to work with us. Within 2 weeks, she came back. And she came back with a very simple idea. Normally, this is your urine cup. Her idea is, you put a strip in the middle, you attach your prostate cancer antibodies in the middle, you put urine sample of the patient here. When the sample is going down, the biomarker, the prostate cancer marker, gets strapped onto the strip. You pull out the strip, and you know whether it's positive or negative. Very simple idea. So initially we thought it wasn't work, but I actually was able to recruit a computer scientist, a 3D printing specialist. Look at the interdisciplinary work. And with them together, we generated lots of models. And finally, we came up with a model which looks like this. You collect the sample on the top, you put a cartridge in the middle, you slide it out, and then you can do any type of analysis in the urine. If you want to see whether there is albumin in the urine, you can see it. If you want to see creatinine, if you want to see bladder cancer, any type of thing. So with that, this kid actually did the pregnancy test kit. Normal commercially available pregnancy test kit, they can only detect up to a certain level. But with our kit, it can detect very low levels of HCG. So you can detect whether you're pregnant in the first 2 weeks of your fertilization. Whereas the regular pregnancy test kits, it takes 4 weeks. So not only that, the most interesting thing was, if you smoke cigarettes, I can catch you. What we did was, we coated that strip with an antibody against a substance called continin. When you smoke a cigarette, the nicotine in the tobacco is converted into continin and comes out through your urine. But we made a strip that can detect that continin. So if you are smoking, for example, I think girls won't smoke in India, but in US, lots of girls also smoke. So if you smoke a cigarette, you come home, and your parent will give a cup that is designed by others. And then you pee in the cup, and within 2 minutes, you pull out the strip. If the line comes up, we know that you are smoking. And this is now taken up by insurance companies. because insurance premiums go up if you are a smoker. So that's another interesting project. And January 2023, a patent was issued on this. All these are patents. All these are intellectual properties. And if you can see really the names, can anyone read the name? Can you see the name here? Anyone? I'll read it. The first author is Srinivas Pentyala, which is me. The second author is Sahana Pentyala. It's my daughter. She is the one who went and complained to my chairman, and I got yelled by my boss. And she's, I mean, who has the audacity to go and tell about their professor, right? But she has the guts, and she went and complained on me. But I challenged her, and because of that, for 2 years, we are constantly fighting in the lab and at home. And my wife took my daughter's side, and sometimes I didn't even have food sometimes. But that's a success story. It's one of my most privileged moments in my scientific career, having a patent together with your daughter. And actually, it's her idea, but I stole the spotlight a little bit by sharing the idea, okay? So with that, I think I should stop, right? Do you want me to stop? I don't know. Okay. Maybe final. Another interesting, very simple, you want me to stop? Okay, maybe final. One day, actually these are all 3 real stories that happened to me or my colleagues or ideas given by my students. A few years ago, 3 years ago, I had some pain in the wrist area, and I think I know everything about orthopedics because I'm one of the faculty members. I have a joint appointment. I thought it is a sprain, I thought it is a fracture, I thought it is a gout, I thought it is a pseudo-gout. Then I went to my orthopedic chairman, and he said, it's nothing, it's just a sprain. But he said, why did you think that it is gout? You know what gout is? When you build up uric acid in your body, it crystallizes and forms in the joints. The crystals will accumulate in the joint, and the joints will fuse, and you get gout. And there is no diagnostic method to detect gout rapidly in the clinic. And that's the challenge given to me by my orthopedic chair. I didn't even pay attention to it. And after 2-3 months, I got a stupid idea. I don't know whether you do it in your chemistry, but when I was doing my undergrad chemistry in AC College in Guntur, we used to have an experiment where we actually stain uric acid with silver nitrate. So when uric acid and silver nitrate come together, the uric acid turns black. And that's my BSc second year experiment in chemistry. So I took that idea, and then we collected the joint fluid of a gout patient, and then we put that fluid on a silver nitrate bed. So the joint fluid of a gout patient have uric acid crystals. When it touches the silver nitrate, it turns black. Within 1 minute, I can detect whether you have gout or not. And with that, I thought this is a good project to get some money. So we developed a very, very innovative technology, combining iPhones and gout detection together. So we take our iPhone, attach a small microscope to it, and when we get a patient sample, we put the sample on a silver nitrate bed and take pictures. No need for microscopes, no need for trained technicians. Anyone can detect gout rapidly in less than 1 minute. The good thing about this project is when we wrote the grant, we asked for 10 iPhones for all my students. And my students are very happy because they only use it in the lab for 1 hour, and for the rest of the day, they can take it home and use it as an iPhone. So some things good come out sometimes too, okay? So these are the crystals we were able to find with the iPhone. You don't have to send it to the lab and wait for two days. So what I'm saying is, think beyond. When you look at your bench to bedside and bedside to practice, we are taking technologies that we discover in a basic science lab to the clinic and implementing in a clinical practice. And that's what translational research is. So translational research can go both ways. Either you can start at the basic science and go to the clinic, or look at the things that are happening in the clinic and come back to basic science and try to understand this, okay? So my job is to bridge the divide between the basic science and the clinical practice. And that's why we are called translational researchers. With that, I'll stop here. Thank you. Any questions?
Thank you very much for your presentation, sir. After attending your session, I'm feeling very proud. I'm feeling I'm very luckiest person as I have got so many points from you. I have so many doubts, but you covered all the topics. As neurology you covered, biomedical devices you covered, so many projects. Whenever I'm seeing your name, I want to title it as a large titles, small stories, great insights. Thank you very much, sir. Thank you very much. If any questions from delegates or students?
Any questions from the students or the delegates?
Stay back for a few minutes, sir. It's time to honor our eminent speaker, Dr. Srinivas Pentyala sir. In this regard, I would like to invite patron of this international symposium, Dr. U. Shaurili Garu, onto the dais. I would like to call upon patron of international symposium, Reverend Sister G. Nirmal Jyoti Garu, onto the dais. And convener of this international symposium, Dr. B. Pamul Reddy sir, onto the dais. I request Dr. Hare Krishna Roy sir join us onto the stage. I request co-chairpersons and chairpersons of this session onto the dais, please. Thank you very much, sir, for your valuable session over today. Once again, thank you very much, sir, for your interactive, valuable, knowledgeable session. It's time to honor our chairperson, Dr. Surya Prabha Madam. Thank you, Madam, for coming to the International Symposium. It's time to honor our co-chairperson of this session, Dr. G. Sopna Madam. I thank all the dignitaries, eminent speaker, chairperson and co-chairperson of this session. Thank you all.
Thank you so much, the dignitaries, for the proceeding you have done. Thank you so much, sir. I apologize for cutting short your time. I took 30 minutes. No, sir, I apologize, sir, from my end. It's my constraint.
Thank you very much, sir.
Without any delay, we'll be going to the last offline session for today. So post-lunch anyways, we'll be having online sessions here itself. So we'll be continuing with The next session, the speaker we'll be having today here is Dr. Jitendra Patel, sir, from Datta Meghe College of Pharmacy, DIMA chair, deemed to be university from Wardha, Maharashtra. For this session, we have the presiding members, Chairperson Dr. Hema Kumari and Co-Chairperson Dr. Vijay Kushore. I request the Chairperson and Co-Chairperson to please honor your chairs. Thank you so much. Before we start today's final session, let me take the opportunity of introducing our co-chairperson to all of you. He is very well known to everyone. He has been visiting to our institution all the time. We have Dr. K. Vijay Kishore. He is working as an assistant professor in the Department of Pharmaceutical Chemistry in University College of Pharmaceutical Sciences, Acharya Nagarjuna University. He has teaching experience of 14 years. He has published research publications in various journals, international and national. He has 4 book chapters to his name and authored a textbook for pharmaceutical medicinal chemistry. He holds various academic positions in Nagarjuna University. He is the assistant coordinator, Board of Community Development through Education. He is the deputy chief warden of Boys Hostel in Nagarjuna University. He also served as coordinator of sports in ANU and coordinator for examinations of ANU College of Pharmaceutical Sciences. Dear Vijay Kishore sir, it's very pleasureful for you to have you here with us. I request you to come on to the dais please to introduce today's chairperson.
Thank you, ma'am. Thank you for your introduction. Now I take the privilege of introducing my chairperson of this session, Dr. N. Hema Kumari, designation Associate Professor and HOD, Department of Pharmacy Practice, Hindu College of Pharmacy, and her total teaching experience is around 14 years. And she has publications in various reputed national and international journals, and she has guided nearly 20 PharmD and B-Pharmacy projects. Ma'am, it's up to you.
Thank you so much, sir. A very good afternoon to all. I would like to take an opportunity to express my gratitude to the organizers for giving me this opportunity to act as a chairperson to this session. I take it as my privilege to introduce the speaker of this session, Dr. Jitendra Patel, Associate Professor, Datta Meghe College of Pharmacy, Wardha, Maharashtra. Sir did his B-Pharmacy from Peters Ravishankar Shukla University, Chhattisgarh in 2007. Sir completed his M-Pharmacy in the Department of Pharmacognosy, Biju Patnaik University of Technology, Odisha in 2009. And Sir has completed his PhD in Pharmaceutical Sciences, JNTU Hyderabad, from the University JNTU Hyderabad in 2017. Sir has a total experience around 16 years of teaching experience. He worked as assistant professor in Bharat College, Hyderabad from 2009 to 2015. Sir worked as associate professor and HOD, KVK College of Pharmacy, Hyderabad in 2015 to 2018. Associate professor, Avanti Institute of Pharmaceutical Sciences, Hyderabad from 2018 to 2021. And now Sir is working as associate professor, Datta Meghe College of Pharmacy, Wardha from 2024 to till date. Sir is awarded with Talent Award from Educational Minister Chhattisgarh in 2021, Young Scientist International Travel Grant in 2016, Conference Grant recipient CSIR New Delhi, and Conference Grant from DST deemed to be Datta Meghe College of Pharmaceutical Sciences and deemed to be in university.
Sir is having more than 40 research and review articles publications with H-index 9 and research interests nanotechnology for hepatoprotective activity and patents. Sir has various patents and he wrote many books also. With this brief introduction, I invite Dr. Jitendra Patel sir to continue the session. Thank you very much.
I just want to play the video for my organization so that
Enter a vibrant world built to inspire curiosity and empower young minds to take confident strides into the future. Datta Meghe Institute of Higher Education and Research is the vision of a tireless trailblazer, Shri Dattaji Meghe. Education and health are the twin pillars that can truly transform the future.
No benchmark is expected to be such which will be remaining unattainable for the onward march of this university.
DMI HER is a deemed university offering over 270 multidisciplinary programs to more than 10,000 students. Away from the hustle and bustle of the city, the campus is a self-sustaining complex. It also has an off-campus college at Nagpur. The infrastructure-rich, full-fledged medical colleges along with large hospitals facilitate hands-on learning. Allied sciences students have an array of non-medical streams to choose from. The students benefit from leading national and international collaborations and the opportunity to partner with some of the world's best institutions. Exchange programs and combined research initiatives empower the students to grow and gain greater recognition. With a proven track record of graduates and postgraduates in 25+ countries, DMI HER students are prepared for a global career. The environment is gender equal and rich with cross-cultural interactions with students from numerous countries and continents coming together. The campus offers a complete life to its students with facilities for sports, workouts, simulation labs, research house, literary society, new age auditorium, NSS activities, and more. With 661 plus full-time faculty, 795 plus PhD scholars, and 48 plus fellowship students, DMI HER offers one of India's most leading and evolving research ecosystems. Highly advanced facilities, grant opportunities, seed fund support, co-working space, advanced computing facilities, research guidance, publication help group, fabrication laboratory, innovators mentorship, and startup ecosystem are some of the key aspects creating a holistic research and entrepreneurship culture. DMI HER also offers a world-class incubation center where winning ideas are already taking shape. At DMI HER, a fully dedicated center has been created to empower students to make winning choices. Be it career, internship, fellowship programs, research programs, taking next steps into the future, or soft skills training, the center is here to partner students. New age and technology-led learning environments have been created for students to understand concepts in an interactive manner. The simulation lab infrastructure rivals the best globally with real human-like procedure experience. DMI HER has undergone multiple cycles on NAC re-accreditation and has achieved the highest grade in all cycles. We are proud of the institution that we have meticulously built. Come, be a part of this ever-evolving journey at DMI HER.
Thank you. Yeah, so in last over, I have to make 10 runs, something like that. Okay, so most of the part, it has been completed by the eminent speaker. So I will just try to summarize those things. Basically, we are talking since last many series about the nanotechnology, right? So when we talk about nanotechnology, it is clear up to now that the particular active ingredient or active moiety, we want to make it in the small particles or small size so that we can enhance the maybe solubility, maybe bioavailability, or maybe therapeutic efficacy, right? So I just want to streamline my area where the nanotechnology, like how it is nature to nano. So when we talk about nature to nano, I will take it in the respect of liver diseases because it is my area. So basically, when we talk about liver, it is the largest organ of the body, which is when we talk about any metabolism or any kinds of first pass or trademark which body accept, like it has to go inside and whether it is suitable for the body or not, like that thing. So liver is the organ which is approving that whether it is right or wrong. And millions of people, they are affected by the liver disease or liver disorder worldwide. And there is a challenging what kind of medication has to go on, and so many research it is going on, like how we can enhance when we talk about the natural products, right? So most of the natural products is having the problem like maybe solubility problem, maybe bioavailability problem, right? So to overcome out of those problems, we need to go for these kinds of advanced formulations, right? So basically that only I will cover up, right? So when we talk about the liver damage, it may happen by the so many reasons, right? It may be biological, maybe by the chemicals. The chemicals may be whatever the drugs we are using, say for example in anti-tuberculosis, right? So in the case of TB patient, they need to have the drug for maybe for 6 months, maybe for more than that. So when the continuous consumption of those kinds of medications, definitely it is going to impact onto the hepatocytes, and the hepatocytes may damage, right? So in that case, maybe biological, maybe virus, or it may be any pathogens, like on physical cause, maybe trauma, or maybe some radiation exposure by the particular individuals. So these are the main various reasons where and there are certain identified plants, because I am talking here about the natural to nano, right? So nature is given us so many plants which has already proven the hepatoprotective activity. Here are the few examples. Maybe silymarin is a very good example for in the case of hepatoprotective. Apart from that, there is a turmeric where the curcumin is playing the role, licorice, and maybe ginger, ginseng, then even in the Phalanthus niruri, green tea and endography. So these are the reputed, well-established and already reported hepatoprotective plants, right? So whatever the research has been carried out till today, it was done into the crude extract, right? Maybe different part they have taken and it has been screened into the different animal models, right? So when we talk about the hepatoprotective, it may be the multiple approach, maybe from the nanotechnology, what we are talking here, right? When we are trying to reduce the active ingredient into the smaller size, we can enhance, and in the genomic level also there is the CRISPR and the DNA-like technology on in the mechanistic side. We can change the cell signaling, and in likewise we can have the impact onto the antioxidant defense or maybe in anti-inflammatory, because these are the way where the liver is getting damaged, right? So when we talk about how the things happen, like when we are taking the pharmaceuticals in the different form, maybe synthetic drug or maybe some kinds of drugs, so they are definitely during the metabolism they are going to impact onto the hepatocytes, or maybe viruses. As I told you, the biological cause also may be damaging the liver. So these are the things, trauma, fatty liver. So what is happening, like cytokinin release, which is the inflammatory signal, that is the one cause. Maybe necrosis, where the accumulation of the some fatty materials is happen into the hepatocytes, then osteosis, apoptosis. So these are protein misfolding, right? So in this way, liver is getting impacted, and there will be the challenges to overcome it, right? So oxidative stress is one of the main mechanism of the hepatotoxicity. So how the oxidative stress it happened, like maybe it may be the internal triggers or it may be the external trigger. When we talk about internal triggers, it may be the mitochondrial activities, maybe inflammation or maybe cellular the system, and they are going to damage the maybe DNA, or they will cause the lipid peroxidation and protein degradation, right? So there are certain way where the nanoparticles, they are helping us to enhance the efficacy. Maybe when we talk about the liposomes, so liposomes generally it helps us for the absorption of the active ingredient or enhance the bioavailability. When we talk about the solid lipid nanoparticles, they improve the stability and during the life cycle of the products, and polymeric nanoparticles, they are useful for the controlled release when we want drug to be released whenever required and however required. Nanoemulsion is for the enhance the solubility and dispersion. So each and every formulation, they are having their own advantage, and likewise when we are delivering the active constituents to the liver, whenever the liver problem exists, we may get the better result and better therapeutic efficacy also. So there are some case study onto the natural products. It may be from the, when we talk about the garlic nanoemulsion, if we are going through, so it may have the antibacterial property, and which has already proven by the various researchers, and even in curcumin, whenever curcumin, which is obtained from turmeric, it will be incorporated into the nanoparticles. The anti-inflammatory or anti-cancer efficacy can be enhanced, and it will help us, like, and sesame polymeric nanoparticles. So likewise, whenever it is going for this certain advanced formulation, it is giving the better result. And genomic instrument and technology is being used in the research for the nanotechnology. Like we are using the genetic variation analysis, and the transcriptome profiling also may be done, and gene expression measurement for the targeted therapy and personalized medicine. My previous speakers, they have already mentioned that the individual persons required individual types of the treatment, so that can be possible based on the problem exists onto the. And synergistic effect, one of the important terminology which is very important in the natural product case, like whenever we are using certain pharmaceuticals with the natural products, we can expect that one constituent may increase or decrease the another constituent effect. So whenever it will come into these kind of formulations, we can get the unexpected result which may be beneficial also, right? So determining the mechanism exactly involved, what kind of mechanism it is there, that is one of the difficult tasks actually. It's not that the nanotechnology or nano formulation will always give you the better result. There are the research reported already by certain researcher that nanotechnology also giving the some toxic effect, right? It may damage to the cell in the different ways, like it can also, you can see the mesoporous nanoparticles also involved in the endocytosis, and micelle also passive diffusion, whereas the cell membrane disruption can be seen. Here also you can see the interaction of nano materials with the cellular organelles and how it is going to damage the certain, whatever the things we are expecting always into the nano formulation, that may not be always true. It depends on the case to case and what kind of ingredient has been taken to formulate the nano formulation and what kind of mechanism is involved, right? So like that, you can see in liposomes, whenever the nanoparticle it is going, it may leads to the endocytosis, whereas in mitochondria it may also leads to the generation of reactive oxygen species, which is actually which we don't want actually, right? Whenever we are going for such kind of formulation, so which the different content of the cells, it may have the different types of interaction that also need to analyze in the coming research, right? So you can see the pathway of oxidative stress of the whenever whatever the reported paper, they are showing always that it is the reducing the reactive oxygen species, reducing the oxidative stress and reducing the inflammation. But it may not always true that it may also involve into the apoptosis, fibrosis and inflammation. In the case of liver related formulation, we are expecting all the things should not happen. That is the target, but there are reported paper which suggest that it may also impact severely to the hepatocytes. So what can be the measure for the risk assessment to the nano formulation? So there are the exposure route, we need to analyze whether it is inhalational or dermal contact or ingestion or maybe the kind of thing. Toxicity evaluation is very important. Whenever we communicate such kind of paper, they generally reviewers suggest that what is the toxicity profile and what is the regulatory hurdles, right? So that kind of review generally we get because of these reasons, because nanomaterial characterization supposed to be done. In vitro toxicity test definitely has to be done, not only for the active pharmaceutical ingredient, even for the formulation. And in vivo toxicity study, generally it has to be done because by following the OECD guidelines, and whatever we do for the normal extract, that has to be done for the formulation also by following the regulatory guidelines, right? But bioaccumulation studies also need to be done. And risk assessment is very important because hazard identification, what kind of things, dose response analysis, that should be also analyzed, exposure assessment and risk characterization, that has to be done. So I will summarize with this all, like phytomedicine potential. We know that there are formulations available into the market and it is already reported by the certain researchers. It may not be always beneficial. We need to go for the proper analysis, assessment of those formulations so that we will be able to have the proper formulation which can give you the proper hepatoprotective activity. Thank you.
Thank you, sir. Thank you so much for enlightening our students by giving a great lecture regarding the natural products that are useful in hepatoprotective activity. Thank you very much, sir. If anybody are having any doubts, you can ask sir.
As sir rightly pointed out in the start of the session, sir, you have scored half century in the last over. It was well explained, sir, and it's very informative.
If no further questions, we will try to wind up the session. Sir, thank you so much. Please stay back for a minute, sir. Now it's time to show a small token of gratitude towards our eminent speaker, Dr. Jitendra sir. Sir, we are extremely happy for having you here with us. Now I request the chairperson and co-chairperson to come on to the dais for the felicitation of the speaker. I request Principal sir, Shishir sir and Ashmita ma'am to join the same. I request Roy sir to join, please. I request Dr. Jitendra sir to please honor the chair. Sir, we are extremely happy. Thank you so much for all the knowledge you shared with us. I request Dr. Amit sir to join the dais, please. It's a pleasure to have such wonderful sessions, sir. Thank you so much. Thank you, sir. Give a big round of applause. It's an extreme honor for us to have such eminent personalities amidst us today. Now it's time to honor the chairperson of today's session, Dr. Hema Kumari Madam. Madam, please accept a small token of love and gratitude from our institute. Thank you, Madam, for your time and interest in our institute. We hope to have your collaboration in future. Thank you, ma'am. Now it's time to honor the co-chairperson of today's session, Dr. Vijay Kishore, sir. So please accept a small token of love from NCPA. Thank you so much, all the dignitaries. Thank you, all the dignitaries. You can move off the dais, please. Give us just a couple of minutes.
হ্যালো হরি হরি এটা ধরো তুমি ধরো হরি ভিডিও ভিডিও অন করো আমি তোমাকে ইন্ট্রোডিউস
Post lunch, we'll be having the poster presentation. Students can go to your respective places. Immediately after your poster presentations or oral sessions, you can come back here for the continuing sessions.
Hello. In this scientific session, our honorable speaker is Dr. Nebojsa Pavlovic, Department of Pharmacy, Faculty of Medicine, University of Novi Sad, Serbia. In this opportunity, I would like to introduce Honorable Chairperson of this session, Dr. Ashmita Samadar, University of Kalyani, West Bengal, India. So, Dr. Ashmita, please introduce the respected speaker for this scientific session. Thank you.
Thank you, Professor Nandi. It's an honor for me to introduce our esteemed speaker of the session, Dr. Nebojsa Pavlovic, who is an assistant professor and teaches courses in pharmaceutical technology and pharmacy practice at the Faculty of Medicine, University of Novi Sad, Serbia. His main research interests are novel drug delivery systems and technologies in pharmacy. He is a participant in several projects in national and international level, including 2 Horizon Europe projects, Medilem and CellSet, 1 Cost Action, Enable, and 1 project funded by the US Agency for International Development, Momentum. He also conducted 1 educational project funded by International Pharmaceutical Federation, FIP, in 2022. He has published more than 50 articles in peer-reviewed international journals with more than 1,000 citations in the Scopus Index and Hirsch Index of 19. He worked as a visiting research faculty at Faculty of Pharmacy, Curtin University, Perth, Australia in 2017 and 2023. Besides, he attended 1-month seminar, that is Seminar on Pharmaceutical and Medical Investment and Cooperation for Developing Countries, organized by Ministry of Commerce of China and Company, Sinopharm, in 2016. He was awarded several grants by international scientific societies. In 2019, he obtained the prestigious award Dr. Zoran Dignic for the best researcher in autonomous province of Vojvodina, Serbia. In 2022 and 2023, he was awarded for excellence in science for the highest cited researchers in autonomous province of the state. Besides, he was awarded by Faculty of Medicine, Novi Sad for achievements and results both in science and teaching. We welcome the speaker on board.
You can see my presentation. I hope everything
Nebo,
is right.
Dr. Nebo, you are mute. Can you please unmute?
Okay.
Dr. Nebo, you are muted. Please unmute.
Just a second.
Our esteemed speaker, your microphone is muted. You are not audible. You are not audible. So can you unmute your microphone, please? Dr. Nebo, you are requested to unmute your microphone.
Do you hear me now?
Yes, yes.
Great. I will share my presentation if it's okay. Is it okay now? Do you hear me now and do you see my presentation?
Yes. Hello? Yes, it is. Please.
Excellent. Thank you very much. I feel honored to be here today and to give my lecture. I would like to thank to the organizer for this invitation, especially to Dr. Roy and Dr. Nandi. And I would wish even better if I was in person in beautiful India now, but hopefully some next time. My topic is pretty wide, and I will try to focus on some main results of my research, but it's mostly based on drug pharmacokinetics and investigation of pharmacological activities of drugs. This lecture is divided in two parts. First part will focus on in silico methods in assessment of drug pharmacokinetics, but also pharmacodynamics. And the second part will focus on the influence of gut environment, especially microbiota on drug pharmacokinetics. Those are two main projects that I've been involved or I was conducted in the recent years. First, let's say that pharmacokinetics is a study how organism affects drugs. So not how drug affects human organism, but the opposite, which means how the drug is absorbed, distributed, metabolized, and eventually how it's eliminated from the body. Of course, all drug candidates that should be commercial drugs one day should have acceptable pharmacokinetic profile. If so, pharmacokinetic data enable us to have the optimal dosing, efficacy, and safety of the drug, and of course with the final aim of having successful therapeutic outcomes. Generally, when we talk about in silico methods, they're very important in all phases of drug development process, and its main goal is actually to select only the most promising drug candidates that should succeed in some later stages of drug development. So-called high throughput screening. Sorry, I hear some echo, so that's why I thought that maybe someone else said something, but I will continue. So yes, for in silico methods, it's very important that we filter from large chemical databases and select only those most promising chemical compounds. The basic assumption of in silico methods in drug development is that structurally similar chemical compounds have similar activity, which gives these methods predictive character. And it's very important, and we can see that it's a hot topic in science for more than 20 years, and even from 2003, one of the first publications on this topic was asking, is this pharmacokinetic in silico modeling, is it some kind of prediction paradise? Nowadays, we can say that definitely in silico methods are very important in drug discovery and development process. However, they did not manage to completely replace other methods like in vitro, in vivo studies. They said it's a very wide topic. There are many books on these topics, and I just wanted to show that many aspects of drug discovery and development are can be a part of in silico investigations, from pharmacokinetic assessment and prediction, drug activity, toxicity, etc. When we look also at the number of publications, we can see in the last 5 years there is a sharp increase in number of publications, with more than 10,000 publications per year about in silico drug development, especially when we look at the subject areas, mostly pharmacology, biochemistry, medicine. When we look about geographical regions dealing with this topic, there are developing countries, so really everyone is dealing with this important topic. There are really many in silico methods. They can be divided into two main categories: ligand-based, that we identify some functional groups that are important, for example, for some pharmacological activity, and the second ones are structure-based methods. The main example is molecular docking technique, where we explore the mechanism of binding some potential drug molecule with pharmacological target, with some protein, either it is an enzyme or transport protein or something else. As I said, the main goal of in silico methods is to identify the most promising drug candidates, and I'm very happy about these publications that we had in 2019, that we actually tested structural analogs of resveratrol and identified oxyresveratrol as the most promising analog that could be used in metabolic disorders, especially obesity and similar metabolic disorders, because they act on PPAR gamma receptor. And I'm happy because the next year, a group of Chinese scientists actually confirmed our results and experimentally, and show that there is anti-adipogenesis effect of oxyresveratrol mediated through this receptor that we investigated. But the focus of this lecture will be on another naturally occurring compound. It's quercetin, flavonoid that is widely present in vegetables, fruits, etc. It's lipophilic molecule that we can see its structure. It has well-established, in the first place, antioxidant and anti-inflammatory activities, but I will focus here on anti-cancer and neuroprotective effects. What's the problem with quercetin? In its ugly form, without sugar moieties, etc., it has really low aqueous solubility, and in that way it has poor oral bioavailability. Another thing very important is that even that small fraction that is absorbed in the intestines, the really small fraction of that can pass through the blood-brain barrier. So yes, there is much evidence that quercetin can exert neuroprotective effects, but actually its concentration in the brain is really small. That's why we conducted two different studies. I will not explain all the details because we don't have enough time. But first, in 2017, it was experimentally demonstrated through X-ray crystallography that quercetin is a ligand for one protein, urokinase type plasminogen activator, that there is a scheme how it's involved in angiogenesis and cancer metastasis, but it's important nowadays pharmacological target for anti-cancer drugs with anti-metastatic activity. What we did, we explored around 50 quercetin structural analogs and identified several analogs of quercetin with better UPA inhibitory activity, which means that it would be it would exert even more pronounced anti-metastatic activities, and with improved pharmacokinetic properties when compared to quercetin itself. The most promising drug candidate is this one. You can see the structure. Very interesting when you look at these structures. Actually, it's very similar to quercetin, but the number of hydroxyl groups is the same, but their arrangement is different, and that stereochemistry of this molecule actually leads to better water solubility and on the other hand, because of their arrangement, amphiphilic moment is higher, so it actually also has better membrane permeability. Another study is also very important since it was experimentally demonstrated that quercetin can act on some multi-kinase in the brain, that enzyme is very important in in progression of neurodegenerative diseases. As I said, quercetin itself has very poor intestinal absorption, but also permeation through the blood-brain barrier. We did several in silico methods. We don't have time to speak everything about it. It was published last year. This study, blue circle, this is this diagram. The figure is so-called boiled egg figure, and in the gray area, there are compounds that have very low intestinal absorption. In the yellow, the white part is with acceptable intestinal absorption, but yellow part are the compounds with good brain distribution that pass blood-brain barrier. Quercetin itself is the blue circle that is close to this gray area, so it's on the edge between a low and good intestinal absorption, but none of the tested around 50 compounds has good blood-brain barrier permeation, but some of these structural analogs are very close to this yellow part. What it means, that's why we performed principal component analysis. We actually studied the influence of different molecular descriptors on blood-brain barrier permeation. And we actually saw the 4 compounds with the best brain distribution grouped together. Why it is important? Because it can be used as a guide for medicinal chemists to synthesize even better quercetin analogs based on these molecular descriptors that will have in future much better blood-brain barrier permeation. So yes, this whole principal component analysis is very important for future synthesis. Another part of this lecture is focused on the influence of gut microbiota on drug pharmacokinetics. For me personally, it's very important. It's very interesting topic because when we talk, for example, about drug metabolism, we always talk about the liver, but we forget about 2 kilograms of bacteria in our bowels that have such a strong metabolic capacity, we should have in mind that composition of gut microbiota differs between different people, so it can contribute largely to individually different drug responses. That's why one of the strategies that emerged is that oral co-administration of probiotics with conventional drugs can actually modulate microbiome and diminish that inter-individual variability in drug response, so that we have more uniform drug response in different people. There are several studies that show that co-administration of probiotics can change drug pharmacokinetics with different drugs. But when we talk about that, we should not forget the influence of bile acids. When we talk about gut bacteria, it's always in some complex network with bile acids, because bile acids control the growth and the composition of gut bacteria. On the other hand, gut bacteria metabolize bile acids and create that complex network that so they both individually and together influence, for example, drug pharmacokinetics. Bile acids are not just physiological surfactants that aid in lipid absorption. Nowadays, it's well known that they act also like signaling molecules acting on nuclear and membrane receptors, and we focused on two natural bile acids, cholic acid and deoxycholic acid, that differ only in one hydroxyl groups, but it contributes to different behavior in vivo. Bile acids are very important for us because they have many absorption-enhancing mechanisms. The main mechanisms are they can act both on the drug molecule itself, for example, through ion pairing. It's very important to say that bile acids can improve absorption of both lipophilic and hydrophilic drugs. On the other hand, bile acids can actually exert their effects on biological membranes through increasing their fluidity of phospholipids, etc. So it's a really complex topic, but very important for drug pharmacokinetics. We focused in our studies, several studies, on glycoside as very lipophilic oral antidiabetic drug that is weak acid, but we focused on this drug because it has really wide inter-individual variations in oral bioavailability. Those variations cannot be explained only by genetic variations, but also it suggests that there is a contribution of gut bacteria on drug pharmacokinetics and in the final stage drug response. So we wanted to determine the effects of probiotic bacteria and bile acids on glycoside absorption and to evaluate the possible impact of bile acid structure on that drug transport. We used several probiotic bacteria and we tested absorption of glycoside alone or in combination with probiotic bacteria and in combination with two bile acids, cholic and deoxycholic acid. We used in vitro pampas system with artificial membrane at 3 different pH values representing different parts of our intestines and incubated up to 6 hours. We measured the glycoside concentration both in donor and acceptor compartment in order to calculate apparent permeabilities. In another study, we tested if glycoside is accumulated or maybe metabolized by probiotic bacteria. And we also tested glycoside bile acid interactions through molecular mechanics force field calculation to see if they really form some complexes. When we look at the concentrations of glycoside in acceptor compartment, that's the fraction that actually was absorbed, we can see that higher pH values, probiotic bacteria actually significantly improved glycoside absorption, that absorbed fraction, and the bile acids could diminish partially that effect. When we look at concentration time profiles of glycoside, we can see that after 6 hours, the influence of probiotics is significantly, concentration significantly higher than in the testing groups without probiotic bacteria. We also calculated the permeability coefficients, and we can see, for example, that the most relevant pH value, 7.4, actually probiotics increase 3 to 4 times absorption of this anti-diabetic drug. This is very important result of our study because it shows that when we calculated the total glycoside mass after 6 hours of incubation, that in the group of glycoside alone with probiotic bacteria, we, let's say, lost some part of glycoside. What can be the cause? So maybe the glycoside accumulated in bacteria or it was degraded metabolically by probiotic bacteria. That's why we conduct, but before another study, we also tested through molecular mechanic calculations glycoside bile acid interaction. And we saw that the potential energy of these complexes is lower than the sum of energies of individual compounds, which indicates that creation of these complexes result in stabilization of the system, especially in the case of deoxycholic acid. That's why deoxycholic acid had more pronounced effects in our study than folic acid. In another study, we tested glycoside penetration or accumulation into probiotic bacteria, and in the right figure, you can see that pretty high concentrations of glycoside inside bacteria. So we experimentally confirmed accumulation of the drug in probiotic bacteria, while deoxycholic acid could diminish that effect. When we also look at the total concentration, intracellular plus extracellular, we can see that this green line, which represents glycoside plus probiotic bacteria, that again, some part of the glycoside is missing. But because it's both in intracellular, extracellular compartments, so definitely we confirmed that a part of glycoside was degraded in probiotic bacteria. That's why we tested glycoside biotransformation through the use of two softwares. One, this is MetaPrint, that shows in different colors the probabilities of different metabolic reactions, and another one, EAVEC, that let us identify 5 potential metabolites that can be formed through metabolic reactions through the activities of enzymes that exist in probiotic bacteria or in gut bacteria in general. In conclusion, we definitely demonstrated that probiotics increase the permeability of glycoside across the intestinal membrane. Bile acids could decrease that permeability through both intestinal membrane and bacterial plasma membrane. And very important, we experimentally showed that definitely one part of this antidiabetic drug is metabolically degraded within probiotic bacteria. Altogether, we can say that significant interaction between probiotics and bile acids with glycoside may have influence on the bioavailability of glycoside and, of course, eventually on the drug response. But it also opens some new pharmacological strategies because each individual has a specific bacterial fingerprint that nowadays is not so difficult to determine, so we can know our bacterial fingerprint. It opens up new possibilities of therapy individualization for probiotic supplementation that would be adequate for our gut bacteria composition. In the end, I would like to thank you for your attention, and of course to thank to all the funders of these studies. You also have, if you have any questions, feel free to ask. You also have my email address here, and if you have any thoughts, ideas, I would happy to. you can always send me to discuss further these topics. Thank you once again.
Hello. Hello. Thank you very much, Nebo. Dr. Nebo. Hello. Can you hear me?
Yes.
Hello.
Yes, yes.
Interaction.
Coming, sir. Thank you.
Thank you very much for your bright presentations on in silico pharmacokinetic modeling, taking structure-based drug discovery, pharmacophore modeling. Hello, Nirmala College of Pharmacy is muted.
Yes, I didn't hear the whole questions, just the
Okay.
parts.
Hello.
Yes.
Can you hear me, Nebo?
Yes.
Okay. And I am very happy that you are with us today in this scientific session. You are honorable speaker. You are my very good friend. Our young scholars, young faculties have been benefited a lot. Now, I would like to give this session to honorable chairperson, Dr. Asmita, to please.
Thank you, Mr. Vedo. Both Professor Nandi and myself, we work in collaboration where we undertake in silico as well as in vivo and in vitro techniques to, for creating some treatment strategies using phytocompounds or some other synthetic compounds. It's again muted.
There was some echo, but now I think I hear you better now.
So we thank you for hello. We thank a lot on behalf of Nirmala College of Pharmacy and organizing committee. We thank you for your great talk, and I think all the students and all the audience this August gathering has been benefited by your talk, and we expect potential collaboration from your end with this institute so that we can go for an academic excellence with the students and the upgrowing of the future researchers together from your end and from our end. Thank you so much.
Thank you very much.
Now this session is going to end. Thanks one again. Thanks.
Very good afternoon. Respected Raghavendra sir, am I audible to you? Hello sir, good afternoon. Sir, can you hear me?
Yeah, I can hear you now. Can you hear me?
Hello, sir. Very good afternoon, Dr. Raghavendra Ashok Bohra. This is Dr. Asha from Nirmala College of Pharmacy, your host for today and the chairperson for today's session. Hearty welcome to you for this online session. On behalf of the organizing committee of Nirmala College of Pharmacy, give me just a couple of minutes so that I'd like to introduce you to our audience. Dr. Raghavendra Bohra is a distinguished academic researcher and mentor, consistently recognized among the world's top 2% scientists by Stanford University in 2023 and 2024. He currently serves as a senior scientist at CranMed Limited and as an associate professor, where he operates at the intersection of cutting-edge research and impactful teaching. Dr. Bohra's illustrious career is marked by numerous accolades, including Royal Society of Chemistry's highly cited author award and fellowships from the Indian Council of ICMR, Irish Research Council, and Science Foundation Ireland. He has also served as a visiting scientist at the Maine Medical Center Research Institute, USA. Dr. Bohra has played pivotal editorial roles, including associate editor for Hela and Elsevier, Biomaterials Journal, and BMC Biomedical Science from Nature. He has also edited the influential book Hybrid Nanostructures for Cancer Theranostics. His research focuses on magnetic trigger drug delivery and cancer nanomedicine, bridging fundamental science with translational applications to address critical challenges in healthcare. With a unique ability to integrate academic excellence and pioneering research, Dr. Raghavendra Bohra continues to significantly contribute to healthcare, nanomedicine, and education. Dr. Raghavendra, we are extremely happy to have you here. We are privileged to have your session today. The student delegates and participants will be benefited with your presentation. Dr. Raghavendra, session will be over to you. You can start your session, please. Sir, I think you're muted. Can you just unmute yourself, please? Dr. Raghavendra? Sir, can you please unmute yourself, please, Dr. Raghavendra?
Absolutely sorry about that. So,
I request Dr. Raghavendra to please unmute yourself.
I did. Can you hear me now? There is no mute from my end now. Can you hear me now?
Yes. Yes.
I am not able to pick your voice. Can you hear me now? The voice is muffled, so your response is muffled. Can you hear me now?
Yeah, it is audible.
Audible, perfect. So, thank you very much, and a very good morning to everyone. Good afternoon in India, and I'm really thankful for the committee members, Roy sir, and all the team members to just give me opportunity to present myself here for a short period of time. Once again, thank you very much for this long introduction. we are not habitual for such a long introduction. It's all around, we are all on the same platform. So today I'm here merely not just to discuss my research work, which is which is revolving around hypothermia in cancer therapy with the help of magnetic nanoparticles. But around that, I also want to just ensure that we are not restricted to one research problem, but more importantly, it's the thing that what we can offer to your department or your college from our end, so that further collaborations can be strengthened. So whatever I am showing here, merely this can be reproduced, or this can be further taken from the guys which are really interested to continue similar research topic, or they have a similar flavor in their mind to continue the research. So just to move ahead now, so the topic is hypothermia. It's a new normal in cancer or not. So that's what we are going to see in the coming slides. It's all been said, so no more wastage here. Yeah. So currently I'm working as a senior scientist in CRENMED, and as well as I'm associate professor in the university. So I'm based in Ireland, and you can see why Ireland. So Ireland is merely now getting a global hub identification, because 9 out of 10 companies are in Ireland right now. And you can see, like, nearly, if you take top 10 companies from world, they have their headquarters in Ireland. So it's be Boston Scientific, Abbott, or Medtronics, or Stryker, even MSD. So these are all companies which are really major players of R&D. And to name out certain products, like all the drug eluting stents, all the medical device, they are being manufactured in Ireland. And so that's why it's 9 out of 10 global companies that contributes in the medical device segment. And I am also a part of medical device segment here in CRENMED. Yeah. We all know why cancer happens, what are the major factors that are responsible for cancer, and how we diagnose cancer, and what are the risk factors which are associated with cancer. So I will just walk on these slides. Something really you need to focus here, that as an part of our society, the latest ICMR report says that nearly 1.7 million patient people have been diagnosed cancer last year, and now many more are around that corner, and the numbers are growing up. And if I want to just name few cancers, like oral cancer, esophageal cancer, breast cancers are among the top list of this trend in India. We all know what are the current treatment options for cancer: hormone therapies and immunotherapy. These are something which are newer terminology from cancer treatment options. For surgery, radiation therapy, chemotherapy, these are age-old practices which are currently used or which are currently practiced. So this is a beautiful picture I found in nanoconvergence paper. So if you see this paper article, this was a nice cartoon which was there. So here you can see that the doctor is asking that radiotherapy and chemotherapy, they have solved everything. So, but patient has nothing left apart from the head. So what this cartoon trying to address, that those drugs are really effective, but the main problem arises that due to their poor pharmacokinetic property, non-targeting ability, and immense toxicity, which are more serious at some point of the treatment regime where the disease mere subsides. So you need to take care that this contributing factor should be absent when you design a newer treatment option in cancer therapy. We all know what is cancer nanotechnology, and we all know what the nanoparticle means. So just walking through this slide again. Again, important slide from the research perspective. When you say a cancer theranostics, that means the particles which can be used as a therapy or and as well as for diagnosis. When I say diagnosis, So in cancer therapy, we use different modalities for diagnosis like MRI or PET CT scan. So when you use your material which helps to detect or to diagnose the tumor location or the intensity of the tumor, then this entire system or this entire uh, material itself can be called as a theranostic material, which has the dual properties of diagnosis as well as imaging properties, diagnosis as well as treatment properties. So yeah, so if you know, the title was around hypothermia therapy for cancer. And so what is hypothermia? So hypothermia is nothing but this is a simple instrument which we have learned in a graduation physics or maybe in a chemistry, in a physical chemistry, where you can see that whenever you have a electromagnetic coil or electromagnetic generator, and when you place any conducting material or any material that can lose the ion when they have been kept under this electromagnetic generating coil here. The coil is an example which can be influencing factor. So when I use such material, when you apply a AC magnetic field, that is alternating current magnetic field, the material, mostly rare earth material or magnetic material, they have a tendency to lose their finite electron from their shell, and that electron, in response to this electric magnetic field, it just vibrate or oscillates in a motion that generates the heat energy. The heat energy which is generated due to this oscillation can be captured or can be used to kill the cancer cells, because it has been long said that cancer cells are susceptible to heat beyond 41 degrees Celsius. Your normal cell can sustain the temperature above 41, 42, 43, but cancer cell, due to their leaky vasculature, due to their abnormal vasculature, due to the tumor infrastructure what they have, they cannot sustain this temperature. So the concept is absolutely revolving around this. So you have your material which you design in a way that when you keep them in this system of or in this magnetic field, they start to lose the heat, they start to lose the electrons. The electrons will start to oscillate in a motion, in a van der Waals direction, or in a motion, or eddy current loss, what we call in a proper terminology. And that eddy current loss or that van der Waals losses cumulatively generate the heat, and that heat can be used to kill the cancer cells. Now, here two important questions are arising. Is that heat is controlled? Is that heat can be managed? So, because all this rare earth, they can jump from 50 to 90 in a seconds. I'm repeating again, all this rare earth metal, all this rare earth material, when you keep them under this AC magnetic field, they can jump from 50 to 90 in seconds. You don't want that to happen, because that can be a thermal ablation, not merely heating. So the entire research is focused around to ensure that you have a way controlled heating where the temperature remains around 41 degrees Celsius throughout this entire process. So this entire therapy is called as magnetic hyperthermia therapy, and it started from Gilchrist in 1957. And in 2019, MacForce, a company in Germany, which has been approved by FDA in 2015 and then later in 2019, to ensure that the iron oxide nanoparticles can be used in brain glioma and as well as in prostate cancer. So now we are just a foot ahead, or just a foot in a door, to ensure that this therapy is reaching towards the FDA door, and very soon we can expect that this can be a new normal in cancer therapy. So just moving ahead with the slide. So that was I was saying, that when you have very controlled heating, you don't have this side of. So the question is, you should have a very controlled heating to ensure that you don't have a thermal ablation when you treat your patient. In light of this, our like a few year research, we have designed a material called as cobalt zinc ferrite nanoparticles that has been designed with the proper composition of cobalt and zinc. Both belong to a transition state material, where the composition was such in a proper way that when they were exposed to this heating element, they showed a very uniform and controlled heating system. So these are the basic developmental like a material characterization of the cobalt zinc ferrite. So you can see XRD, which conforms with the normal JCBDS pattern card number 221086 for COFE204 and ZnFe204. So all those like students from, they have a background of material chemistry or some sort of a material background, they can understand what JCPDS and what this XRD means. FTIR, which is very common, shows that those relevant peaks which are there in the material. EDS spectrum confirms the presence of zinc and other elements in the material, and the zinc, and the TGA confirms the presence of the coating which was used at that time. It was PEG, if I remember correctly. Yeah, it was PEG. We had a series of coating there. Yeah, on the substance. Zeta potential confirm the size of particle, DLS, everything confirm, and these two special properties, C and D, they shows that they have that proper magnetic hysteresis, because these are the magnetic properties of the material. When you have a saturation of magnetization around 60 msg, that confirms that material will not keep magnetic property on its own. Whenever that they have been used as a magnetic material, they will work as a switch, and whenever you turn off the magnetic material, they won't keep the heat up to them, they will release the heat. So there is no heat holding in the material, and this property is confirmed by the C and D graphs, where they have inline or very defined materials magnetic saturation. A simple biocompatibility studies confirm that on L929, and by using different assays, they are very biocompatible and within the FDA norms of biocompatibility. So this is something, when you see, when we kept this material in under this magnetic field, if you can see, they have a very precise and controlled heating. When you see sample A is around 42 degrees Celsius for 10 minutes. Sample C has a very like shooting hitting pattern. You can see it went up to 46, 48, which we don't want, but sample A had shown a very desired pattern of hitting up to 40, up to 40, 38, 42, 41. So this was something which we're expecting in the material, and that has shown. Moving ahead, the same material, when we did in vitro magnetic hypothermia, so the cells were MCF7 were selected, and they were incubated with the material, and they were placed under the magnetic field, and you can see after 10 minutes and 60 minutes the cells started dying, and you can see the presence of ROS production has been incrementally increased over the time point from control to 60. Again, that confirms that the magnetic heating, the death is due to production of ROS due to the heat. Similar, if it was continued here to understand more, if drug was loaded in the system, and when you have a dual therapy, like the beads are carrying drug as well as they are also capable of hitting, you see a 2 times more percentage of death in the MCF7 cell at time point of 30 and 60 minutes. Then what we did an animal study where those material were used for in a treatment to understand the what exactly the beads, when the magnetic particles were injected for a similar model of MCF7 breast cancer in a mice model. And you can see after, because we continued for 40 days, because the treatment was there for 3 days, but till 40 days we were seeing the mostly histology images and the tumor burden, and at the end of like maybe 40 days, we saw that combined therapy with the drug loading and hypothermia, the tumor burden was almost nullified. But it was not merely achieved with the hypothermia, but when with the combination, we had that effect which was seen. So we went down further to understand what was the mechanism, and of this. This is certainly not we are just hypothesizing this mechanism, because whenever you apply heat, the heat generally just initiates the heat shock protein 70, heat HSP 90, and these are the proteins which plays a vital role in apoptosis of cancer cells. And by just inhibiting the BCL2, and then just upregulating the caspase-3 and the cytochrome 3 from mitochondria, you encourage the apoptosis in the cancer cells. At the same time, there is another saying that which we are trying to understand, that your innate immune system is trying to respond to this heat shock protein, and then those responses include the presence of T cells. We haven't seen T cells accumulation around our tumor site because we were not sure around that, but definitely now the new research say that when you have this microenvironment, the T cells which are dormant in the previous condition where the tumor cells are highly activated, the microenvironment is highly motivating for tumor, the T cells are in dormant stage. But the moment you start shedding off those HSP70s and the apoptosis is triggered, T cell presence is activated, and that T cells generates innate immune system, and that adaptive immune response further contribute to cancer killing cells. So eventually it gives you a two-arm, where your first arm is due to apoptosis, and the second arm you are activating the adaptive immune system and innate immune system that activates T cell, and that enhances the cell death in the entire process. Yeah, so just to conclude what we are saying, and now this conclusion is very much important take-home message for you guys as well, because now when I show you certain sort of like what the flavor of research is, all you can think around this process and try to think that can further steps can be taken here to understand how this process can be done better, how immune system can be involved in a more aggressive way, or how the material can be used in a minute quantities, or in a way that a minute material is required to generate that particular heat to kill the cancer cells. Yeah, so the intelligent design and synthesis of library of nanomaterial can be done to have a more physicochemical properties, and to ensure that they have a proper surface properties that make sure that they go to the cancer cell directly. Magnetic heating and drug delivery, these are two different aspects, but when you use practically, there has to be certain protocol where, and that protocol should be reproducible, where guys can really think and make progress to be more robust. Yeah, if you are really interested in further reading, I recommend you go ahead for this, see this Doxil, the first FDA approved nano drug, how the lessons have been learned. I recommend you to read this article. I recommend you to read our book, which is there again in 2018, and similar papers.
So, good afternoon, Bora sir.
Yeah.
Is it audible? My screen visible to you?
Yeah, you are audible.
Okay, yeah. And good afternoon, sir. And it is very pleasant to have a discussion with you and with our students. And obviously, you want to learn a lot more things from you, and especially related to the nanoparticle, the development, the synthesis, your guidance and all, which will be required for our laboratory. And obviously, as we know that we have collaboration, so that collaboration we are expecting to continue for long.
Yeah. So the only thing that Roy sir, what I just try to show the flavor of research, what we did previously and what we are currently doing. So I keep motivating guys that if you are really interested around this topic, you can definitely take further any of this topic which I shown in my presentation. Like ask me if you need any further help around that particular project. I am really I can really extend my collaboration via you, and then we can really move ahead in any further topic. Guys can do either as an if they want to do organic material, they can really go for organic material. If they want to use organic inorganic hybrid, they can go for this type of combination. Or they want to literally go for organic material like liposomes and PLGs, they are welcome to do, and we can play around and see that how I can extend collaboration so that we can come out with really nice science and impactful science, which is really needed for the society right for the moment. What I understand that, see, this work is merely, you can see few slides, but the entire theme was designed in a way that it should be appealed to FDA. all the components were chosen in a way that FDA should not reject in terms of, because all these are metallic material and FDA don't prefer to use them, don't help to give a permission around them. So everything was designed around that particular aspect, and we may miss something, but if your students or your faculties are really interested to carry forward around this space, they are really welcome, and I can initiate all lot of suite of experiment which is there with us, and we can progress ahead in this way.
Yeah, obviously. Thank you, Bharat sir.
And
Thank
anyone
you
in the and anyone want to have any questions around that particular space, even small questions, or I'm really happy to take and answer to the best of my abilities.
5 minutes. Ready to. 5 minutes. Managing. 5 minutes. Managing it. Just go.
By accumulation.
By manage. Or editor.
Hello?
Hi, hello.
Yeah, thank you for your talk. I just have one question that is there any specific procedure of managing bioaccumulation of the nanoparticles that have been seen to produce a small kind of toxicity in whatever organisms we deploy them?
I couldn't figure out your question. Can you come back again, please?
Is there any procedure to combat the toxicity which is come by the nanoparticles that are being deployed in any organisms?
See, toxicity again, it's depend upon the initial material what you choose. So your initial material is very important player in the approval. So if you choose, so if you have a material that really appeals or which is listed by already green signal given by FDA, you need to choose around that. And then the components you are using should be again user friendly or FDA friendly or biocompatibility friendly system that you have to select and pick around that. So the starting material always plays an important role, and you need to be very much choosy around that particular aspect.
Yeah, thank you, sir, for your nice speech and presentation, sir. And we are very much generous that you received. You have accepted our invitation to be one of the premium speaker for our conference.
Yeah.
On behalf of my organizing committee, I am very much thankful. And once again, heartful, thankful and appreciable. And I appreciate your effort and for giving such a precious time with us.
No problem, Professor. I really thank you for all the management and to you and principals or everyone to just giving me opportunity and presenting myself in a short way.
Thank you, sir.
Yeah, thank you very much.
I will call you back. Thank you, sir.
Okay.
Thank you, sir.
Thank you, Dr. Raghavendra.
Okay.
Thank you so much. Hello? Hello, Dr. Uttam. Am I audible to you? Dr. Uttam? Hello, Dr. Uttam Prasad. Sir? Sir, kindly let us know if we are audible to you. Sir, kindly unmute yourself if you are speaking.
Yes, yes, it is audible, it is audible.
Good afternoon, Dr. Uttam Prasad.
Good afternoon, madam. My voice is audible, madam.
Good afternoon, sir. Sir, this is Dr. Asha from Nirmala College of Pharmacy, your host for today for the online session. Very hearty welcome to you on behalf of the organizing committee and the whole fraternity of Nirmala College of Pharmacy. Just give me a couple of minutes to introduce you to our audience, sir. Dr. Uttam Prasad Panigrahi is an associate professor at Faculty of Pharmaceutical Science, Assam Downtown University, Guwahati, Assam. He completed his B.Pharm and M.Pharm with distinction from Biju Patnaik University of Technology, Odisha, MBA from Periyar University, Salem, and PhD from Jawaharlal Nehru Technological University, Hyderabad. He has 16 years of teaching and 6 years of research experience. He has 72 research paper publications in Scopus, Bentham, Elsevier, Springer, Wiley, Taylor and Francis journals. He has 6 Indian patent publications, 4 PCI syllabus books and 1 Elsevier book chapter. He has presented 23 research papers in national and international conferences. He has guided 40 B.Pharm, 36 M.Pharm and 5 PhD projects. He has attended 30 workshops and 12 FDPs in India, and he's a lifetime member of APTI and INC Bangalore. Presently, he's a member of Faculty Research Committee and NAC Criteria 4 Coordinator at Assam Downtown University, Guwahati. He's a recipient of Research Excellence Award 2022 and also qualified faculty eligibility test 2010 conducted by JNTU Hyderabad. He's an editorial board member and reviewer of various journals of pharmacy. He's involved in teaching, research in various areas of pharmaceutical analysis and bioanalytical studies. He has hands-on experience in instrumental analysis comprising LCMS, HPLC, IR, and UV spectrophotometer and many more. Dr. Uttam Prasad, we are highly privileged to have you here with us today. Hearty welcome to you, and session is over to you.
Good afternoon to all.
Dr. Uttam, you can take over the session. The session is over to you.
Am I audible?
Yes, yes, please. You can please proceed.
Hello.
One room at your holiday in New Delhi International Airport. So, holiday in
1000.
Am I audible?
Yes, sir.
I'm audible. Am I audible? Am I audible? Hello.
Continue, sir.
Am I audible, madam? Hello?
Sir, audible, sir.
Audible?
Yes, sir.
Okay, okay. So my screen is actually visible, no?
Yes, sir, your voice is audible, sir.
Okay, okay. So my screen is actually visible there, my screen. Hello?
Yes, sir.
Yes, yes. So the slides are open.
Sir,
Is it
you are
visible,
audible.
the
You
slides?
can start the session.
Yes, yes. My slides are visible there, my slides. Hello? Okay. My slides are visible there, madam.
Yes, sir. Yes, sir. Please continue.
Okay, okay, okay. Okay. Good afternoon to all. Good afternoon. Good afternoon to all. Good afternoon.
Sir, good afternoon. We can please proceed, sir.
Slides are visible, madam, there?
Yes, sir. Yes, yes.
Okay. So good afternoon to all. Thank you for giving me the opportunity for an international symposium on sustainable drug design and nanoparticle development, quantum and computational perspectives. And I am delivering a presentation today on this occasion, LCMS and its application in phytochemical analysis. LCMS actually, it is a combination analytical technique between a liquid chromatography and mass spectrometry, where liquid chromatography is having the separation power and mass spectroscopy is having the detection power, where we can detect the mass of the separated components. And LCMS is nothing but it is an integrating model. We can tell it a hyphenated model which combines both chromatography and mass spectroscopy. We know that simple LC is nothing but it is a high performance liquid chromatography which consisting of several components like column, sample inlet system, pump, degasser and solvent system, and with a mass spectroscopy which is attached by an interface which is very, very important connector between a LC and a mass spectroscopy, and particularly the interface is very, very important for phytochemical analysis. So there are several points we have to take care when we will perform the phytochemical analysis. First, we have to focus on the interface. So interface is nothing but this is a connector between a HPLC with a mass spectrophotometer. A successful interface will make a good separation as well as mass detection. So what requirements you have to focus on a basic selection of the interface is, first thing is that maintaining chromatographic performance. Second thing is that high transfer efficiency from LC to MS, and there will be no degradation in mass spectrometric performance. So there are several types of interfaces we are using for LCMS. Mostly we are using direct liquid introduction, moving belt system, thermospray, continuous flow, fast atom bombardment. We will discuss, there are some most common interfaces we are using in LCMS studies. First one is electrospray ionization, second atmospheric pressure chemical ionization, then atmospheric pressure photoionization. Now we will discuss what is electrospray ionization. In this case, the interface consisting of one nebulizer, and there is a electron spray ionization tip. At the tip of the nebulizer, there is a ESI tip is there. So when the sample is coming, separated sample coming outside from the HPLC system, and it will enter inside the interface. So these nebulizers will convert them into a droplet system, and these droplets will be exposed to a 3 to 5 kilovolt ampere voltage. Then these droplets will convert it into a energy, means electrical energy. So charged droplets we can tell. So these charged droplets undergoes desolvation by the help of a drying gas. So these droplets will convert it into a positive ions. So these positive ions will be migrated towards the mass spectroscopy. So this is electrospray ionization, which we are using, commonly used in phytochemical analysis. Next we are discussing about the atmospheric pressure chemical ionization interface. This is another type of interface which will connect both LC with mass spectroscopy. In this case, the sample which is coming from the HPLC, it will enter inside a nebulizer, and this nebulizer is connected to a heater, and this heater will convert the sample into a charge. So what happens, these droplets when enter inside the heater, it will be undergoes evaporation, and there one corona needle will be there. So here one corona needle is there. So this corona needle will expose it to the droplets, and these corona needle will give some electric supply. So these droplets will convert it into a positive charge, and these positive charge will migrate it towards the mass spectroscopy. Next, we are going to discuss about atmospheric pressure photoionization. So here the sample is entered similarly like APCI. Here also the sample will be entered inside the heater. So these heater will convert them into a droplets. Through this nebulizer, the droplets will come outside, and it will be exposed to the vacuum UV ultraviolet. So these vacuum ultraviolet will convert them into a charged molecules, and these charged particles, positive charged particles, will go inside the mass spectroscopy. So there are three types of interface which we are using in phytochemical analysis. Most of the cases we are using. And what is the basic difference between electrospray ionization is mostly useful for polar compounds, and atmospheric pressure chemical ionization is useful for nonpolar compounds, and atmospheric pressure photoionization is useful for moderate polar compounds. And now we are discussing about a schematic diagram of a LCMS system. Here there is a two equipment. See, in the left side of the from my left side, from your side it is right side, that is LC, and the right and the left side is the your mass spectroscopy. So liquid chromatography is basically useful for separation of complex compounds, complex mixture into individual components, and these individual components, when it is passed through the interface, there you will get electrospray ionization, atmospheric pressure chemical ionization, and atmospheric pressure photoionization. It depends upon the samples. So now this interface will connect the HPLC with a mass spectroscopy and enter all the ions, positive charge ions, inside the mass spectrophotometer. Inside that mass spectrophotometer, the positive ions will be analyzed by mass per unit charge, and it will be detected, and due to the fragmentation of the ions, you can find what is the molecular weight of that particular
Hello, sir. Is there any problem? Please continue the session. Hello, sir. Is there any problem? Please continue the session.
Good afternoon, students. Now, Mrs. Sai Lakshmi Garu is going to take the session. Okay, we have with us two young researchers. One is Sai Lakshmi ma'am and second one is Asha. Now, Sai Lakshmi Garu will going to take the session. Ma'am, please come on to the dais.
Good afternoon to all. Myself, G. Sai Sri Lakshmi, working as Associate Professor, Department of Pharmaceutical Analysis in Nirmala College of Pharmacy. I am the one of the research scholar in Wales University, Chennai. My work is presented as a research. So today we discuss about my research proposal to our students. First, title of the research work is Analytical Method Development and Validation of Stability Indicating RP-HPLC Method for Simultaneous Estimation of Sovazavir and Daclazavir in Bulk and Pharmaceutical Dosage Forms. These drugs comes under antiviral category. Next, this is the introduction about the Sovazavir. Sovazavir is the most powerful antiviral drug. So it is a new innovation to develop the RP-HPLC method for. So antiviral drugs are most popular in at the time of COVID 2019. So we decided to work on antiviral drugs for further diseases. So another drug is daclazivir. This is a combination of daclazivir and sobazavir combination. So what are the methods and materials we have to use for the analytical method development means, first one, what are the instruments we have to take in, and what are the chemicals we have to use for the method development and validation of sobazavir and daclazivir. These chemicals are acetonitrate and HPLC grade orthophosphoric acid water purchased from Merck Life India Private Limited. And what are the instrumentations means liquid chromatography. Already finally students have the knowledge about regarding RP-HPLC methods and LCMS like that only. In first year students we don't know about the HPLC and other instrumentations. Next, what are the chromatographic conditions? These are the chromatographic conditions, that means 0.1% of orthophosphoric acid and water X breaks, and C18 is the column. The column centimeters and diameters is 150 into 4.6 millimeters and 3.5 micrometers. And what is the main objective for this means to develop a new drugs for the method of RP-HPLC and UPLC and LCMS. This is the my result of chromatographic, that means blank condition, and means blank means without drug. That means already we know heard about the placebo. Placebo means without drug, like that only. This is a without drug, that is called as blank determination by RP-HPLC methods. In UV conditions although we have to determine the one of the blank determination. That's why I also used for HPLC method also. This condition is called as specificity. And next one, linearity. What is the linearity means? Linearity range means to maintain the UV conditions 40 to 600 microgram per liter. And next one, this is the linearity results. Accuracy. Accuracy means it is achieved by the measuring of recovery experiments of 3 conditions. That means 50%, different percentage of drugs we have to take in, and different times and different days we have to use for the determination of different combination of drugs. That is the accuracy. That means different percentage means 50% of drug, 100% of drug and 150% of drug, like that only we have to determine. Next, intraday precision and interday precision. Intraday means within the day. What are the percentage we have calculated like 50%, 100% and 150%? That is calculated intraday, means single day we have to complete the calculations, that is called as intraday. Interday means another day. Intra means within the day, inter means another day. What are the calculations we have done? 50, 100, 150 and 200%. This is calculated by single day, that is intra. Another day is another combination we have to take in, that is interday. Interday precisions. This is the interday precisions of sobazavir and daclazivir combinations. Next, LOQ and LOD. These are the another type of specifications for sobazavir and daclazivir combination of antiviral drugs. Next, robustness. These are the parameters we have to develop each and every single method in analytical methods also. Next, degradation studies. Degradation studies means temperature, room temperature and environment temperature. So like that only, that is whether the drug is stable or not. That is called as degradation studies. Next, conclusion. This method is prescribed the quantification of. Quantification means how much amount to be percentage. The combination of sobazavir and daclazivir in bulk drug and formulation. This is the new method development. In previous days there is no development of this type of development for antiviral drugs. This is the new first and developed method for combination of sobazavir and daclazivir combination for treatment of antiviral diseases, that is done by my work in PhD. Thank you for giving me this opportunity. Thank you Principal Sir and management for giving this wonderful opportunity. Thank you for your patience listening.
Thank you so much, ma'am.
Yes, yes, yes. Is it visible, the slide? Okay, okay, okay. Now it is okay. Now the screen is showing, madam?
Please do survey.
Okay, okay, okay. Now screen is showing?
Sir, is visible, sir?
Yes, yes. Is it visible? The screen is...
Sir, screen is okay, sir?
Okay, now the screen is visible?
Yes, sir. Yes, continue.
Okay, okay, okay.
Dear students, we have a small announcement. After this session, we have one more session by a young researcher, an alumni student, Miss Asha. She'll be presenting her research work in 5 minutes for all of you. After that, you can go for a tea break. After tea break, by 4 o'clock, we'll be having culturals. Will be continued. 15 to 20 minutes we'll have culturals. After that, valedictory will be continued. In the valedictory, again, we'll have culturals. So I request you all, after going for a tea break, you have to be back, but wait for this session to be completed. Right? After this session, we have one more young researchers session. After that, you can go for a break. I'll intimate you when to leave. Thank you.
Rotary evaporator. So after evaporation, these extracts are injected inside the LCMS system. So LC will separate individual components, and these individual components will be molecular determined by the mass spectroscopy. Let us take an example of the solanum retroflexum. It is a solanaceous fertility plant. First, this solanum retroflexum leaves is dried under shade, and it is grinded to powder, and it is kept in a room temperature in an airtight container. After that, these powders are extracted in 3 different percentage of solutions: 40%, 60%, and 80% methanol is to water. After that, these 3 extracts we have to go for evaporation. After by rotary evaporator, these evaporated samples will be kept in a prior to -20 degrees centigrade. After that, these samples are ready to go for optimization in HPLC and mass spectroscopy. First, we have to select the mobile phase. That mobile phase, we have chosen solvent A is formic acid with deionized water, and solvent B is acetonitrile with 0.1% formic acid. So here the technique we have used is isocratic, means gradient elution. Gradient elution, why we are using? Because in phyto compounds there are several components are there. So we should select a different proportion of the mobile phase. So that's why we are using here gradient elution method, and this gradient method is 30 minutes. First we have taken 98% A, solvent A, for 1 minute we have run, and some sample components are separated. Then we have used for 3 minutes 97%. Then at the 21 minutes we have taken 92% of the solvent A. Then at 25th minute we have taken 50%. Then 26th minute, for 2 minutes we have to run. Then we have taken 5%. Then at the last 2 minutes, at the end of the solvent A with mixture with the solvent B, there are several components that are separated out through the HPLC system. Then when the components are separated, then it is introduced inside the mass spectroscopy through an interface. Already we have discussed here. The most important interface we have used is electron spray ionization, and there are several optimization points we have to maintain in mass spectroscopy. That is capillary voltage, cone potential, source temperature, desolvation temperature, cone glass flow, desolvation glass flow, multi-channel plate detector potential. So these are all the parameters we have to follow. These parameters are specifically designed for particular sample. So there are different samples we can use, and we have to maintain these optimal parameters. Next, after separation, these are all the important components we found in solanum retroflexum. The most important components we are found here. This is the liquid chromatography chromatograms, and this is the mass spectroscopy. And here are the results I am showing here. The most components we are found in retroflexum, that is chlorogenic acids and caffeolyl quinic acid, then feruloglycoside, then dicafeolquinic acid and caffeolyl quinic acid, and para-coumarolinic quinic acid, and coumaroyl hexose. These are all the components which is detected as well as separated from the solanum and retroflexum. Some other components we are found as ferulonyl quinic acids, flavonoid derivatives, and alkaloids. Mostly we found that is solasolinine and solamargin. Let us take another example. LCMA studies of methanol extracts of seed cells of Archidendron bubalinum. This is another natural sources. We have taken the seeds and we have extracted these seeds. This is the method. First, these seeds are collected and it is peeled to separate the seed cells. And these seed cells are thoroughly washed and sun-dried for 4 hours. After that, it is soaked in methanol, 1:10 weight by volume, and it is extracted by maceration process for 24 hours. After that, these extracts are filtered, and these filtered substances, again it is concentrated with a rotary evaporator at 50 degrees centigrade. After that, these samples are injected inside the liquid chromatography and mass spectroscopy, and here we have used the solvent is methanol with 0.3% formic acid, and we are maintaining the flow rate at 0.1 ml per minute with an isocratic elution, because here we are detecting only the specific component. That's why we are using the isocratic, but the previous sample we have used the gradient one. Now here we are using the sample in interface to make the sample in a dry conditions in interface, that is nitrogen gas. So here we have used the nitrogen gas in electron spray ionization. Next, sample injection. So here we have used 0.5 gram of sample extract diluted with methanol. Then it is filtered with 0.22 micrometer nylon filter. Then after filtration, 5 micrometer volume of extracts are injected inside the mass LCMS. Next, these are all the mass spectrum, and here we found 5 compounds. See here, candidate 1, 2, 3, 4, 5. So these are all the compounds we found at 3 different retention times. What are the 3 different retention times? 0.4, 1.28, 1.90, 2.63, and 2.80. So these are all the retention time, and these are all the masses. So first candidate we found at 65, 436, 247 and 450. So these are all the mass of that particular compound. So after that, we found that these are all the mass spectrum of 5 components which is present inside the methanol extract of that seeds. So we found that the measure retention time of this particular Archidendrum bubalinium, we found that 5 retention time, 5 chromatograms with 5 mass detections, and the most identifiable mass detection is 436 at 1.90 and 450 at 2.80. The 1.90 retention time, it is predicted that that is proligin, and 2.80 it is detected as acetylbin. So these are the most important proposed compound found in Archidendrum bubalinium seed cells. And after that, the separation is taken place. The florigin, which is a component of the particular seed, natural seeds, it is fragmented, and we found that 274 is the molecular weight of that particular florigin by mass spectroscopy. And another one is that acetylbin. We found that at 268, it is the mass detection of that acetylbin. So these are the 2 important fragments we found in mass detection of that particular Archidendrum bubalinium seed cells. So like this, the LCMS is very, very useful for phytochemical analysis. Not only for phytochemical analysis, LCMS is also very, very useful for bioanalytical studies, forensic science. We can trace the elements as well as the components. Also, we can detect the bio compounds from the biological fluids like serum, urine, saliva, different cerebrospinal fluid. So here we have special emphasis on the phytochemical analysis, and it is a very good equipment for different types of separations and mass detections. So thank.
Hello?
Hello?
Sir, am I audible?
Yes, yes, audible, madam.
Sir, thank you so much for your wonderful talk. All the students hope they've got benefited with your presentation. We are really privileged to have you, sir. Thank you so much for your time and effort you have put in collaboration with our university, with our campus. I hope we'll have a future collaboration as well, sir.
Sure.
And someday it would be good if we could see you live in our campus. So we'll have a plan further in future. Thank you so much once again on behalf of the entire College of Nirmala College of Pharmacy.
Thank you, madam. Thank you.
Thank you, sir. Madam, we'll have one talk by the young researcher of today. She's going to give a talk on her research work. Now I invite Ms. Asha Gummadi, our alumni student. She's our student from here. We are very proud to call her onto the dais. Obviously, she'll be proud to present in the college where she has studied. Give a big round of applause. We hope to see you as students again coming back here. Hearty welcome to you, Asha.
Good afternoon, one and all. I'm not going to take much of your time. I'll just complete within 5 minutes. So my topic is related to the application of AI in the pharmacy sector. So these are the various fields where the AI is going to work. So I'm going to explain some of these topics here. See, first, we all know that Google has AI Gemini, and there is one more Gemini called Med Gemini, where we can know the, ask any questions related to your, like for example, you don't know how to read the CT scan, then it is going to, you are going to insert your images and you ask it, like, can you give me the radiological report of my CT scan? So it is going to give you the radiological findings, and researchers have found that these are similar to the radiologist's written report. So the similarity is 91.1%. Then the second one is, where the AI is working is Molly. She is a virtual nurse. So this is specially designed for the cardiac patients. She is also known as a healthcare avatar because she is a friend to the heart patients. How she is going to tell the patients is, like she is going to ask, how is your signs, how is your day, is your day good, what are your vitals, is your blood pressure is good. Based upon the reports, she is going to advise the patient what medication need to be taken and what action need to be taken. And the third one is IBM Watson. This is specially designed for the treatment of cancer. So how it is going to work is, IBM is collaborating with several cancer institutes. Different clinicians, well-expertised clinicians are also involved in this. By using this tool, total 9 studies was identified, which including the patients of 2,463 patients. So what is the process? So first, AI is going to analyze based upon the patient data which I have given from the medical literatures or clinical trial data and patient's record. And it is give you the data based upon the evidence only. It is not going to give any prediction model, but it is going to provide you the data based upon the evidence only. Then finally, the clinicians who are involved in this, they are going to finally make a decision which medication treatment plan is best for the patient. The next is, the AI is going to give you the 3 different buckets. That is green bucket, yellow bucket and red bucket. Green bucket includes the treatment plan based upon the evidences from the clinical trial data. Whereas yellow bucket includes, say, what are the alternative methods other than the first green bucket. It is going to give you the what are the alternative methods of the treatment for the patient. Whereas in the green bucket, it is going to give you the contraindications, what medication should not be taken to the particular patients. Again, that is also based upon the evidence only. Next, how AI is going to helpful in the drug discovery process. These are some of the example AI tools. I'm going to explain the free tools, AI tools, which are going to be useful for you in future. So this is one of the tool is Swiss ADME tool. So I have a new, imagine that I have a new compound. I don't know the ADME properties of my compound. So this tool is going to helpful. How it is going to helpful is, just I'll draw my compound, or I'll enter the smiley format of my compound here, and I'm just going to click run option. Here after run options, I'm going to get the physicochemical properties of my compound, lipophilicity of my compound, water solubility and pharmacokinetics, whether there is any GI absorption is there, whether my drug is going to pass the blood-brain barrier, all these things are going to given by this tool. And the next tool is, for example, I got to know my ADME. The next I am to check whether my drug is similarity is there or not. Already my same compound is available in the library or not. In order to check that, this tool is going to helpful. That is Swiss similarity. By using this tool, you can identify whether my drug is similarity is there or not. There is another tool called Swiss bioisostery, where you can make the replacements of your existing drug and check the structure activity relationship of your drug, and you can also predict the efficacy of your compound. And this is one more tool called Swiss target prediction, where you can predict the target, which target I need to work on it. So just need to enter your molecules and you have to click the predict target. Then it is going to give you the what targets you need to work on. Based upon that, you can go for the further analysis. So what is the further analysis is, you have to check whether how much my drug is going to activity on the particular target by using the docking. So you are going to use a docking process and you are going to check the whether there is inhibition percent is there. If inhibition, how much percent is there, we can know that. And it is also helpful in the targeting drug delivery system by generating the predictive models based upon your physico-chemical properties and biological properties of your compound. And this is one of the interesting topic, that is smart drug delivery system. Smart drug delivery system indicates that here AI is going to helpful for the formulation. Like we are going to create a drug in such a way that the drug is going to be released only on particular conditions. Like this particular pH only the drug need to be released, this particular temperature only the drug need to be released. For example, a gastric tissue is there. So gastric tissue pH is different, the enzymes present there is different, and the temperature is different. So I am going to design in such a way that the drug is going to release on the particular pH and particular temperature. And this AI also helpful to detect the changes in the target site after the drug release. Whether there is any target size changes is there or any physiological state changes is there or not, AI is going to helpful to detect. From this I conclude that AI is very useful in the drug discovery process, drug release system, targeting strategies. With continuous research collaborations, AI is going to helpful in the precision medicine. Thank you. Any questions? Any questions from the audience? For example, I have a new compound with me. What I need to do? What is the drug discovery process? You want to know anything? Feel free to ask me. No questions? I think you understood my concept. Thank you.
Thank you so much, Asha. It's pleasure having you here again back. She was in my class, now again along with me on the stage. It's an honor for all of us. Very proud of you. So thank you everyone for your kind attention for all the technical sessions. You all were very patient. Thank you so much. Are you all happy with what you learned during the sessions? If you have felt any point of time boring, in the future later you'll understand why you neglected this. But anyways, thank you. So we'll now have a quick tea break. By 4:20, can I expect you all back? I know you'll be back because it's going to be culturals. If it is a scientific session, you would be late, but as it is cultural, go have a tea break. Come back quickly by 4:20 sharp. Inform all your friends who are outside that sessions are over, they can come back. A very good evening to one and all present here.
Once again, very good evening to one and all present over here. A very good evening to respected dignitaries, esteemed guests, eminent speakers, my dear faculty members and dear student delegates. As we come together today to mark the conclusion of this memorable event, it is both an honor and a privilege to stand before you and introduce the valedictory address. This movement serves as a reflex of all the hard work, dedication, and shared experiences that have brought to us. It is a time to celebrate achievements, acknowledge growth, and look forward to exciting future that lies ahead. The valedictory address is not merely a closing statement, It is a tribute to the journey we have undertaken together. It allows us to express our gratitude for the moments we have shared and the lessons we have learned. We also recognize the bonds that have been forged and the impact that this experience had on each of us.
As we approach the culmination of this enriching today symposium, it's time to lighten the atmosphere and celebrate the success of this event with a touch of cultural brilliance. To add color and vibrance to this valedictory function, I now invite the talented dance team to take the stage and mesmerize us with their special performance. Dear dignitaries, here are our students coming up to showcase the importance of the Indian festivals, and I feast for all of us. Hearty welcome and come on to the dais, dance team.
Really excited to experience various festivals which you people celebrate every year.
Absolutely, it's our honor and I'm happy to hear it. In spite of vast diversity and cultural richness of our country, we celebrate many festivals, some of which are
అజమాయిషీలే
Thank you guys. Your performance really vibrates the environment with the grandeur of the festivals. Let's give them a big round of applause. It's time to turn up the energy and witness an incredible display of talent and grace. Please join me in welcoming our third Family girls for a spectacular dance performance.
थोड़ी फुर्सत भी मेरी जा कभी बाहों को दीजिए, थोड़ी फुर्सत भी मेरी जा कभी बाहों को दीजिए। आज की रात मजा हुस्न का आँखों से लीजिए, आज की रात मजा हुस्न का आँखों से लीजिए। वक्त बर्बाद न बिन बात की बातों में कीजिए, वक्त बर्बाद न बिन बात की बातों में कीजिए। ఈ అల్లరి గాలేమో అల్లుకు పొమ్మందే, మాటలతోటి కాలక్షేపం మానేమంటుంది. కబడ్డీ కబడ్డీ అంటూ గుర్తుకు వచ్చేనా, ఏమంటాయి జీవాలు మళ్ళీ ఎప్పుడో అవకాశం, ఎంచక్కా బాగుంది చుక్కల ఆకాశం. సోసీలారా బాగుందే నీ సహకారం, ముద్దులతో తెరిపేద్దాం నీకు నాకు మధ్యన దూరం. గోదావరి గట్టు మీద రామసులకనే నీ జంట కట్టుకున్న సందమామనే. ఉత్తరం ఉప్పు మీదుంటే దక్షిణం దంచి కొట్టిందే, తూర్పు తుక్కురెట్టిందే, పదర పక్క వేసి పైకి 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 రమ్మందే. వాస్తు బాగుందే బేబీ వాస్తు బాగుందే, కాస్త 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 దోస్తు రమ్మందే. గతంగా వాస్తు బాగుందే బేబీ వాస్తు బాగుందే, కాస్త 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 దోస్తు రమ్మందే. వచ్చుండాయి ఫీలింగ్స్, వచ్చుండాయి ఫీలింగ్స్, వచ్చి వచ్చి చంపేస్తుండాయి ఫీలింగ్స్ ఫీలింగ్స్. వచ్చుండాయి ఫీలింగ్స్, వచ్చుండాయి ఫీలింగ్స్, వచ్చి వచ్చి చంపేస్తుండాయి ఫీలింగ్స్ ఫీలింగ్స్. మల్లిగా వాడితే అమ్ముగడో, కన్నుల తుమ్ముగడో, అంగడి తుమ్మిల నాముగడో, పుంజిరి తుమ్మిగడో, ముల్ల మలర్మణి చెట్టుగడో, తిమ్మడి చంపుగడో, తెందేని తెత్తుగ వండుగడో, తిమ్మిన తుండుగడో.
వసినార జమన్రే పలకదోర జాంపళ్ళ బండి, పూల పంటకి వచ్చి పాల పెట్టి చిక్కల్లో పక్కేసి లాగిస్తానా బండి.
మాస్తో పెట్టే పుట్టే మడతడిపోతే,
అది
ఒంట్లో ఒక్కో నెల తెలికడిపోతే. చిరుతల్లే నువ్వు చేసుకున్న చిరునవ్వే చిందించే నీ చికిత నేనేనట. మెగా 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 మీటర్ నీలో ఉంది, మెగా మీటర్ నువ్వే నాకు లక్కీ మ్యాటర్. కొట్టేద్దాం రా పోస్టర్, మెగా 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 మీటర్ నీలో ఉంది, మెగా మీటర్ నువ్వే నాకు లక్కీ మ్యాటర్. కొట్టేద్దాం రా పోస్టర్.
Wow, what an electrifying performance. A huge round of applause for their energy. It's said that the music suits the mind and it makes rest. Now let's invite G. Priyanka of 5th Family for a wonderful singing performance.
Good evening everyone. I am Priyanka from 5th year family. అస్తమానం నీ లోకమే నామై మరపు చేతనైతే నువ్వే నన్నావు నా నిద్ర కుల్లాస నీ కలలకిచ్చేసా నీ కోసం వయసు వాకిలి కాసారా నా అసలు పోగేసా నీ గుండెకు అచ్చేసా నీ రాకకు రంగం సిద్ధం చేసా ఎందుకు పుట్టిందో పుట్టింది ఏమో నువ్వంటే ముచ్చట పుట్టింది పుట్టాని నీ పిచ్చి పట్టింది నీ పేరు పెట్టింది వయ్యారం ఓని కట్టింది గోరింట పెట్టింది సామికి ముగ్గులు కట్టింది చుట్ట మళ్ళీ చుట్టేస్తాంది చుట్టేస్తాంది చుట్ట మళ్ళీ చుట్టేస్తాంది చుట్ట మళ్ళీ చుట్టేస్తాంది తొంటరి చూపు ఊరికే ఉండదు కాసేపు మత్తుగా మెలేసింది నీ వరాల సిరి హత్తుకోలేవా మరి సరసున చేరే వస్తుగా పెంచానిట 100 కోట్ల సొగసిరి ఆస్తిగా లేసుకో కొసరి కొసరి చేయరా ముద్దుల దాడి ఇష్టమేని సందడి ముద్దడిచ్చి ముట్టేసుకోలేవా సరి చేజారి నీ బంగారు నక్లీసు నా ఒంటికి నచ్చట్లే నీ కౌగిలితో నన్ను సింగారించురా ఏ వెన్నెల జోలాలి నన్ను నిద్ర పుచ్చట్లే నా తిప్పలు కొంచెం ఆలోచించు ఎందుకు పుట్టిందో పుట్టింది ఏమో నువ్వంటే ముచ్చట పుట్టింది పుడతానే నీ పిచ్చి పట్టింది నీ పేరు పెట్టింది వయ్యారం మోని కట్టింది ఊరింట పెట్టింది సామికి ముగ్గులు కట్టింది చుట్ట మళ్ళీ చుట్టేస్తాంది చుట్టేస్తాంది చుట్ట మళ్ళీ చుట్టేస్తాంది చుట్ట మళ్ళీ చుట్టేస్తాంది తొంటరి చూపు ఊరికే ఉండదు కాసేపు Thank you very much.
Thank you, Priyanka. Your voice just filled the auditorium with an elegance. Dance is not just a movement, it's an art, a story, and an emotion of your life. I invite Kaushik from 4th B Pharmacy for a solo dance performance.
to clap. Clap to clap. Boys and girls. పదహారేళ్ల వయసు పడి పడి లేచి మనసు పదహారేళ్ల వయసు పడి పడి లేచి మనసు పట్టుకో పట్టుకో కళ్ళెత్తి పట్టుకో 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 పక్కమేసి కట్టు
పుష్ప అంటే నేషనల్ అనుకుంటారా ఇంటర్నేషనల్.
One man
One voice, one legacy of the King of Pop, Michael Jackson.
Yeah, massa.
Wow, Kaushik, that was a great performance. It just needs a great round of applause. Now we have another spectacular solo dance performance by Ravi from 6th Farm Day. Let's invite him with a great round of applause.
కన్యాకుమారి కనబడద దారి కయ్యాల మారి పడతావే జారి పాతాళం కనిపెట్టేలా ఆకాశం పని పట్టేలా ఊగకే మరి మతిలేని సుందరి
నేను ఇప్పుడు లాగి పెట్టి కొడతాను, మూసుకుని కొట్టించుకోవాలి.
అది నువ్వు కొట్టే బంగారం.
నీ కయ్యికలే కన్ను కొట్టాయా, నడుం లకలకలే కచ్చగట్టాయా. నీ కయ్యికలే కన్ను కొట్టాయా, నడుం లకలకలే కచ్చగట్టాయా. మజాగా మడదేస్తివే పీచు మిఠాయా, అమ్మో పీచు మిఠాయా, అయ్యో పీచు మిఠాయా. మజాగా మడదేస్తివే పీచు మిఠాయా, అమ్మో పీచు మిఠాయా, అయ్యో పీచు మిఠాయా. సోజావో పడకేస్తా రాయ సుబ్బనాతి అమ్మ, వాటంగా పిలిసినదే వాటర్ బాటిల్ మోతే.
పచ్చిమిరపకాయ నేను నీ పంటి కిందికొస్తే, నీ ఎకసకాలు చూస్తే నా సోకు నీకు రాస్తే, నా సోకు నీకు రాస్తే, నా సోకు నీకు రాస్తే, నా సోకు నీకు రాస్తే, నా సోకు నీకు రాస్తే.
అన్న ఒక్కసారి ఆ ఎస్పి బాలసుబ్రహ్మణ్యం గారి సాంగ్.
కాస్త నువ్వు నిన్ను నేను తాకుతుంటే, తాకుతున్న చోట సోపు నిప్పు రేగుతుంటే,
రేగుతున్న చోట భోగి మంట మండుతుంటే, మంట చుట్టుముట్టి కన్నె కొంపలంటుకుంటే,
నరాల్లో లవ్వు గివ్వు జివ్వు పుడుతుంది.
పెదాల్లో నవ్వు కివ్వు పువ్వై పూస్తుంది.
That's a lovely performance, dear Ravi. Thank you so much. We were having speeches and sessions by so many personalities. What do you think about a person who was talking about the subject? He was raising questions, he was making us understand so much about the topic. Now, the same person is going to give a special performance for us. It's my honor to invite Dr. Shishir Nandi sir onto the dais for a special song performance. Of course, he needs a wonderful applause for the initiation. Thank you so much. Hearty welcome to you, sir. The stage is all yours.
Hello. Hello. Echo. Hello. 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 Echo. Today. We are very much happy. I could remember well at the time of my university life when, like you are doing, we have also done. We have studied, playing on the playground, dancing, singing. That's why I would like to sing a song. Kishore Kumar ji, the legend of songs. I am very blind lover of Kishore Kumar. Echo. The meaning of the song is purity, love, and truthfulness. key principle of happy life. Never say goodbye. Say we are all connected by heart. साउंड बढ़ाओ, साउंड बढ़ाओ, साउंड चल चलते चलते मेरे ये गीत याद रखना, कभी अलविदा ना कहना, कभी अलविदा ना कहना। चलते चलते मेरे ये गीत याद रखना, कभी अलविदा ना कहना, कभी अलविदा ना कहना। तोते हंसते स्टॉप प्लीज, स्टॉप शुरू बिगिनिंग प्लीज, सब में से याद रखना, कभी अलविदा ना कहना, कभी अलविदा ना कहना। चलते चलते मेरे ये गीत याद रखना, कभी अलविदा ना कहना, कभी अलविदा ना कहना। तोते हंसते बस यूं तुम गुनगुनाते रहना, कभी अलविदा ना कहना, कभी अलविदा ना कहना। प्यार करते करते हम तुम कहीं खो जाएंगे, इन्हीं बहारों के आंचल में थक के सो जाएंगे। प्यार करते करते हम तुम कहीं खो जाएंगे, इन्हीं बहारों के आंचल में थक के सो जाएंगे। सपनों को फिर भी तुम यूं ही सजाते रहना, कभी अलविदा ना कहना, कभी अलविदा ना कहना। चलते चलते मेरे ये गीत याद रखना, कभी अलविदा ना कहना, कभी अलविदा ना कहना। हमें दिलबर, बिछड़ जाए कहीं हम मगर, हर सुनी से लगे तुम्हें जीवन के ये डगर, बिछड़ा हमें दिलबर, बिछड़ जाए कहीं हम मगर, हर सुनी से लगे तुम्हें जीवन के ये डगर, हम लौट आएंगे तुम यूं ही बुलाते रहना, कभी अलविदा ना कहना, कभी अलविदा ना कहना, चलते चलते मेरे ये गीत याद रखना, कभी अलविदा ना कहना, कभी अलविदा ना कहना। कभी अलविदा ना कहना, say no goodbye. We all are connected by heart. Thank you, thank you.
Thank you so much, sir. That's a wonderful performance and you have changed the atmosphere of the event. और शिशिर दा, मैं कल तक किशोर दा की फैन थी, आज से आपकी फैन बन गई हूँ। तो आपने अभी वादा किया है कि हम बुलाते रहेंगे और आप आते रहेंगे। Give a big round of applause. कभी अलविदा ना कहना शिशिर सर, निर्मला कॉलेज ऑफ फार्मेसी से। Now we have another performance lined up. We'll be moving ahead soon. We have Prasanna from Diploma for Pharmacy for a performance. We'll be moving ahead and soon we'll be continuing with the valedictory event. Thank you, Prasanna. We are coming up to the end of the cultural program of today. So before that, We know we all are patriotic. We love our country. We have respect for the legendary personalities of the nation. So when you love your country, we say Ettara Janda, right? So let's all make the sound of Ettara Janda. Let's call upon the dance team for the same song performance.
నెత్తురు మరిగితే ఎత్తర జెండా సత్తువ ఉరిమితే కొట్టర కొండ నెత్తురు మరిగితే ఎత్తర జెండా సత్తువ ఉరిమితే కొట్టర కొండ జెండా కొండ కత్తి సుత్తి గిట్ట కోత కొమ్ము కోడే
ಒಂದ್ಸಲ ನೀ ಕೋಡೆ ಸಿರಿಗಲ್ಲ ಕೋಡೆ
సిరిసిల్ల కోడే
ಎಲ್ಲ ಎಲ್ಲ ಕೋಡೆ
చైనా కోడే
రత్కల్ల దట్టిది రాయలసీమ కోడే
నెత్తురు మరిగితే ఎత్తర జెండా సత్తువ ఉరిమితే కొట్టర కొండ ఉరుం 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 నెత్తురు మరిగితే ఎత్తర జెండా సత్తువ ఉరిమితే కొట్టర కొండ
రయ్య రయ్య రకతం లేలే మనే
రథము టము గుండెల కెగదన్ననే
కూరరం పిర్రు పిర్రు బిగిసనే అరసింహాసి కటి ముప్పంతా ముగిసనే
ఇప్పుడు కాకుంటే ఇంకెప్పుడు అడాలా
తప్పుల మేళాలు మహా గొప్పగా మొగాల
మూత కూత కొత్త కోట తూట వేట తురుము కోడే
కసిగల్ల కోడే
కలకత్తా కోడే కుచుకల కోడే గుజరాతీ కోడే
కతిలటి కోడే
ఎత్తూరు కోడే
తిరుగేలేనిది తిరునల్వేలి కోడే
నెత్తురు మరిగితే ఎత్తర జెండా సత్తువ ఉరిమితే కొట్టర కొండ
చుట్టర చుట్టూ తలపాగా చుట్టరా పట్టర పట్టు పిడికిలి బిగబట్టరా
చెప్పలు రెండు చరిచి జై కొట్టరా మన ఒక్కొక్కరు కోట్లాది పెట్టురా
చూడర మల్లేస చుడమైనది భరోసా కుమ్మర గణేశ గుడగట్టర కుల్లాస
అస బుస పుట్ట గిట్ట గింజ గుంజ కలుసుకు కోడే పల్లె పంతమున్న కోడే జావి కోడే
రతన్న కోడే
రముదూరి కోడే
పౌరుషాల కోడే
అల్లాసి కోడే
విజయ విహారమే
వీరమరంగ కోడే
Thank you so much, dance team, for reminding us about such the legendary personalities of our country. ప్రకాష్ ఆ చివరికే నాన్న
వాటర్ బాటిల్స్ ఇప్పుడు పెట్టేయడమేగా
పైకి వచ్చి కూర్చుని ఒక తర్వాత ఒకళ్ళే వస్తారు కదా ఎలానో ఎవరు వస్తే వాళ్ళకి ఒకే తర్వాత ఒకే తర్వాత ఇది ఇచ్చేయండి Respected dignitaries, esteemed guests, faculty members, and dear participants, once again a heartfelt welcome to all of you for this valedictory event of the 2 days International Symposium. Over the past 2 days, we have been privileged to witness a confluence of ideas, innovation, and collaboration. As we gather here to conclude this remarkable journey, let's begin the valedictory function by welcoming the esteemed dignitaries who will grace the days with their presence. First and foremost, it's my privilege to invite Reverend Sister Dr. U. Shaurilu Garu, Provincial Superior of CSA, Guntur Province, patron of this symposium and Director of Nirmala College of Pharmacy, onto the dais, please. With a warm round of applause, please welcome Sister onto the dais. Sister, your guidance and leadership have been a source of inspiration for this event for all of us. We are deeply grateful for her constant support.
I take this privilege to invite our esteemed chief guest for today's International Symposium for valedictory, Dr. K. Venkatramana Sir, Principal of ASN College of Pharmacy and President of APTA. Hearty welcome, sir. Acknowledge the honor of your presence and request you to join on to the stage.
Indeed, a great honor next to invite not just my favorite, who has become everyone's favorite here, Professor Dr. Sisir Nandi Sir, the guest of honor of today's event, onto the stage. Global Institute of Pharmaceutical Education and Research, Kashipur, Uttarakhand. Sir, we are delighted to have you here with us today for your countless efforts you have put into this event, for all the energy you have put in. Hearty welcome to you, sir. The next esteemed dignitary I would like to call upon to the dais is the sweet person we have met on the day one, Dr. Nikhil sir, onto the dais, please. The special guest of today's event. Hearty welcome to you, sir. We are very much honored to have you here, and we can say that we will miss you after you leave from our place.
Now I would like to invite Secretary and Correspondent of Nirmala College of Pharmacy and co-patron of this International Symposium, Reverend Sister G. Nirmal Jyoti Garu. I would like to extend a warm invitation to join us onto the stage.
Welcome to you, sister. Now it gives me great pleasure to invite the convener of this symposium, Dr. B. Pamal Reddy sir, whose dedication, vision and leadership have been instrumental in the success of this event. Hearty welcome to you, sir. I kindly request you to take your place on the dais, please. Thank you, sir.
It's time to invite Head of the Department of Pharmaceutics and Organizing Secretary of International Symposium, Dr. Hare Krishna Roy sir, onto the stage.
Thank you, respected dignitaries, for gracing the dais. Your presence has enlightened the shine of this stage this evening. As we have our guests arrived onto the dais, how come we leave them without listening to their messages? it's my privilege to introduce a distinguished personality who has made remarkable contributions to the field of pharmacy education and research, Dr. K. Venkatramana sir, Principal of ASN Pharmacy College, Tenali. Dr. Ramana sir is not only an exemplary leader, but also serves as President of APTI Andhra Pradesh State Branch, guiding the profession with his vision and commitment. His vast experience and dedication to advancing pharmacy education have been an inspiration to many. Sir, we are honored to have you here with us today. May I now kindly request you to share your thoughts and address the gathering. Ladies and gentlemen, let us welcome Dr. K. Venkatramana sir with a big round of applause as he takes stage for his speech.
Thank you, madam. Good evening. Respected Shauroli madam garu and respected Jyoti madam garu and principal, my brother Dr. Amul Reddy garu and other speakers on this dais, and my dear faculty members of Nirmala College of Pharmacy and all over the state of Andhra Pradesh, my dear faculty members, all the colleges attending here, dear students. The 2 days program, International Symposium, the very good speakers from first 2 days and good deliberations is completed, and the students also presented so many scientific presentations. Definitely this program is fruitful and good knowledge you students have gained came from various institutions across the state of Andhra Pradesh, and also outside state students also joined in June. Because this program is associated with the government 2 departments, one is the CSR and another one is the Department of Biotechnology. This type of the programs students are attended, we exchange your views, ideas and thoughts, what is going on and what you have to share to the other students and other speakers, very senior most speakers across the world. Already one sir came from Egypt, another so many members first 2 days came from across the state of different states in India. So this type of the knowledge is very useful to the student community, because those days earlier in first 25 days back, this type of the programs were not attended, because those days is very small programs is conducted different areas, only few programs. Nowadays technology is transfer, technology is well developed and systems also very fast. So definitely student community, everything in your hand across the world. మీ అందరికి కూడా బాగా తెలుసు నాకంటే, మీ అందరూ కూడా ప్రతి వాళ్ళ చేతిలో కూడా ఆండ్రాయిడ్ ఫోన్స్ ఉన్నాయి. ఇప్పుడు అరిచేతిలో ఫోన్ పెట్టుకుంటే ప్రపంచం మొత్తం చూసేయచ్చు. వాట్ ఈస్ గోయింగ్ ఆన్, వాట్ వర్క్ ఈస్ గోయింగ్ ఆన్, వేర్ వీఆర్ స్టాండింగ్, అంతా కూడా మీ చేతిలోనే ఉంటుంది. కాలేజీ ఈజ్ ప్రొవైడింగ్ ద సమ్ ఫెసిలిటీస్, ద అదర్ రిమైనింగ్ సైడ్ యూ హ్యావ్ టు లెర్న్ మోర్ థింగ్స్ you have to attend the different programs like the symposiums, seminars, conferences and presenting the presentations, oral presentation or poster presentations or whatever it may be. మనం చెప్పినప్పుడు తప్పు ఒప్పు ఉండి డెఫినెట్గా నేనైనా కూడా ఫ్రమ్ బిగినింగ్లో తప్పులు చెప్పి ఉండొచ్చు, నాకు తెలియనప్పుడు గ్యారంటీగా 1 or 2 ప్రోగ్రామ్స్కి అటెండ్ అయినప్పుడు డెఫినెట్గా మనము రెక్టిఫై చేసుకుని ఇంకా బాగా చెప్తాకి ఛాన్సెస్ ఉండేవి. సో ఆ విధంగా మనం ఎవరు కూడా ఇక్కడ ఉన్న ప్రతి స్టూడెంట్లో టాలెంట్ అనేది 100% కాదు, 200% ఉంది. కానీ షై ఫీల్ అవుతారు, మనం ఏమైనా తప్పు చెప్తామో, మనం ఏమైనా అనుకుంటారేమో అని. అలాంటిది ఏది అనుకోవద్దు. ఎందుకంటే ఈరోజు నాడు 76% యుఎస్లో ఇండియన్స్ ఉన్నారంటే వాళ్ళు ఈజీగా ఏం వెళ్ళలేదు, నాలెడ్జ్తో ఉన్నారు కాబట్టి అక్కడ సస్టైన్ అవ్వగలిగారు మన వాళ్ళు. ఇప్పటికీ స్టిల్ ఈ నిమిషం వరకు కూడా డెఫినెట్గా మీ అందరికీ కూడా మంచి నాలెడ్జ్ ఉంది. ఆ నాలెడ్జ్ని ప్రాపర్ వేలో చక్కగా గుడ్ పాత్లో తీసుకెళ్ళి మీరు మంచి భారతీయులుగా, భారత పౌరులుగా ఎక్కడున్నా కూడా ప్రపంచంలో ఐఎమ్ ద స్టూడెంట్ ఆఫ్ నిర్మలా కాలేజ్ ఆఫ్ ఫార్మసీ, ఐఎమ్ స్టూడెంట్ ఆఫ్ సో అండ్ సో కాలేజ్, అంతేగాని మీరు కాలేజీకి ఏదో ఇస్తారని కాదు. కాలేజీ ప్రోగ్రాంలు కండక్ట్ చేసేది మీరు బాగా షైన్ అవ్వాలి, నలుగురిలో మీరు నిలబడాలి అనే ఒక మంచి ఉద్దేశంతో మేనేజ్మెంట్ ఎంత కష్టమైనా సరే, రిస్క్ తీసుకునైనా సరే ఈ ప్రోగ్రామ్స్, ఈ టైప్ ఆఫ్ ద ప్రోగ్రామ్స్ కండక్ట్ చేస్తున్నారంటే దాని మీనింగ్ మీకు మంచి ఒక గుడ్ మెసేజ్ ఇవ్వాలి. మా స్టూడెంట్స్ మంచిగా షైన్ అవ్వాలి, మంచి పొజిషన్లో ఉండాలి అనే కోరుకోవటంలో ఏ మేనేజ్మెంటు, ఏ ప్రిన్సిపల్, ఏ టీచరు కూడా వెనక ఉండరు, ముందే ఉంటారు అనే దానికి సంకేతమే ఇట్లాంటి ఇంటర్నేషనల్ సింపోజియమ్స్, డిఫరెంట్ స్పీకర్స్. మనం ఈజిప్ట్ వెళ్ళాము కానీ సార్ ఇక్కడికి రావటం జరిగింది. సో ఎంతో రిస్కు, ఎంతో కాస్ట్లీ, ఎంతో టైము అయినా సరే వారిని ఇక్కడ తెప్పించి మీకు సెమినార్, ఈ ప్రోగ్రామ్ కండక్ట్ చేయించారంటే ఒక మంచి డెలిబరేషన్, ఒక మంచి లెక్చరర్, తప్పనిసరిగా అది మీకు జీవితంలో మలుపులు తిప్పటానికి, మీరు ఒక మంచి పొజిషన్కి వెళ్ళటానికి ఇలాంటి ప్రోగ్రామ్స్ తప్పనిసరిగా ఉపయోగపడతాయి. సో ఒక 1000 రూపాయలు కట్టామో 500 కట్టామో అని ఆలోచించకండి. తప్పనిసరిగా మనీ అనేది శాశ్వతం కాదు. మీరు జీవితంలో ఎదగటమే కాలేజీల యొక్క ముఖ్య ఉద్దేశము, మంచి పొజిషన్లో ఉండటమే. అది మైండ్లో పెట్టుకుని చాలా బాగా ఒక మంచి గుడ్ స్టూడెంట్గా, గుడ్ నాలెడ్జ్తో కాలేజీ నుంచి బయటకు వెళ్ళాలి. ఈ ఫార్మసీ ఫీల్డ్ ఈజ్ ఏ ఎవర్ గ్రీన్ ప్రొఫెషన్. ఏ ప్రొఫెషన్ అనే ఫ్లక్చువేషన్స్ ఉండొచ్చు, కానీ అవర్ ఫార్మసీ ప్రొఫెషన్ ఈజ్ ఏ నో ఫ్లక్చువేషన్స్, ఎవర్ గ్రీన్ ప్రొఫెషన్. ఈ ఎర్త్లో మనిషి భూమి మీద ఉన్నంత వరకే మన ప్రొఫెషను ఇంకా పైకి వెళ్తూనే ఉంటుంది. ఆల్రెడీ ద ఎవిడెన్స్ జస్ట్ 3 years back people all are observed in Corona 2020 and again second time also ఇండియాలో 2 ప్లేసెస్లో, వన్ ఈజ్ ద సీరం ఇన్స్టిట్యూట్ అండ్ వన్ ఈజ్ ద భారత్ బయోటెక్. మన తెలుగు వాళ్ళు, మన రాష్ట్రం వాళ్ళు, భారత్ బయోటెక్ వాళ్ళు కోవాక్సిన్ అనేది వ్యాక్సినేషన్ ఓన్గా తయారు చేశారు. అలాగే సీరం ఇన్స్టిట్యూట్లో ఆ తయారు చేసిన దాంట్లో మన ఫార్మసిస్టులు కూడా భాగస్వాములు అయి ఉన్నారు. మన కాలేజీలో చదవని ఇంకా మన ఫ్రెండ్స్ కాలేజీలు ఎక్కడన్నా ఉన్న విద్యార్థులు వాళ్ళందరూ కూడా ఉన్నారు. అది ఈజీగా ఏమీ రాలేదు. సో ఫార్మసీ ఈజ్ ఎవర్ గ్రీన్ ప్రొఫెషన్ అని అందుకనే. సో డెఫినెట్లీ మై డియర్ స్టూడెంట్స్, టీచర్ గారు తిట్టారనో, ప్రిన్సిపల్ గారు తిట్టారనో, లేకపోతే తల్లిదండ్రులే కోపడ్డారనో మాత్రం పెట్టుకోకండి మనసులో. ఎప్పటికీ మీరు బాగుండాలి అనే కోరుకునే దాంట్లో తల్లిదండ్రుల తర్వాత సెకండ్ నెక్స్ట్ ప్లేస్ ఇమ్మీడియట్లీ టీచర్. అంతే. మిమ్మల్ని కొట్టలేము, కానీ పెద్ద వయసు మీది, మేజర్లు అయిపోయారు, కానీ మీ మీద గట్టిగా మిమ్మల్ని దండించగలిగే హక్కు తల్లిదండ్రుల తర్వాత టీచర్కే ఉంటుంది. You take the role model examples of the Dr. Sarvepalli Radhakrishna, Dr. APJ Abdul Kalam. You take the good personalities. ఎక్కడికో తీసుకెళ్ళిపోయారు ఈ దేశాన్ని. బెస్ట్ ఎగ్జాంపుల్స్. అలాగే ఎక్కడో యుగోస్లేవియాలో పుట్టి ఇండియాలో కలకత్తాలో మదర్ తెరీసా గారు వారు ఇక్కడికి వచ్చి ఇండియాలో సర్వ్ చేశారు, మీ అందరికీ తెలుసు. సో ఏదైనా సరే ఒకటి సాధించాలనుకున్నప్పుడు డెఫినెట్గా ఒకటి వదిలేసుకోవాలి. అలా వదిలేసుకోమంటే అన్నీ కాదు, తప్పనిసరిగా మన గోలు మనం రీచ్ అవ్వాలి. అంటే కొంత కష్టపడాలి. కష్టపడినప్పుడే దాని యొక్క స్వీట్ ఫ్రూట్స్ డెఫినెట్గా మనం తీసుకుంటాము. దానికి ప్రతి విద్యార్థి ట్రై చేయాలి. యాజ్ ఎ టీచర్గా నేను ఈరోజు ఇక్కడికి వచ్చిన ఒక గెస్ట్గానో, ఒక ప్రిన్సిపల్గానో, లేకపోతే ఇంకో హోదాతో చెప్పను. నేను ఇక్కడ స్టేజ్ మీద నుంచున్నా ఒక టీచర్గానే మాట్లాడతా. ఎందుకంటే నేను కూడా క్లాసులు చెప్పున్నా కాబట్టి ఒక టీచర్గానే మీకు చెప్తున్నాను. తప్పనిసరిగా మీరందరూ బాగుండాలని కోరుకునే దాంట్లో ప్రథమ స్థానంలో మొదటగా నేనుంటా. అది మీరు ఎక్కడున్నా, అరే మాస్టర్ గారు మా ఒక కాలేజీకి వెళ్ళామే, ఎంతో చెప్పారే మన కోసం, మనం బాగుండాలని, దానికే మీరు ట్రై చేయాలి. ఆల్ ద బెస్ట్ ఫర్ కమింగ్ 4th years బీఫార్మసీ ఎగ్జామ్స్ ఉన్నాయి విత్ ఇన్ షార్ట్ వెల్, అవుట్ గోయింగ్ బ్యాచెస్, అలాగే అవుట్ గోయింగ్ బ్యాచెస్ ఆఫ్ ద ఫార్మ్ డి స్టూడెంట్స్ అందరికీ కూడా ఆల్ ద బెస్ట్. ఆల్ కాలేజ్ స్టూడెంట్స్ ఉన్నారు ఇక్కడ, అందరికీ ఆల్ ద బెస్ట్. బాగా చదవాలి, చదివి కాలేజీలకి మంచి పేరు తండి. మీరు ఏ దేశంలో ఉన్నా, ఈ ప్రపంచంలో ఎక్కడ ఉన్నా టీచర్ని మర్చిపోండి. ఎప్పుడు వచ్చినా ఈ కాలేజీలో ఈ స్టేజ్ నుంచి మీకు ఏ కాలేజీలో చదివినా ఆ కాలేజీ స్టేజ్, ఆ కాలేజీ మీది. ఎప్పుడైనా మీరు రావచ్చు, ఎప్పుడైనా మీరు వెళ్ళచ్చు. థ్యాంక్ యూ ఫర్ గివింగ్ దిస్ ఆపర్చునిటీ మేనేజ్మెంట్ అండ్ మై బ్రదర్ పాముల్ రెడ్డి గారికి అండ్ అవర్ faculty members and non-teaching staff of Nirmala College and dear students, thank you for giving this opportunity. Thank you one and all.
Thank you, sir, for your valuable words. It's time to invite our guest of honor. It is now time for us to hear from someone whose presence today truly adds immense value to this occasion. We are deeply honored to have you with us, sir, our esteemed guest of honor, Professor Sisir Nandi. It is great pleasure to have you here, sir. We invite you on to give an invaluable message with us.
Wish you very good afternoon. It's just before the evening, so wish you very good evening as well. It's my privilege to be one of the keynote speakers of this international symposium on sustainable drug design and nanoparticle development, quantum and computational perspectives, organized by Nirmala College of Pharmacy. It has been really a very brainstorming sessions contributed by multidisciplinary research scientists, young students, scholars, teachers, academicians, talent persons from industries. We have support from all of us. It's our great opportunity to learn from all of you. I show my deep respect to President Ma'am, President Sister Ma'am, Director Sister Ma'am, Jyoti Ma'am. I really thankful to my colleague, respected Principal Sir, Professor Reddy, my affectionate dear brother Hare Krishna Rai. I show my deep respect to very senior teachers and very great administrator and principal, Professor Dr. Venkata Raman sir. Thank you very much for your support. If we see from the beginning of the session from yesterday, Professor Dekil emphasized on application of multidisciplinary research activities, applying multidisciplinary means integration of multidisciplinary sciences and technology to explore the sustainability for the human mankind and economy and ecology, environmental and protections. Thank you very much, sir. We have learnt a lot, particularly, from the Dekil sir lecture, we have learned that the fish which is cultivated in our pond, in our river, if fish is under disease, there is no treatment methodology in India. So from this valedictory session, we would like to send our message to our corresponding government department, state government and central government to make curriculum for designing of such courses so that this kind of treatment facilities must be in our country. Thank you very much, sir. Myself, I have presented the screening of chemo preventives to combat the colorectal disorders, which is very common to our family, utilizing ligand-based drug design, structure-based drug design, chemobioinformatics, computational modeling that can reduces the drug design development cost and reduces the time, labor, manpower. Dr. Ashmita ma'am from Kalyani University, West Bengal, very, very young and dynamic researchers, teachers. She developed nanoparticles utilizing natural components like piperine, curcumin to combat the lifestyle disorders like, diabetes, cancer, Alzheimer's, which are very common nowadays. Very important. The young scientist, very dynamic, Dr. Amit Rai, he screened bioactivity of natural components like hesperidine to combat the leishmania topoisomerase, utilizing different instruments, technologies. Professor Nagaraju sir explained the significance of new drug delivery system to improve the drug safety and efficacy. He has given a very constructive future perspective on the discovery of nanorobotics to target the specific disease cells like cancer, diabetes through design and development of nanosensors. We have learned. Dr. Srinivasan sir from IICT Hyderabad, very young and dynamic scientist, has given a direction how to design the vaccine utilizing mRNA technologies. I have a lot of discussions. We have a lot of discussions with Dr. Srinivasan sir, and it is possible through mRNA therapy to increase the lifespan of a person. It is very possible. So our science and technologies, where it is going on due to application of our brain. Dr. Jitendra sir, Dr. Raghavendra sir discussed the formulation of nano drug utilizing natural resources, natural components. Dr. Srinivas sir from USA, very senior scientist, he delivered on bone grafting technology to minimize the very common issues of human to get benefit of the society and human health. He has developed many rapid detection kit for early diagnosis of the diseases, which is very, very important nowadays. If disease is early diagnosed, then the treatment can be started taking particular personalized medicines to minimize the problem. Dr. Nevo from Serbia, he has summarized the applications of in silico pharmacophore modeling, structure-based, ligand-based drug design, Dr. Uttam sir has delivered on impurity detection and phytocomponent separation and detection through LCMS, liquid chromatography mass spectrometer. And the most important part and beauty of the symposium is participation of young researchers like you, young PhD scholars, young students. You are the main part of the symposium. Your oral presentations, your poster presentations really motivate us. Our lecture motivate you more than we have been motivated by you based on your fertile brain, based on your talented brain. Don't keep yourself within the four wall, within the looking mobile. No, don't waste a single time, because Android mobile with internet that kill the brain memory, incorporating silent diseases like internet addiction disorder, internet gaming disorder, artificial neural network of the brain is silently killed. So youth talent, you are the main part of our society. You are the spinal cord of our country. You are the spinal cord of the institution and family. You motivate, you wake up, and you produce, you generate talent, you generate new science, new technologies. Really, I am so surprised. Your energy, your presentations, really, we really appreciate you. I would like to give one important message that youth, early in the morning you wake up, activate yourself before coming into the institution through yoga, do exercise, take breakfast, come at the institute. Through discipline, you attend all your classes, interact with all of your teachers. After coming back to home from the institute, you open your books, you study, you pay at least 2 hours, taking equivalent importance of each and every subject, what you have been taught by your teacher at the class. You will be tough in the university, you will be tough in our country, everybody. That must be. And you have a dream. What dream you have? After dinner, when you go to sleep, you have a dream. Next day, I have class. Go to the class, go to the class, go to the class with discipline. That should be key principle of your life, because time is most important. If you lost this time never come back in your life. Very crucial for you. For me, I am working in Uttarakhand, north part of the India. We have computer-aided drug design laboratory, well-established laboratory. We have many softwares installed. We are giving training on ligand-based drug design, structure-based drug design, biochemical activity predictions, compound synthesis, structure-based modeling, vaccine designs. We have modules, expertise. Why would you not opt that opportunity? It is open to all for you. You come at our institute, learn. Even I can give you training through online session, which is very beautiful. I have some modules which are free, which don't know you. If you learn through online session, from Uttarakhand, I will install at your computer at this institute through desktop sharing, computer internet technologies. I will teach from there, you will learn from here. We shall be in touch by telephone, through email, through WhatsApp, through call. Then you will be expert. You do something. At your 4th year, you have project work. What you are doing? Teacher is assigning you the project title. Don't make any copy paste. You have to learn how to do the literature review, how to do the literature search. That is sufficient. You target one component, you target one topic, one component of the topic, learn. Then you explore in the master degree. Then you explore it more explicitly in your doctoral studies. You know, we have a great teachers. We have great teachers like C.V. Raman. We have great teachers like APJ Abdul Kalam, who always says, if you salute your job, no need to salute others. But if you pollute your job, you have to salute everybody. Why? I would like to build myself. I would like to study. I devote myself through study and hard work. So solely devotion through study and hard work, that should be key principle in your life. Print it in the brain, print it in the heart. This is the temple of Ma Saraswati, goddess of education. We are blessed by Ma Saraswati. In this temple, eminent academicians come from different parts of the globe in this symposium. Professor Dikhil came from Egypt. Very young, talented scientist. What idea he has shared. It's great opportunity for us. Great opportunity. So this opportunity, you gulf, you opt, you catch, you snatch from us. It is only possible through collaboration, connect towards the global collaboration, integration of science, technology, multidisciplinary research, technologies, sciences, academia, industry, collaborations. So this platform is open to all for the collaborations. If you have idea, which I don't know, you share. We collaborate to generate new thing. You know, through study, if you know how to maintain your family, how to maintain your health, your family will be benefited. If each and family is benefited, our country will be strong. We have a great teacher, Swami Vivekananda ji. You know, education is the manifestation of all perfection already in man, written by Swami Vivekananda ji. So, my dear students, wake up, proceed till goal is achieved. Thank you very much.
Thank you so much, Sishit sir, for your wonderful message to all our students. We all want to know the secret of your energy. Like, you have been so energetic from day one. We have been seeing your energy. Please tell us your secret before you leave, sir. Thank you so much. Give a big round of applause for such a wonderful personality, for a thought-provoking message for all of you. It's with great honor and respect, and now invite Reverend Sister Dr. U. Shaurulu Garu, the Provincial Superior of CSA Guntur Province and the patron of this symposium, to deliver a message to all the students. Hearty welcome to you, sister.
A joyous good evening to one and all. Esteemed guests on the dais, of the dais, honored speakers, valued principal, secretary correspondent, organizing secretary, all the teaching and non-teaching staff of the host college. I am very much pleased to stand before you just to have the feedback, and on behalf of our Superior General, Sister Japamala Vatti, and on behalf of me, as the President and Director of this college, I am very happy to share some of the views and faculty members from various institutions, all the principals, and all those who have present here, dear ever-loving students. I say ever-loving students. I am a student, always a student, to learn something new. And I love students, especially those who learn and those who are aspired to do something special in their life. And dear guests on the days and of the days, and I take this opportunity to thank each and everyone for taking part in this 2 days program on sustainable drug design, nanoparticle development, and quantum computation perspective. And before I thank each and everyone, we need to know the spirit of hard work behind. The hard work that all the speakers had in their lives to come up to this stage. And the hard work, how much the faculty members have been having. And the hard work that all the participants and privileged students are putting in. It's not a comparison, my dear students, but we need to know the spirit of hard work behind to come to one stage and to boldly stand on the dais to put forth the knowledge before the audience. And today, I would like to quote, "The heights by great men reached and kept were not attained by sudden flight, but they, while their companions slept, were toiling upward in the right." So it's not one day or 1 year, it's not some years, but the learning is a lifelong process. To find out any drug design, to design for the health care, it's a very, very hard work that we put in. So my dear, as we take up this quote, to go to the heights in everyone's life, especially the participants and all the students who have presented today and taking part in these 2 days, have taken part in this 2 days program. And this quote reminds us that the great need of maintaining the spirit of hard work. To come to one stage, everyone need to work hard. Working hard doesn't mean that simply sitting and learning. No. We are in the modern technological world. As we are in the modern technological world, you are engaged much more with the modern technology than learning. You are depending on the modern technology than perceiving or pursuing the new subject by your own. You might be referring, but that referring modern technology may help you to some extent. But your self-learning through books, through references, and through all that is kept for you to learn is much more important to become to one stage, to stand on the stage. So my dear participants, and it's the high time for you to think over all the topics that were presented and that the well-versed speakers have spoken, and it's your time to take something of their knowledge that they have expertise. So you take that to your heart, to your mind, and to put into practice in your everyday life so that you can stand firm. And this is the age to learn. This is the life to enjoy the subject. Since you have taken the life sciences, it's nothing but pharmaceutical sciences. To learn or to become a great pharmacist in your own subject, you need to put your interest, you have to be very enthusiastic, and you need to work hard to fulfill the target that you put forth. Without target, without a goal, without a good idea, nothing will take place. So my dear participants, so it is not something that comes too naturally. No. Naturally it may come, but without your efforts, nothing will happen. So I don't want to take much of your time, but as a management member, I would take this opportunity, I take this chance to express my gratitude to the speakers. And it's my privilege to stand before you again to point out the subject that you have put in and the theme of the today's program, Sustainable Drug Design and Nanoparticle Development, Quantum Computational Perspective, is of immense significance, and yesterday's and today's presentations and discussions have greatly enhanced your knowledge and our understanding of this cutting-edge field. So we need to understand, they have put something before you, and it is you who have to sharpen your knowledge. A special acknowledgement to our distinguished speakers who have generously shared their time, expertise, and insights with our students and all the participants and the faculty members. Your thought-provoking discussions have provided a deeper understanding of how quantum computational techniques are revitalized or revolutionizing sustainable drug design and nanoparticle development. We deeply value your ability to simplify complex concepts, making them accessible and engaging for all participants. Your presence and contributions have inspired researchers, students, and professionals to explore innovative, creative solutions for a sustainable future in pharmaceutical science. A warm thanks to you, my dear students, and all the researchers, the participants of today's program, and the professionals too, to present your studies, findings, and perspectives that have taken place. Your dedication to scientific exploration and knowledge sharing has been evident through the high quality research and presentations development. Each presentation added significant value by addressing emerging needs, challenges, and opportunities in sustainable drug design and nanoparticle development. The insights shared have not only expanded our knowledge, but also paved the way for future research and collaboration in this evolving domain. The enthusiasm and active participation of our students have been truly admirable. Your curiosity and engagement in discussions demonstrates to a promising future for scientific and advancement in this field of pharmacy. The questions, insights, and innovative ideas shared by students reflect your deep interest in quantum computational applications in pharmaceutical science. We appreciate your eagerness to learn something new every day and contribute to the society and think critically about the sustainability aspects of drug development in day-to-day life aspects. Behind every successful event, there is a dedicated teamwork which has been taken place in our College of Pharmacy. And they have been working tirelessly to ensure smooth execution and impeccable discussions. My sincere gratitude to the organizing committees that includes NCPA, Nirmala College of Pharmacy, Secretary Correspondent, Sister Nirmala Jyoti, Principal, Dr. Pamal Reddy Garu, and Roy Sir, Dr. Roy, organizing secretary and co-organizing secretaries, all the MCs and various committees of food, reception, decoration, cultural and media and press, electronic media and press, and all those who have involved, especially the students who have performed the cultural program yesterday and today. My sincere and joyful appreciation, and it's really worthwhile to praise them and to take into account the services that they have rendered all these days and to prepare a pavement for yesterday's and today's program. Your commitment to ensuring seamless arrangements, inviting distinguished speakers, and fostering an engaging environment deserves high praise to you, sir. The success of this event is a testament to your hard work, dedication, and attention to deal with each and every aspect that took place. This gathering has not only served as an academic and research proportion or platform, but also as an inspiration to work towards more sustainable and efficient pharmaceutical development. The discussions have highlighted the potential of quantum computational techniques in transforming drug discovery and nanoparticle research. The event has opened doors for future collaborations, networking, and interdisciplinary research among professionals, students, and researchers. By bringing together scientific minds from various backgrounds, this event has strengthened the continuous for informative solutions and sustainable advancement in the industry too. As we come to an end of this event, let us take forward the knowledge, ideas, and inspirations shared in these 2 days and apply them in our respective fields. I repeat, as we come to the end of this event, let us take forward the knowledge, ideas, and inspirations shared in these 2 days and apply them in our respective fields. We encourage all participants to continue engaging in meaningful discussions, research, and collaborative efforts to drive progress in sustainable drug development and nanoparticle development. The importance of sustainability and computational advancement in pharmaceutical sciences cannot be overstated, and we are confident that the insights gained today will shape the future of tomorrow. The insights you gained today will shape the future of your field. Once again, we extend our deepest appreciation to the eminent speakers, researchers, students, all the privileged participants, and organizing committees for making this event a remarkable success. Your contributions have made a significant impact, and we look forward to more such enlightening programs and discussions and groundbreaking research in the future. May God bless you. Take these research speakers into consideration to come up in your life. Thank you.
Thank you very much, sister, for your warm and heartfelt greetings. Heartfelt thank you to everyone. We are always honored to have you here all the time for your support you extend towards us. Now it's time to recognize the students who have come all the way from various colleges and our host college who have participated in various oral and poster sessions. Now I hand over the session to the scientific committee member, Ms. Swati Madam, to take care about the prize distribution ceremony.
So good evening, one and all. Myself, Swati, working as Associate Professor in the Department of Pharmaceutics, acting as a scientific committee member for this international symposium under the scientific coordinator of Dr. Faizan Ali Sir, HOD of Pharmacy Practice and Pharmacology. In the international conference, I want to give a glimpse of presentations. Total, we have received 400 presentations from various colleges in oral, poster and e-poster categories of different departments: pharmaceutics and regulatory affairs, pharmacy practice, pharmacology, pharmaceutical analysis, and pharmaceutical chemistry. So, from the pharmaceutical analysis, we got 26 abstracts, in chemistry 35, in pharmaceutics and regulatory affairs, we received around 133 presentations. In pharmacy practice, we received 98 presentations. In pharmacology, we received 53 presentations around. So we have conducted a two-day international symposium for the presentation in the scientific committee. So in this regard, I take the privilege to announce the prize winners of oral and poster categories for these 5 departments. So I would like to invite our scientific coordinator, Dr. Faizan Ali Sir, if available, join the session, please. So in this regard, we are request the guest of honor, Dr. C.C. Nandi Sir, to come forward to give away the prizes. I request all the participants, please be attentive. If any participant is not available, the faculty who are taking care of them, please come and take the certificate and the prize. So first, in the Department of Analysis, under poster presentations, the first prize goes to Mai Muniza from VV Institute of Pharmaceutical Sciences. Second prize, Meenakshi Komaraju, KVSR Siddhartha. Third prize, Suresh Babu Virla, Nirmala College. Muhammad Muniza, VV Institute of Pharmaceutical Sciences. Meenakshi Komaraju, KVSR Siddhartha, and Suresh Babu Virla. If anybody is not here, the classmates can come and take the prize. Please, we are running out of the time, so any one of them can come and take the prize. Suresh Babu Virla. Anybody come can? In oral, Dr. E. Pravalika from ANU. Dr. E. Pravalika from ANU. SSV from CHIPS, Raviteja Killana from Nirmala. From oral presentations, Dr. E. Pravalika from ANU, SSV Chebrol Hanamaya Institute of Pharmaceutical Sciences, Raviteja Killana from Nirmala. prize Raviteja Killana, Nirmala College. All the prize winners, please wait. After that, we have a photo session for all the prize winners in all departments. So, please stay here after taking your prizes. Thank you, sir. And next, we request our chief guest of the session, Dr. K. Venkatramana, sir, to give away the prizes. I also request our principal, sir, and correspondent sister to accompany. And also I invite Reverend Sister Dr. U. Shaurili Garu, President CSA, Patron of International Symposium. Okay, Pharmaceutical Chemistry, Oral first prize. Oral first prize, D. Abhilasha from Hindu College. Second prize, B. Pujita from Vijay Institute of Pharmaceutical Sciences. Third, Raga Likita from Malineri Perumalla Pharmacy College. I again repeat, Abhilasha, B. Pujita and Raga Likita in oral pharmaceutical chemistry. Of Malineni College, please come and collect V. Raga Likita Award. Anyone from Malineni Perumalla can come and collect V. Raga Likita. Okay, next we move on to the poster. V. Rajashekar, School of Pharmacy, Anurag. V. Rajashekar, School of Pharmacy, Anurag, got the first prize. Second prize, G. Beulah, MITS College of Pharmacy, Suryapet. Third prize, Chunduri Pujita from CHIPS. I again repeat, first prize, V. Rajashekar, School of Pharmacy, Anurag. Anybody from Anurag College. Second prize, Beulah from MITS College. Third prize, Chunduri Pujita from CHIPS. If the persons are not available, please, the faculty representative, please come and take the award. Thank you, sir. Please take the chair. Next, we request Reverend Sister Dr. U. Shaurili Garu, President CSA, Patron of International Symposium, to give away the awards in the pharmaceutical department, pharmaceutics department. So, sister, please come forward to give away the prizes. Poster, Pharmaceutics and Regulatory Affairs. Poster, Viresh from Nirmala College of Pharmacy. I also request Pamuledi sir to join the prize distribution. Next, Kiranmai Kesam Setti, ASN from ASN. Satya Sirisha from ASN. Satya Sirisha from ASN. Okay, next in e-poster, Kirtana Ramireddy from KVSR Siddhartha. Kirtana Ramireddy, Gollampudi Anjali from Hindu, Mustafa from AM Reddy. Mustafa from AM Reddy. Kirtana Ramireddy, Gollapudi Anjali, Mustafa from AM Reddy. Next, oral, Naureen Syed from St. Mary Group of Institutions. I request for the oral presentations, Arshia Naureen Syed from St. Mary Group of Institutions, M. Jhansi Rani from Sri Padmavati Mahila Vishwavidyalayam. Next, Amzad Bai from Nimra College. Padmavati Mahila Vishwavidyalayam, Jhansi Rani. Amzad Bai from Nimra College of Pharmacy. Thank you, sister. Okay, now I request our organizing secretary of International Symposium, Dr. Hare Krishna Roy sir, for the prize distribution, and our Padmavati ma'am, HOD of Pharmaceutical Chemistry, to come join for the prize distribution in the Department of Pharmacology. So, in poster, Chukka Unmilaran, Chips College of Pharmacy. Second prize, K. Kirti, ASN College of Pharmacy. Third prize, Gayatri Priya from AM Reddy College of Pharmacy. Also request our eminent speaker, Dr. Muhammad Dikhil sir. Sir, I request to join the session, please. Pharmacology oral, first prize, P. Kanchana, Hindu College of Pharmacy. Second prize, Dundi Leelavati, St. Mary College of Pharmacy. Third prize, Avinash from Sims College of Pharmacy. First prize, P. Kanchana. Second prize, Leelavati, St. Mary's. Third prize, Avinash from Sims.
I request Sister Sirisha Rani to come on to the dais, please. Sister from principal from CBSE Nirmala School. As a part of her PhD work, she has been a special presentee here today. Sister, congratulations to you. Thank you for your presence, sister. We are pleased to have you here. Thank you so much.
Thank you, Padmavati ma'am. Thank you, Roy sir. I request Asmita ma'am and Amit sir, please join for the prize distribution. So, Amit Roy sir, please join for the prize distribution, sir. So, kindly request. And And I also request Sister Agatha Garu. Pharmacy Practice Department. Oral presentations. L. Maunika from Aditya Pharmacy College, Kakinada. Second prize, Martha Grace from Vikas Institute. Third prize, Nehasri from Nirmala. Third prize, Nehasri from Nirmala College. Second prize, Martha Grace from Vikas Institute. First prize, Aditya Pharmacy College, Kakinada. Next, pharmacy practice poster presentations. First prize, Shaheen from Nirmala College of Pharmacy. Hemant Kumar from Vikas Institute of Pharmaceutical Sciences. Third prize, B. Bhargav from SIMS College of Pharmacy. Bhargav from SIMS. Shaheen got the first prize. Second, Hemant Kumar. Third, Bhargav. I request Dr. Swapna ma'am to come and join the prize distribution. I request Dr. G. Swapna ma'am and Dr. Naresh sir to come join for the prize distribution. Thank you Asmita ma'am and Amit Rao sir. Thank you. I request Raga Likita from Malineni. I request Raga Likita. So please anyone, Raga Likita in the Department of Pharmaceutical Chemistry from Malineni College. And also Reshma from same Malineni College. Madam, I request Asha ma'am to come forward for the prize distribution. Nuncho ingokal. Thank you, sir. Thank you, madam. And thank you all for giving this privilege. And the session is then handed over to the hosting people.
Congratulations to all the participants and thank you for all the remaining participants for your time and efforts you have put in. We encourage you and want to see you in future events as well. Now it's time to recognize the institutes who have supported us through this event. As a gesture of appreciation for the invaluable support extended by our esteemed institutions, we would like to present a token of gratitude to those institutions. In that list, first of all, we would like to invite the CHIPS College faculty and student representative to come on to the dais, please, to receive the token of appreciation from the college. Representative faculty member and any student representative to join the stage from CHIPS College. I invite President Sister and chief guest of today's event and guest of honor to give away special appreciations to the institutions who have sent a good number of students here today for the support they extended. Thank you very much, CHIPS College, for being here with us, followed by SIMS College. SIMS College, Prashanti Evangeline ma'am and co-faculty, please come and join the dais. Madhavi ma'am, please come on to the dais. We request you to bring your students as well. Prashanti ma'am, Madhavi ma'am, along with your student volunteers. Thank you so much, SIMS College, for the support you extended. Followed by Vikas College. Vikas Rajamandri, faculty member and student volunteers as well from the institution. Thank you, Vikas College, for being here with us today. I request all the students to stay back. Please do not leave for a short while. St. Mary's, the next institution who has given support for us is St. Mary's. I request the faculty member and the student coordinator. Thank you, Vikas College. We hope you all will convey our wishes and regards to your managements. Next in line we have one more, the best supporting institute for us, St. Mary's. St. Mary's College, Guntur. We request the faculty member representative. Followed by Hindu College. We have the faculty members from Hindu College of Pharmacy, Guntur. These are the appreciations for the support they extended towards us for the success of this symposium. Next, I request ASN College of Pharmacy. ASN College of Pharmacy. Faculty and student representatives, please come on to the dais. Followed by ANU. We request the ASN College faculty members and students. Thank you so much. Thank you to the bunch of students, those who are here from ASN. Thank you. KVSR Siddhartha. We have ANU in line. After ANU, we have KVSR Siddhartha. We request the faculty members and the student representatives to come on to the dais. Acharya Nagarjuna University is waiting for us. Thank you so much. We have the representatives from Acharya Nagarjuna University coming up. University College of Pharmaceutical Sciences. Next we have KVSR Siddhartha. After that, I would like to call upon Malineni College. Representatives from Malineni. Last but not least, we have Nimra College of Pharmacy, Ibrahimpatnam. Representative from Nimra. Malineni, if you are available, please come on to the dais. We have Nimra College of Pharmacy for their support here. We have one more institute, St. Xavier's. St. Xavier's, please come on to the dais. St. Xavier's, thank you so much. A heartfelt thank you to all these institutions who are behind the success of this event, all the managements, the faculty members and the students who came long away from long distance. That's the spirit of Nirmalites who are here to encourage the outside institutions who are here. It's a very privilege for us to honor and express gratitude to the chief guest of today's event, Dr. Venkat Ramana sir, who's here all the way, who's been waiting long since evening. Sir, thank you for your extensive patience you had during our valedictory event. I request all the dignitaries on the dais to join in felicitating our chief guest today. Give a big round of applause for our wonderful chief guest who's been waiting for us today. Thank you so much, sir.
It's time to felicitate our guest of honor, Professor Sisir Nandi. I request all the dignitaries join in the felicitation ceremony.
Now I request our beloved patron of this symposium, Reverend Sister Dr. U. Shaurila Garu, to accept the token of gratitude. Sister, thank you for all the support you've extended. You're the backbone of the institution for being with us for the successful conduct of this event. Thank you, sister. I request the co-patron of this event, Reverend Sister Nirmala Jyoti Garu, to take the chair, please. Thank you, sister, for your unwavering support for the conduct of this event. I request the convener of the event, Professor B. Pamul Reddy sir, to accept the token of gratitude for your unwavering, extensive support you have provided, sir. Your guidance has made this event possible. Thank you, sir, and congratulations for the successful conduct.
Time to express our deepest gratitude and respect to our HOD of Pharmaceutics and Organizing Secretary of International Symposium, Dr. Hare Krishna Roy, sir.
I request all the faculty members to come backstage, please, as we are getting late. Now the appreciations will be given to the LOC members, the local organizing committee. So I request all our staff members to get up from your seats and be backstage immediately. Please wait at the backstage. We'll be calling up the names. We have Professor Dikhil sir here, who has been very sweet with us, who has been spending his time and energy with us since 3 days. Thank you so much, Mohammed sir.
Dr. S. Padmavati ma'am. Dr. G. Swapna madam. Dr. Faizan Ali sir.
I request all the Nirmala faculty to come backstage immediately. Dr. Swapna madam, Faizan Ali sir.
Dr. B. Lakshmi sir. Dr. U. Spandana madam. Dr. K. Saujanya madam. Dr. Asha Begum. Dr. R. Naresh Babu sir. Mrs. R. Ratna Manjula ma'am. Mrs. M. Preshanti madam. Mrs. Swati madam. Dr. R. Narasimha Naik sir. Mrs. G. Sai Sri Lakshmi madam.
accept the token of appreciation. Mrs. G. Sai Sri Lakshmi. Mr. V. George Babu. Sister Veena. Miss Pavani. Krishnaveni Madam. Pavani. Jyoti Madam. Jyoti Madam, followed by Lakshmi Shanta Madam. Lakshmi Shanta, Mrs. M. Shanti. Mrs. M. Shanti, Mrs. I. Supriya. Miss D. Pramila. We have here Mrs. M. Shanti, I. Supriya, Pramila, Dr. Joel. Dr. Joel. We have Pramila, Miss Pramila. Maharshi, Dr. Maharshi sir. We have Dr. Joel. Dr. Maharshi sir, followed by Miss Parvati. Dr. Pranita. Miss Parvati, please. Miss Swarupa Rani. Deepthi Nancy. Bhargav Gopi sir. Bhargav Gopi sir. Mrs. Tejashruti. Mrs. Tejashruti. Mr. Subani. Mrs. Vijay Kumari. We have Mrs. Reshma Madam here. I request all the faculty members to stay back. A clear instruction that we should have a group photo at the end after the session. I request all the faculty members to be available near the stage. I request all of you to keep standing for national anthem.
जनगण जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे